Mangalore is known as an education hub. Many students from different places come to Mangalore just for study purposes. So just take a round around a famous private research and skill-focused university, that is Srinivas University.
International Conference. We hope yesterday's sessions enriched our perspectives, sparked meaningful discussions, and set the perfect tone for another inspiring day ahead. As we step into day 2, we carry forward the same enthusiasm, curiosity, and academic spirit that define the gathering.
As we begin the first scientific session of the day, let us carry forward the spirit of inquiry and excellence that defines this conference. Our first session is titled Gastrointestinal Multiomics Identify Microbial Drivers of Polyposis. The topic will be delivered by renowned expert Dr. Shahid Umar. To chair this session, we are honored to have two distinguished academicians, Dr. Shiran Shetty and Dr. Krishna Sharan. Requesting Dr. Anand Kumar, Professor, Division of Research, to do the rapporteuring for the session. I request Ms. Rashmi to introduce our chairpersons.
Dr. Srinivas is a professor and unit head, Department of Gastroenterology and Hepatology, Kasturba Medical College, Manipal Academy of Higher Education. He is also a Deputy Medical Superintendent, Kasturba Hospital, Manipal. He holds an MBBS from Bangalore Medical College, Rajiv Gandhi University of Health Science, Bangalore, an MD in General Medicine, Rajiv Gandhi University of Health Science, Bangalore, and an DM in Gastroenterology, Dr. MGR Medical University, Chennai. Dr. Srinivas Shetty is the Fellow of Royal College of Physicians, Edinburgh. He has received 2 ICMR grants. He has a prolific publication record of nearly 85 research articles in reputed international journals. We also have with us Dr. Krishna Sharan, who is a professor and HOD, Department of Radiotherapy, Shema, Nitte University. Dr. Sharan has pursued MBBS from KMC Mangalore and MD in Radiotherapy from KMC Manipal. His areas of interests are head and neck malignancies, gynecological malignancies, and brachytherapy. He has played an excellent supportive role in research by presenting 84 publications in his domain. With this brief introduction, I welcome you both, sirs.
Very good morning and thank you for the kind introduction. I think we are heading for an exciting day today. I think the first topic is multiomics approach to identify microbial driver of polyposis, which will be delivered by Dr. Shahid Umar, who is a renowned scientist, professor of surgery and the vice chair of research in the Department of Surgery at the University of Kansas Medical Center. He has built a significant academic and research career with his background of Ph.D. in biochemistry from Lucknow, MS in biochemistry from Aligarh Muslim University, and also is specialist in gastroenterology at the Kansas Medical Center, and he's been a faculty since 2001. And I think his focus is basically on entering pathogen in cell proliferation, cellular transformation, and neoplasia. He explores how gut microbiome alteration influences in cancer development and also the shift in resistance. He is engaged in developing newer therapeutics, including the brain probiotic approaches to strengthen the immune system. Through his extensive work, I think he is the right man to talk on this topic. So we welcome you, sir. Sir, you're on. Morning, sir.
Mr. Chair, Vice Chair of Research Department of Surgery. I would like to thank the organizers for giving me an opportunity to tell you a little bit about one of the projects in the lab. My apologies, I couldn't be there in person. But it's definitely a pleasure to see you and see everyone virtually. So, with recent bursts in multiomics approaches, I thought this would be a great distinct microbiomes on each body side, diaphragm, skin, and mucosa, for example. But then there are other areas such as liver and pancreas where not much of bacterial flora is found. However, they do receive metabolites through systemic means. The human gut is home to about 100 billion bacteria. There's actually almost 1 trillion bacteria in the colon itself. And most microbes have two major phyla belonging to Prometheus and Bacteroides. And that actually is the composition of the core microbiome, which is already 18 species. Why do we need them? Well, the host provides immunity, and then the intestinal mucus provides a source of nutrition. But bacteria segregate by fermenting the digestible carbohydrates, biotransforming drugs. There is a lot of vitamins and successful, they metabolize drugs, but more importantly, they educate our immune system. And so, if you look at the structure of the human intestinal microbiome across the life cycle, there is a lot of variability when the child is born. And from that to 2 years, there is the microbiome stabilizes. And then by the adult age, we can have a composition which remains small unless it was part 18, for example. There is a period of extreme diversity when women are pregnant, and that's a good thing because it helps them navigate the pregnancy through changes in various metabolic activity. So what happens when things go wrong, for example, right? So if you see here on the left, there is a long colon, whereas on the right there is an inflamed colon. So I'm just using the colon as a prototypic example. Basically, what will happen on the colon is a homeostasis dominated by bacteria which are primary fermenters, right? So in other words, there is a maintenance of hypoxic condition in the lumen of the gut, and this is dominated by the pie chart shown here, which is basically all primary fermenters. So they utilize the oxygen and then convert this into carbon dioxide. So the level of anaerobic or hypoxic condition is maintained. However, when this changes to a stage where there is loss of this control, and then the oxygen diffuses through the vasculature into the lumen of the gut, that changes the microbial environment in the lumen, which becomes more anaerobic or facultative anaerobic, for example. And then you can see the shift in bacterial profile from fermenters to facultative bacteria. And this can lead to several diseases, some of which are mentioned here. So speaking of diseases, one of the publications in my lab is looking at the role of microbes in polyposis, which is what the title was initially. So my lab works with the gentleman at Children's University Hospital here in the Kansas City, United States. We work on a disease called familial polyposis, or Ph.D., which is a rare disease, 1 in 8,000, followed by loss of process of certificate on ABC. This could be a combination of those germline as well as an acquired mutation, in which case there is a defective ABC protein, which leads to an activation of the immune pathway. There is a transcriptional coactivator downstream or beta-18, which goes to the nucleus and promotes cell proliferation. And this is definitely a process that sets the ball rolling for neoplastic transformation. Okay. So a couple of years ago, we published a paper where we found a bacteria called fusobacteria, shown right here. And fusobacteria hasn't shown in patients who have colon cancer, but we were the first one to show that it is also upregulated in the polyps of these patients, as shown here is the orange here, compared to the individuals, for example. And what was intriguing was that we only found fuso in the mucosa, but not in the skin samples. And then because one of the proteins in fusobacterium called FAD, it promotes DNA damage, and if you fix capsular cell lines such as SINGH or HT29, with the fusobacterium, it can form bigger tumors, and they also can cause DNA damage. And the studies have now shown in the paper indications where they found that if they can limit this oxidative DNA damage through antioxidants, you can actually help reduce the presence of these significant double-strand breaks. So why is that important? Because one of the agents for release by immune cells during inflammation is the oxygen species or ROS. And what ROS does is causes guanine to oxidize to aoxyguanine. So for the students, if you remember, most of the time guanine binds to tyrosine, right? But when guanine is oxidized to aoxo, it actually binds to adenine. So there is a cytosine to adenine mutation, which sets the ball rolling for neoplastic transformation. So we wanted to study the effect of these condition media on significant double-strand breaks. So first what we did, we grew this particular strain of fusobacterium or fusobacterium mediatum. We collected the condition media and treated the ESC116 cells with the condition media. And compared to control, when the cells were treated with the condition media, there was a significant increase in DNA double-strand breaks as well as the MAGA-2As. And this is the same thing is quantified here as well. So is there an evidence of this significant double-strand breaks in FFP? Because nobody has shown before. So if you just recall from your anatomy labs, you have the colon structure shown here, which is the right colon or the second colon transfer as well as the descending colon. So we collected samples from not only cecum, but also from the three portions of the colon, and we labeled them for both aoxyguanine as well as MAGA-2As. And as you can see, in both the ascending and transverse colon, there was significant increase in aoxyguanine. What was even more exciting was the finding that MAGA-2X is also, which is a marker of the double-strand break, is also present in both ascending and transverse colon. However, there was very little in the descending colon. And so this is consistent with presence or absence of biofilms, which was shown by Simsic group at Johns Hopkins. Here they found presence of biofilms in the right colon of the proximal colon, but not in the left colon. So next, we wanted to see if this damage is correlating with presence or absence of biofilms in the three different areas of the colon. So we strained these colons with antibodies for lipopolysaccharide, which labels gram-negative bacteria, then as well as lipoegoic acid, which labels gram-positive, and then this is the merge. And you can again see that there are significant differences if you just compare ascending versus descending. So basically the right colon versus the left colon, there are significant differences in both gram-positive and gram-negative bacteria, and which is again consistent with presence of biofilm in the proximal but not in the distal colon. And we also found presence of these fusobacterium, in the two areas of the colon as well. So the question is, how does presence or absence of biofilms influence whole cell signaling to promote polyptosis? So for that we performed proteomics, and I don't have to tell this audience what it means. We collected 10 samples. We had 2 subjects, 6 polyps, and then 4 mucosal sites, which were analyzed. So just a map of the overall process. And we found several proteins that were upregulated in the polyp versus proteins that were upregulated in the mucosa. And while we were doing this, there was a paper that came out in Nature where they had already shown that a substrain of fusobacterium more prominent within the colorectal cancer patients compared to the oral. So just remember, as a reference, Fusobacterium is a component of the oral microbiome, but to be present in a different, for a substrate to be present in colorectal cancer patients, actually, this was obviously the fascinating area of research. Fusobacterium has been not only just associated with colorectal cancer, but also with colorectal recurrence, as well as a drug as chemo resistance, actually, right? So the idea now is to identify these substrates which are just described, and then do a protein, identify their proteomic, profile, for example. So that the way the process works, you have these, cells collected, develop them into organoids, and then use various approaches, for example, bacterial chemical labeling, microinjection, labeling the organoids, for example, and this can all be, incubated with, various fluorescent dyes, for example, and then you can image in confocal microscope. So as a first step towards this, we have already developed the organoids from these patients with polyps, and again we had different regional, polyps, cecal transverse versus descending, and as you can appreciate, if you look at the way these polyps divide and grow, is very different. And this is day 10, this is day 30, and so we can also look at we can also confirm that there are indeed regional differences in cecal and transverse polyps as organoids. So you can see that at 30 days there is a lot of growth in the transverse colon, but very little in the descending, for example. And this is again shown here in terms of how many cells or crypts grow from the organoids into cryptal structure, for example. And then we have also looked at various markers, and you can see again that there are some markers which are more abundant in the transverse colon versus some in the descending colon, for example. So in summary, this current study sort of suggests that there is definitely an interplay between the concentration of Fusobacterium nucleatum in patients who have familial adenomatous polyposis, and these regional differences, in double-strand breaks could be the reason or the driver of differences in the way the polyps grow in the proximal versus the distal colon. The proof of concept analysis of affected tissue, we did extensive molecular details of proteins and pathways, and maybe next year I'll share all the data with proteins, that were sort of up and down regulated, and what are the molecular structure of those. And so the idea is to be able to really understand what actually promotes the growth of these polyps, and when do they become cancerous. So I think that's an area of further exploration from my lab. And with that, I got all of my lab members, my different collaborators, as well as my funding from both Institute of Health as well as from University of Massachusetts. With that, I will be more than happy to say goodbye to this conference for now, and hopefully I can see you guys next year in person. Thank you very much.
Thank you very much. I think this was a recorded talk, so you might be, you can actually, if any questions are there, we can just pass on. If there's any comment, you would like to have some comment from the house? I think we can just wind this session. We go to the next talk.
Thank you, sir. We now move to our next scientific session, Radio Diagnostics of Precision Imaging and Targeted radionuclide therapy of cancers. This session will be delivered by a highly distinguished speaker, Dr. Satish Chitney. The session will be chaired by Dr. Srinivas Shetty and Dr. Krishna Sharan. I request the chairpersons to introduce the speaker.
It's my relation to Chitney. Satish Chitney is an associate professor in radiology at University of Chicago, specializing in development of molecular and targeted radiotherapy for polymerase imaging of radiopharmaceutical therapy. This research focuses on creating advanced tools for PET imaging and for targeted radiotherapy using diverse molecular scaffolds and such as small molecules, peptides, antibodies, and radioisotopes using Ph.D, radioiodines, and zirconium nuclei. His work aims to enable sensitive and specific imaging of cancer and disease-related biomarkers. And he has worked in close collaborations with clinicians, chemists, biologists, physicists, and pathologists. His work, he completed his Ph.D. in radiopharmaceutical sciences at the KU Leuven, Belgium, and his publication record is extensive and numerous peer-reviewed articles listed across the Dr. Chitney, to summarize, is a prominent researcher in radiopharmaceutical development and molecular imaging, advancing precision diagnostics and targeted therapeutic strategies through innovative radio-based technology. Thank you very much for this innovation, Chitney. Over to you.
Can you hear me? Yeah. Thanks so much for the kind introduction. So my name is Satish Chitney. I'm primarily a radiopharmaceutical chemist. The research focus of my lab is to develop new radiopharmaceuticals for diagnostic imaging and in the context of cancer as well as for therapy. Moving quickly, so I thought the theme of this conference, is also very well applicable to what I'm going to talk, except the fact that I would like to emphasize the atoms. Molecules are composed of different atoms, but in the case of diagnostic radiopharmaceuticals and therapeutic radiopharmaceuticals, it's the atom is the one that's going to deliver whether it's diagnostic information or the therapy, but nonetheless it's in the form of molecule, which is primarily used for targeting the issue. To set the baseline for students and who are new to this, so I'll be referring to the terms radioactive isotopes, radionuclide, radioactive element. They all mean the same for the purpose of this talk. But simply speaking, you have the isotopes or elements, right? If you remember the periodic table, so you have different elements, and some of those have radioactive versions. So the difference between the non-radioactive or the stable, atom or element versus radioactive element is that they have trying to get pointed here, but so they have the same atomic number, but the mass numbers are different. For example, if you take fluorine, for example, within the nucleus it has 9 protons and 10 neutrons, so it has a specific proton to neutron ratio. Whereas we also have radioactive fluorine, which has 9 protons but only 9 neutrons. So the ratio of protons to neutrons is much higher there, 0.9 here versus 1 there. And because of this, so this is an unstable atom. It's in radioactive form. It's an unstable form. And then radioactive elements tries to attain stability by using a proton or neutron. But in the process, they also emit radiation, which is most frequently a photon or gamma rays, which we use for imaging. So this is, if you remember, this is the periodic table. So, but here we are highlighting the isotopes or elements that have radioactive versions. But within that, we have imaging version of isotopes, which are referred to as PET imaging and SPECT imaging versions. But we also have the therapeutic isotopes on the right hand, beta therapy, alpha therapy, and Auger electron emitters. So they are indicated by the color code. But the bottom line is we have a wide variety of choices to choose from if you are interested in developing radiopharmaceuticals. And a lot of times it depends on whether it's intended for imaging or therapy, and within imaging we have different types of imaging modalities, PET imaging, SPECT imaging, And then within therapy, you have different types of therapeutic radiation uses. So again, frequently radioactive isotopes, because they are unstable, once they are in radioactive form, they try to attain stability by decaying, but in the process, they emit different types of particles or radiation. So, but simply speaking, radioactive isotopes, decay by emitting either alpha particles or beta particles, or photons, which are gamma rays used for imaging. Whereas alpha particles are very heavy, they are very heavy in that they cause greater damage in the distance that they are traveling. But they are also easy to stop because they are heavy, which is essentially a helium nucleus with high energy. Whereas beta particles are high energy electrons, which have less damaging ability compared to alpha particles, but they can travel for longer distances. Whereas gamma emissions, gamma rays are like highly energetic photons which can permeate through a lot of different materials, and you need either thick lead shielding or concrete to kind of stop the gamma rays from permeating. So this image kind of shows the principle of PET imaging. So simply speaking, radioactive isotopes are most commonly used in the form of pharmaceutical. There are some exceptions to it where the isotopes themselves are used for diagnosis or therapy. The best example for that is radioactive iodine, which is inherently taken by thyroid glands or thyroid cancers and things like that. But otherwise, most frequently they are attached to specific molecule which has specific targeting properties on diseased tissue or healthy tissue. But in this case, let's say you have a radiopharmaceuticals, which is as an example is it's shown C11 label PIP on the right side, the chemical structure. So it's injected to a subject after allowing for sufficient time for it to distribute within the body and accumulate in the target tissue, but also gets cleared from the normal organs. The subject is imaged using positron emission tomography, which is represented as ring of detectors here. But positron emission tomography depends on the principle that when C11, for example, decays, it emits two 511 keV gamma rays at 180 degrees angle. So if the detectors, the ring of detectors detects two gamma rays, hitting them near simultaneously, it's considered as one event, or a line of response is drawn with. So after scanning it for 20 or 30 minutes, you have after reconstructing the image, you have an image that represents, whether your targeted molecule is accumulating in the intended tissue or not. Based on that, you can make a decision on, okay, this person has, specific biomarker overexpressed or underexpressed, which is an indicator of a specific disease or condition, or an indicator of effectiveness of therapy and things like that. So getting to the therapeutic isotopes, which I will be focusing on half of my talk on, maybe. So we have alpha emitters, and simply speaking, alpha emitters and beta emitters. Alpha emitters, as I mentioned, are very heavy, but they have high energy, and then they transfer all their energy, within short range, which is typically in the order of like 50 to 100 micrometers. Whereas beta emitters, they are less damaging, but they can, cover larger areas, which is typically in millimeters, 1 to 5 millimeter tissue range. So this cartoon nicely depicts the differences between alpha emitters and beta emitters, but the point is, both these agents, if we formulate them into radiopharmaceutical and then if we deliver to are designed in such a way that they accumulate in specific cancers after binding to specific cell surface proteins or enzymes and things like that. So you have high accumulation of these agents, and then they sit there and they continuously emit these alpha particles or beta particles, which primarily induce DNA damage. That's their primary mechanism. But in the process, as they sit there and they emit these particles, they also create a lot of free radicals, primarily hydroxyl ions and oxygen, which can cause secondary damage through reactive oxygen species mediated DNA damage. So you can also see the range of alpha particles, which is indicated as 40 micrometers to 100 micrometers, whereas for beta particles it's 0.1 to 3 millimeters. My computer is working, but it's not on yet. So that's the difference, because alpha particles primarily cause, significantly higher number of double-strand DNA breaks, which are harder to repair by the cell, DNA repairing mechanism. They are believed to be more potent compared to beta particles, which you will see some examples proving that's true. Yeah, this pointer, laser. Oh, okay. But anyway, so this is an example, to give you visual appeal for what it means for killing cancer cells. For example, we have a cluster of cells there. The isotope that's indicated on top of the those cells is astatine 211, which is an alpha-emitting isotope. So if astatine is present in the tumors, so it has a permeability range that covers like 3 cell layers. A typical diameter of a cell is like 18 micrometers to 20 micrometers. So astatine can deposit all its energy within 3 cell diameter range. Whereas, for example, iodine 131 or yttrium-90, which are beta emitters, they have the ability to kind of cover 40 to 200 cell layers, but they cannot cause as high damage as the alpha emitters. So, but both are equally important, and we kind of pick based on what type of cancer we are treating, what is the expected size of those tumors, and typically it's believed in the field that because alpha emitters can deposit all of their energy within a short range, they are ideally suited for treating micrometastasis like brain metastasis, metastasis from prostate cancer, breast cancer, and things like that. Whereas beta emitters are more suited for large-sized tumors. But there are also new concepts of treating them sequentially, initially administer beta emitter, radiopharmaceuticals followed by alpha emitters and things like that. This cartoon simply shows the different components of a radiopharmaceutical and diagnostic therapeutic. So simply speaking, we have 3 parts here. One is the first and foremost is the cellular target. For example, so in the context of cancer, again, cancers have aberrant expression of a lot of different proteins. Either they are overly expressing them compared to normal cells, which provides us sufficient contrast and sufficient number to target them with radiopharmaceuticals, or simply those cellular targets are present in normal cells, but in case of disease, they are either their expression levels are lower or they are absent, which allows us to do differential diagnosis again. And the second part is the molecule. Now you know which target you want to go after. For example, for developing a radiopharmaceutical or designing new radiopharmaceutical. Now you come up with a molecule that has the ability to lock into that cellular target. So here it's showing as cell surface target, but it could be intracellular or intranuclear, depending on the significance to the disease and what exactly we want to accomplish. But molecules, typically the most commonly used molecular scaffolds are small molecules, peptides, and antibodies. So once you know that you have reasonable assurance that they can bind to the cellular target, then we pick the radioisotope that can match with that type of the molecule. Again, as I showed on the periodic table, we have a wide variety of choices. Sometimes it depends on the type of the molecule we are using, because those isotopes have different half-lives. We have to pick an isotope that matches with the pharmacokinetics of the molecule. For example, if the goal is to use monoclonal antibodies which have a circulation half-life of like in the order of 2 to 3 weeks, you may want to pick radioisotope that has half-life in the order of weeks or several weeks, for instance. Whereas if you are targeting small molecules, we have something like radioactive fluoride, radioactive carbon, which have a maximum half-life of 1 to 2 hours, which are well suited for small molecules. For peptides, you have a little bit of flexibility to pick intermediate range isotopes or long-lived isotopes, as long as the pharmacokinetics are correct. But again, this cartoon shows the fundamental principle of a radiopharmaceutical and how it works. So that's a little bit different from the traditional pharmaceuticals. So in that, most commonly the radiopharmaceuticals are administered at very trace levels. By that I mean, if you talk about the milligrams, micrograms, or molar quantities, they are often like, less than 100 micrograms, the typical dose of a human being with high specific activity radiopharmaceutical. So they are almost always administered by IV with the exception of some which are administered by oral or other means. For diagnostic and therapeutic purposes, most of them on theranostics are administered by IV. So once you have the radio tracer, distributed in the body, it comes across your, targeted tissue, for example, it's brain or tumor tissue. Then, it extravasates and comes across, its intended target. The receptors, for example, work lock-in key mechanism with the molecule you are using. Roughly speaking, because we are not using pharmacological doses, only a small proportion of those receptors are occupied by your radiopharmaceutical. Let's say typically less than 10%, less than 5%. But it also depends on the expression levels of those targets. If they are very small, then you'll be occupying significant proportions of those receptors. And then within a few hours, depending on the molecule, again, whatever is not bound is typically cleared through the blood circulation, eliminated through kidneys or hepatobiliary pathway. That's the typical biodistribution pattern. And now you wait for a few hours for this to happen, and then you get an image where your agent is accumulating in the intended target tissue, and then based on which you can derive information on the diagnosis or use it for therapy. Again, to highlight, so this is the fundamental principle for radio tracer in the sense they are used at trace levels, and for that reason it's very rare to see pharmacological toxicity for radiopharmaceuticals. The major source of toxicity is the radioisotope or the radiation dose rather than the pharmacological toxicity. So now getting into the kind of hot topic in our field in nuclear medicine, radiology, or so is theranostics and radiotheranostics. So if you are in cancer research, cancer care, or are interested in getting into it at some point, it's likely that you will hear this term. So I'd like to introduce what is theranostics and what is radiotheranostics. So simply speaking, theranostics is using a paired diagnostic and therapeutic concept or agents. So in this case, the diagnostic and therapeutics could be structurally different. You're simply using a diagnostic test tool to select, for example, appropriate patients for your therapy. Because these days, the whole pharmaceutical field and pharmaceutical agents are, getting more and more towards precise targeting, right? We're talking about, targeted therapeutics, personalized medicine, precision therapy and things like that. So those agents are not only expensive, but they're also ineffective if those targets are not expressed at sufficient levels in patients. So it's important to select, and these days, a lot of drugs approved by FDA are in fact, targeted therapies are approved in combination with a diagnostic tool. But same concept if you apply to radiotheranostics. In this case, you are using a combination of diagnostic radiopharmaceutical and therapeutic radiopharmaceutical. In most cases, the molecule is very similar structurally, in the sense you have the same targeting molecule that binds to your receptors or enzymes or cell surface proteins, and also in some cases it has a linker which is used to separate the pH-like structure which is required for radiometals to bind. But also to fine-tune pharmacokinetics. You can change the structure of the linker without affecting the binding affinity of your targeting molecule. But in the context of theranostics, so we are using the same molecule, but in this case we are switching the diagnostic isotope after making a definitive diagnosis to radiopharmaceutical that has therapeutic isotope. So best example for FDA approved theranostic agents is using of gallium-68 in a couple of radiopharmaceuticals for diagnosis, but switching to therapeutic, for therapeutic it's lutetium-177, which is a beta-emitting isotope. So we'll see some examples of these in the next few slides. But I want to emphasize is, this is not the whole field in the sense this is a subset of field, but it's making, direct and significant impact on the cancer management of specific cancers, and it's likely to extend to other types of cancers too. But on the other hand, we have a whole range of diagnostics which you could use to implement specific therapies that are not necessarily radioactive. Best example is again Alzheimer's disease. Recent approvals of beta amyloid targeting antibodies. Now we have 4 different FDA approved Alzheimer's imaging agents that they are using in the clinic to select patients for those therapies, because the presence of beta amyloid protein is a requirement to implement the antibody therapies. So that's an example of theranostics, but not necessarily radiotheranostics, because we are not using therapeutic radiopharmacy. So this this shows it's a busy slide with a lot of different isotopes. So again, the numbers represent the molecular weight, and the letter represents the symbol for the isotope. But simply speaking, the arrows indicate rational combinations of diagnostic isotope and therapeutic. And these also go hand in hand with the type of the molecule we are using. Some molecules have more flexibility, but generally speaking, like I said, you want to match the half-life of the isotope to the pharmacokinetics of the molecule we are using. For example, for small molecules, you could use radioactive iodine, you could use fluorine-18. For peptide, you could use scandium, but for antibodies, you are stuck with zirconium-89 for imaging. For therapy, you could use lutetium-177, but radioactive iodine is also a possibility. But the other factor is the availability and cost, which is very important, because some of the radioisotopes are either very expensive or not accessible simply. So that also is one of the factors that goes into it. But speaking about a little bit history on the radiotheranostics, so this concept is not at all new. So it goes back to 1940s, where they systematically evaluated the therapeutic efficacy of radioactive iodine-131. Iodine-131 also has a gamma emission, which means you could image iodine-131 using SPECT or planar imaging. So they did systematic studies of injecting patients with iodine-131 to patients who have hyperthyroidism. So on the left side you see image of a patient with iodine-131 showing normal uptake on the left side, and in case of hyperthyroidism, you have increased iodine accumulation in thyroid gland. But they administered doses from low doses to up to 10 millicuries, and they kind of concluded that if you administer more than 2 millicuries, you have significant destruction of the thyroid gland. This is based on the fact that the treated thyroid glands has only 16% of the uptake injected dose in the thyroid gland, as opposed to 61% in the normal thyroid gland patients. So that's still being used until today, but another good example is in the use of radioactive iodine for the diagnosis and treatment of thyroid cancers. Again, on the left side it's showing images of extensive metastasis of thyroid gland. But after that, there were I don't know if there are any other radiopharmaceuticals approved before 2000s, but this was an example, probably first examples of targeted radiopharmaceutical therapy. So the previous example was using radioactive iodine as such, which is great, but not all isotopes have physiological role. Let's say they are simply eliminated through kidneys or liver, or so they are not targetable to disease as such. But these are two successful examples. The tritiums are Bexar and Zevalin. This was when I got into this field with my PhD. There was a lot of enthusiasm and excitement, followed by a big disappointment, because ultimately, even though despite the good therapeutic efficacy of these two agents, those two agents were not very successful. At least in the US, Bexar was no longer in the market, but Zevalin is still available, I think, but not extensively used. Part of the reason I heard was supply chain issues, and medical oncologists were not really ready to use targeted radiopharmaceutical therapies and things like that. But that was when but this also stimulated a lot of interest in the field to develop these kinds of therapies. That was when the company called AAA, which was incorporated around 2002 or 3, which went on to get approval for somatostatin receptor targeted therapeutics, is later acquired by Novartis. So this is an example of probably the first theranostics based on targeted radiopharmaceutical agents targeting neuroendocrine tumors. So just a brief background. So neuroendocrine tumors have high expression levels of the receptor called somatostatin receptor subtype 2, octreotide is a natural ligand that binds to those receptors, and then chemists and radiochemists have designed an octreotide analog where a chelator is added to this molecule, which is shown in on the right side figure with M in the middle. So that the chelator is called DOTA, which binds to a lot of radiometals. But so in this case, it has the ability to bind to diagnostic metals such as gallium-68, but also to therapeutic isotopes to like lutetium-177 177. So this radioligand has been approved for both diagnosis and therapy. In fact, it's the first example of radiotheranostics approved by FDA. So for diagnosis, we also have now a Ph.D. labeled agent approved, but most commonly it's used in combination with gallium-68, which is available in the form of generator, which you see in the picture on the lower side. So typically hospitals which use this theranostic pair, they buy the gallium-68 generator, which can give gallium-68 for a few weeks or even months, depending on their usage. They dilute it, dilute, and then add to the kit that has that starting peptide with some stabilizers, buffering agents, and things like that. So the mixed gallium-68, I think they heat it at certain degrees, 70 or 90 degrees for half an hour, and then the reaction is complete and ready for use in patients. It's directly injectable. Now same peptide, now if you switch to lutetium chloride, again the kit is slightly different. It's not the same kit, you have to use a different kit, Lutathera, which is manufactured and marketed by Novartis now. But simply speaking, you add lutetium chloride, you heat it again, and then it's done. It's ready for injection. But the way it works is, you have a group of patients with suspected neuroendocrine tumors. They are imaged with, gallium-68 dotatate. It's called the molecular form of it, NetSpot, the brand name. You can, separate patients that have good expression levels of SSTR2 and then put them on lutetium-177 dotatate therapy, Lutathera. And then follow up, with post-treatment, response assessment, either by using gallium-68, or in this case, again, lutetium-177 has imageable gamma emission, which you could use to image by SPECT imaging. And patients who don't have, appreciable expression levels of SSTR2, they could be put on other therapies that saves them cost, and gives them, some additional options for therapies. So the Lutathera was approved based on the interim analysis, which showed significant difference between control group and the Lutathera treated group, both progression-free survival and overall survival. Based on that, it was approved by FDA, but they didn't see significant benefit in the overall survival, which is still meaningful clinical benefit. And then similarly, we have prostate-specific membrane-targeted radiotheranostics. So again, they have what they call PSMA-617. The lower part of the molecule is the one that binds to prostate-specific membrane antigen that's expressed on prostate cancer cells. And it has a linker and gallium-68 binding chelator. And now we have the same molecule, the targeted molecule, but for a therapeutic version of Pluvicto, you have a different linker and a different chelator there, which can bind to lutetium-177. Again, this was approved in 2022. It made a significant difference compared to standard care of treatment, both in terms of median overall survival and progression-free survival and overall survival. But how about alpha emitters? I remember I said they are more potent. So am I done with the time? Okay, 1 minute. So this is an image, that shows, of a patient with extensive, metastasis from prostate cancer that was treated in three cycles of, actinium-225 labeled PSMA, and then one additional cycle is literally disease-free. And this is an example of, direct comparison of the therapeutic efficacy of lutetium-177 versus PSMA labeled with actinium alpha-emitting isotope. Again, initially the patient did not respond to lutetium therapy when he was, put on 2 cycles of actinium-225. Again, literally he was, disease-free, again making a meaningful impact on that. So we have, several other theranostic, potential radiopharmaceuticals, in pipeline. One of them is, called fibroblast activation protein targeting radiopharmaceutical. This works beautifully for imaging, but it's yet to see if it works also for therapy purpose. But right now there are multiple clinical trials ongoing with therapy. So with that, I don't have much time, but we are also working on creating different theranostics and therapeutic radiopharmaceuticals at our university. For clinical use, we are producing different amyloid imaging agents for different clinical trials and for implementing amyloid targeted therapy. This slide, shows our facilities. We have a clean room radiopharmaceutical production facility with hard cells and automated synthesizers and dispensing and QC lab to go through the whole production cycle. But we are also in the process of creating, kind of true diagnostics where the isotope is also same. Some elements have both diagnostic version and therapeutic version. So that's an example. We are also working on zirconium-89 antibodies for new targets. This is a proof of principle imaging in small animal models injected with breast cancer cells. The labeled antibody accumulates in tumors for diagnostic purpose. We are in the process of creating therapeutic version of this radiopharmaceutical. That's in progress. So in summary, radiotracer principle is the fundamental. Remember, we are not using pharmacological doses. And you have diagnostic radiopharmaceuticals and therapeutic radiopharmaceuticals for separate diagnosis and our therapy, but if you use them in combination, you have a pair of radiopharmaceuticals that enable you to accomplish precision diagnosis and therapy. So with that, I would stop and then acknowledge my lab members and different collaborators.
Starting with the basics and going to the more complex. Dr. Nagaraj has a question.
Wait.
Yeah, that's nice. I just want to know if you have any experience of using FAPI for hemangioblastomas, creating hemangioma. I was just exploring my operation.
For glioblastomas?
Hemangioblastoma, integrated and spinal.
Yeah.
I was just going through the FAPI as one of the options if the scan is positive. Any experience? I'm just patients. I was just exploring.
Yeah, I think FAP is still a little bit early. It seems like it's working beautifully in some cancers, especially for the CNS tumors. That's your question. So we also have to take into consideration that some of those molecules cannot get through DBB. That's one additional issue we have to explain. But as such, I haven't seen many papers are in literature on that.
Radioactive nanobore? Do you have an experience with radioactive nanobore? I do see some people who are working on it for prostate.
Yeah. I couldn't get the question yet, but I'll speak to you in person. Thank you.
Thank you, sir.
Thank you.
Yeah, thank you very much. Thank you to the organizers for this opportunity and Dr. Rao for contributing to this university.
I request the chairpersons to please felicitate the speaker. Thank you, sirs and ma'ams. A heartfelt thank you and appreciation to our chairpersons for guiding the session so gracefully. I request Dr. Prashant Shetty and Dr. Sachidip Kotian to do the honors for our chairpersons.
titled Stem Cell Sparing Radiation of Intracranial Tumors and Cognition. It's a privilege to have with us an eminent expert in this field, Dr. Nagaraj. To guide this session, we are honored to have two esteemed chairpersons, Dr. K. Kamalakshana, Dr. Sukesh Rao. Requesting Mr. Samin K. Vidya, Assistant Professor, to be the rapporteur of this session. I request Ms. Rashmi Shenoy to introduce the chairpersons.
Since I've been introduced yesterday, we can skip that after one point.
So,
This is.
a small request.
I've been introduced yesterday. Let's go ahead.
Sir, it's just 15, the time limit for the session is 15 minutes plus 5 minutes. So, speakers are requested to kindly note the first bell will be rung at 15 minutes and the second at 20 minutes. I request.
Good morning. I'm an initial columnist and I do treat quite a few patients. So when it comes to play, important aspects of mental occupational and in fact, we are, because we are aware, we can think, we can experience. If we lose that faculty, then the very existence, the very nature of existence gets jeopardized. So, this is what we understand by cognition. I'll have a few more slides to deal with or talk about what cognition is all about, but this has a very physical basis and nothing very metaphysical about cognition, because a lot of these gurus might be talking about this and that, but what we're talking about is very hardcore, computable aspects of cognition. So it always happens in brain. It always happens in brain. There are not many areas in the brain and different areas with different kinds of activity which is related to cognition. So say, for instance, about touch and vision, even cell is involved in, we all know, so from the cortex, so even the cellular cortex is involved in certain kind of cognition. Okay? So cognition is a very important aspect of the existence. It involves memory, vision, executive functioning, behavior, attention, language, abstraction, delayed recall, orientation. There are multiple aspects to it. Why do we need to understand all this? Is because when we wish to assess cognition in a very, very systematic, scientific way, we need to have techniques and we need to know what we are qualified, right? So there are, some of these tests are routinely used in the clinics, but there are many more than 15 or 20 tests which can be used. But here, very mental test is very, very commonly used because it's very simple. Just 15 questions in the questionnaire. And to execute them is very painful. And we also need to understand that if we need to assess them, the patient must be in a position to answer them. So the very area, very subject of cognition, assessment, treatment is all very, very complicated. I may skip some slides. Okay. So as I said, I'm emphasizing all over again. Cognition is a very complex process and all important attention, perception, memory, learning, language, and thinking. Why I say this all over again? When my patients come to me and I say radiation to the brain, their first major apprehension before deciding to go ahead with radiotherapy is all this. Can I recall? If I cannot recall my very happy memories, what's life worth? So this is not just philosophical, but very, very profound and important for the very existence of the very being. So cognition needs to be understood, addressed in a much more comprehensive way than we are at the very beginning. So since I'm dealing with, I'm talking about gliomas or intracranial tumors, why is it so necessary? When we give radiation, there are two phases, immediate effects and a delayed effect. The cognition may get affected immediately, 4 or 5 or 6 months, or nothing may happen immediately, and over a bit of time, the cognition may get affected. And very often, latter is true. So, when we are talking of intracranial tumors, except for 1 or 2 tumors like the GBM or astrocytoma, most other tumors here, like say for the meningioma or oligo, they have very good prognosis. Which means if I give radiotherapy today, there's a good chance that they will survive for years to come. And if they will survive for years to come, and if I give a certain kind of radiotherapy, and if that's going to lead to impairment of cognition, which will affect the memory, attention, and language, what is life worth? Right? So this is very complex. We need to understand, see, look at this another graphical representation. If when I'm talking about astronauts, astrocytoma pregnancy, people survive for a while. If I'm talking about GPM, everybody dies in 2 years. So it's not a big deal. But as I showed it in that diagram, more and more patients intracranially have a reasonably good prognosis. So cognition becomes a very, very important consequence of radiotherapy. This is not, I think we have something from NASA here. This is also, when you're dealing with astronauts who are in a space capsule for a long time, or a very long-term travel, in the low-dose radiation also might affect cognition, but then I don't have any data on that. So what I'm trying to say is, the consequence of radiotherapy and cosmic radiation is a very, very important area for research, because there's not much that is known, and we don't really have much to offer once it goes haywire. Once it's destroyed, then there's not much to offer. So why am I talking of stem cells and cognition? Because when I was doing MBBS, we were taught that brain is differentiated, it stays there. There is no further repair, there's no plasticity, nothing at all. If it happens, now forever. In the last couple of decades, we have some resurgence of interest in what we call neuroplasticity, because we do see patients with stroke recovering remarkably well. So how does that happen? If brain was done and damaged forever, and there was no scope for repair, how do we see a remarkable improvement in patients with stroke? So then came the idea, the concept of neuroplasticity, and this is how, anyway, the neurogenesis takes place. That starts with, we have stem cells in a subventricular zone, and of course, in order of the ventricle, it starts from here and ends in neurodegeneration. And we tend to see the degeneration as we grow older. Like when people reach my age, very often I forget names. Now that's not deliberate, but maybe my brain is degeneration. There are quite neural cells, neural stem cells, and then they become active and they get differentiated, and both neurogenesis perhaps is possible even in the adult brain, and neuroplasticity. When we talk about neuroplasticity, we talk about the synapses. Can I get the slides back? Okay? So since these are now actually university, we will look at some of the options. So when when we radiate the brain or any spine or CNS, what happens is the disruption of the brain barrier. Inhibition of alveolar damage sites progenitors, which are very important for neurologists. Cellular damage, capillary damage, are called autoimmune response. This is pretty important. That's why when we use steroids, it has such a remarkable effect, immediate effect. It also protects the future data in balance with oxygen and antioxidant levels, inhibition of neurogenesis and demyelination. So these are the various mechanisms by which ionizing radiation, and it is telling of the brain tissue or spine interaction with that, and there are some consequence of that. So, in fact, it's also dose dependent, and this is what happens by the process. But what is really important to do is cell survival. And at a certain dose, in fact, we have Professor Calipers from Canada, we started using a low dose ionizing radiation to treat Alzheimer's. That's why I'm doing this slide to say that yesterday there was a talk of somebody on a CT scan pretty obsessed with the low dose radiation or trying to reduce the dose. In fact, I believe I'm that group of people, radiologists, who doesn't believe, who believes in hormesis or harmonic effects, and he is using this low dose effects, which is anti-inflammatory, it's not pro-inflammatory, so leading to cell death, demyelination, and inhibition of neurogenesis. So here we are, it's in a pictorial form. Now what I'm going to talk about is these neural stem cells in the hippocampus. Yeah. Now, when we're talking of these stem cells in the hippocampus or periventricular zone, they are all dose dependent. More the dose, the greater the cell death, or greater the inactivation or the damage of these neural stem cells. Which means if damage whatever is there, then the subsequent repair, the chances of subsequent repair is far less. And it's a dose dependence, linear. This has been shown, this also depends on the age of the brain. Especially when we're talking of the neonates and the children, generally radiation is not recommended because the consequence of radiation on these children can really be devastating. So not just the dose, but the age of the brain is also of great roles where we are talking about cognition and other aspects of brain function. This is a picture of that. This is those introduction of profile 15, and that's how you see the depletion of neural stem cells. Stem cells are generally far more sensitive than the tumor cells, so that makes it even more complicated. If you want to achieve a cure, and your therapeutic ratio just narrows down so much, which means you must spend all your time and energy in physical optimization of the neurones, such that you protect the neural stem cells because they are far more sensitive than the tumor cells themselves. And this cognitive decline following radiation is seen in more than 50% of the people. It's dose dependent, volume dependent. And decline is volume dependent because if the tumor is very large, very often you have a baseline decline in cognition and protecting the hippocampal stem cells on the niche. It becomes almost impossible, and the dose received by the stem cells will be pretty high. That's why it is volume dependent. So, what are the mechanisms besides what I said before about disruption so on and so forth. If this persists in the latter one, so it happens in other. So if you protect the sum of the plasticity, neurogenesis, and again, cognition. The vocabulary is a stem cell niche, so that's the area within the brain which we should try and protect. The rest of it we need to optimize. And abnormal N-beta-D aspect, the receptor signal, this is very important because we can modulate that. We have some medication which you can modulate that. And it's one of the important receptors which modulates cognition. So addressing this can help us. I'm going to flash through this is the same thing. Maybe this is the vascular aspects of this, and the functional inhibitors can be used because so that the consequence on cognition, consequent to vascular disruption can be used. But already we don't have enough evidence for that. Reduce projection density and increase the excited synapse line density. You can use momentum. This is the one drug which I started giving to theory, and there is now a level of evidence that if you use that along with radiation, and subsequent radiation, probably we do promote genesis and neuroplasticity, and just may help cognition. This is Ross and inhibition of that junction. It is evident not just in the human brain, but across the species. So, skip that. Yeah, there we are. So, when we started looking at sparing these stem cells, we need to identify them in the scans. So, this is how it looks like. They're all distributed in the lateral ventricles. That's the hippocampus, CA3, CA2, CA1. They all have a different role to play, but the composite of all of this is neuromodular stem cell niche. On the scan, this is how it looks like. It's one of the earlier papers. We were treating gliomas, and what you see in the big red, the big tumor. But you can see the lateral, those two small circles. They are you've got niche here. The symmetrical one are the hippocampus on both sides. Yeah, we need to spare them. We have these metastatic tumors, right? So why do we need to spare the stem cells? If you lose spare the stem cells, then the chance of them before the neurogenesis and the cognition. Impairment cognition may be reduced. This is from the original paper of Gandhi, who did a lot of work in metastatic tumor, and we started doing it routinely after that work. So I can't claim that it's my notion of time. Okay? This is the lateral work of the results. This is how the actual results are there. So whenever we sort of contour this paper on the scans, we look at all three-dimensionally and make sure that we include subventricular zone and hippocampus and both sides. It's not always possible to spare this given stem cell formation. We should try as well as external as possible. And how much can we spare? Obviously it cannot be zero. So this is all based on their work, and I mean 8.6 to 9, 12 grade. If we can achieve that in both the hippocampus, that's incredibly good. This is where we are. Dmax is less than 9, acceptable variation is 9 to 10. So when they did this kind of work in the metastatic brain tumor with the whole brain radiograph, they concluded demonstrated that sparing stem cell formation in the brain during radiotherapy can assist in preserving cognition, or the decline of cognition will not be as bad as if you don't look at it. However, when we're doing all this, when we were all exploring sparing the stem cells, there was this thought that maybe, especially the GP, these stem cells, in fact, should be inactivated because they migrate and they become cancerous stem cells, which means you should not be, in fact, spared. So that was a bit of a dilemma, and people were trying to explore. So what we did was, we looked at, we had transition from very conventional radiotherapy to one of intensity modulating radiotherapy and radiotherapy as well. Then we looked at the 3D CRT, and I'm happy that we have tried to save stem cells. And there was significant difference in there was a significant difference in the dose that was given. Okay? So the TDCRT patients got a lot more radiation than IMRT, but there was no difference in survival. That's the difference in survival, which meant when you save the stem cells in the treatment trial, the survival remained unaffected. So we went by those details, and this was before we went into the hypothesis with the computational work, and we using various mathematical models. We look back, and now we do spare the routine, because now we are confident that sparing real stem cells does not lead to decreased survival. Now, can you also have some kind of immunotactics? This is work in progress. Unfortunately, don't really. There's a huge list, except for steroids and now momentum. Nothing much is there. I urge the experts here to come up with something useful protective so that we can just go beyond physical optimization and use the stem cell. And understand one of the tablets that I'm using these are the various techniques, basically physical optimization, material stem cell transport, and just an antibiotic system don't work. Now the one preventive works, this is how it works, and we need to have many more effective neurodetectors. This is how the preventive works in that particular receptor. Yes, this is how it works. It has very minor side effects, so we use it for the long term. We are trying to use amifostin in the past, but it's very toxic drug, so not available anymore. So amifostin also locates itself in the brain. So a very useful drug that is very toxic and mouse. So we need to go beyond medical, we need to go beyond steroids. These are the tools to help me put this slides together. So, cognition as impairment and the consequence of radiotherapy, space travel, so on and so forth, can be very devastating. Can be very devastating. They are not very effective treatments. The idea of neuroplasticity is exciting, and neurogenesis in older or elderly people is very exciting. Probably the low-dose consequence of radiation is quite different from the high dose. If we can spare the stem cells in the dish, in the brain, so much the better. They are far more sensitive than the glial cells themselves. So physical optimization has a limit. Memantine is one of the drugs which is pretty safe, which is used in Alzheimer's routinely, but not all the drugs that you use for Alzheimer's can be used to treat the cognition, decline in cognition due to radiotherapy. And thank you so much. I hope some of the extras will hear questions.
Thank you, sir, for an excellent Dr. Darkar.
scenario in treating pituitary adenomas. What is small lesions? Pituitary adenomas compared to GBM. Second one, by extrapolation, do we have evidences to believe that both proton and carbon ion radiotherapy has any less cognitive deficiencies?
Protons, yes, especially, no, definitely. See, that's what I'm saying. Physical optimization. Now, one of the slides that show you a stereotactic radiotherapy. Now, the pituitary can be now easily varied to stereotactic radiotherapy. What happens when we treat the pituitary at an early age is endocrinopathies which need to be managed appropriately. If we don't do that, then we can have mentally intolerant children. So when it comes to children, one has to be very careful. Protons, yes, that is, especially for many people, AV malformations, acoustics, where we are anticipating a long survival. Where it makes no sense in GBMs and plastics area of time. Very big tumor protons plus one. Yeah.
Follow-up to the same one. Flash is something which has been there for a long time, but lately it's kind of a fad. Whether Flash really has any scope for prevention of cognitive? I am very skeptical about it, but do you have any thoughts about it, number one? Number two is radiation-induced changes. You have shown some of these are there possibilities of directly affecting certain ion channels. That's why probably you are able to use acetylcholine esterase or inhibitors, ACE inhibitors.
Let me, as far as Flash is concerned, it has been there for the last 2 decades, at least 10 to 15 years. The idea of Flash seems to be, there seems to be therapeutic margin, therapeutic ratio. I don't know much about it, but it's work in progress. It's more still in the lab. We don't really have any experience. Theoretically, in principle, maybe, because there's a much more therapeutic ratio if you can physically optimize like we do with other iron methods. Or you have something like an intraoperative Flash. Maybe those things can be looked at. But at this point of time, I think it's got to be.
Yeah, so the ion channels. So are there any direct relevance of radiation-induced damages to neuronal receptors like ion channels, which could change the gases and liquid like water, etc.?
And in fact, memory does that. All the potassium, calcium, gaining channels, it does modulate, and memory does that. So, yes, they do. And I think many of them have an effect on cell membrane, especially calcium.
Thank you, sir. I request the chairpersons.
Sir, thanks for your insightful session. I have one question. When we are using this NAC, can it cause hypersensitivity reaction?
Oh, yeah. Correct, yeah.
And what is the prevalence of this hypersensitivity?
Yeah, that's an area which fascinates me, but there is no hard data in the clinical practice that the low-dose hypersensitivity really is seen. And in the brain, yes, people have said that it could happen, but we don't see that routinely in the clinical practice. Yeah? I'm quite interested myself. I keep asking my MR guys to look at with the advent of IMRs, with the advent of all these techniques, where we concentrate on even SRS, very low, high level, and then we get the levels, right? And here, when we're talking about camps, in fact, very low, if I can achieve very low dose, can be not for anti-calories. So it's very interesting, very complex, not totally else.
Thank you, sir.
Thank you, sir. I request the chairpersons to felicitate the speaker.
We now proceed to an intellectually stimulating session titled Radiation Resistance in Insight, a lesson from transgenic animal model, Drosophila melanogaster. To deliver this session, we have amongst us Dr. Shyam Prasad Varijarabu. To chair this session, we once again have our eminent chairpersons, Dr. K. Kamlaksh Shenoy and Dr. Sukesh Rao. Speakers, kindly note, the first bell will be rung at 15 minutes and second bell at 20 minutes for you to kindly wind up.
Hello. Thanks to the organizing for the meeting. The speaker for this session is Dr. Shyam Prasad Varijarabu, who is an accomplished scientist, expertise in genetics, radiation biology, and molecular biology. He completed the MSc and Ph.D. in the Applied Pharmacy from my university and received the prestigious lab fellowship to pursue research at the University of Germany. He later completed postdoctoral training at the National Lab Institute of Neurobiology and worked as a research scientist at the US Medical School and Neuroscience Research Partnership in Singapore. In 2015, he was awarded the DPT Ramalingaswamy and DST Ramanujam DMT fellowships, which enabled his return to India. At Mani University, he established a Drosophila Neurogenetics Laboratory and was honored on Foundation Day 2022 for his contributions. In 2023, he joined the Yellowboy Research Center as Senior Associate Professor, where he leads a well-linked lab focusing on genetics, molecular biology, and radiation biology using Drosophila models. His lab is recognized for training students and faculty under the Indian Academy of Sciences Summer Research Fellowship. Dr. Varu has nearly 95 publications, including one in Nature, with a total impact factor of about 500, along with 22 book chapters. He is a frequent resource person for academic programs and maintains strong national and international collaborations with funding from PRRS, ICMR, and DBT. He also serves as an invited reviewer for reputed journals in neurobiology, radiation biology, and physiology. With this introduction, I hand over the stage to Dr. Raghu for his talk.
So, good morning. I hope all of you can hear me. So first of all, thanks for this great invitation from the organizing committee, and also I would like to thank Suchita madam for this invitation. So I will talk something completely different what other speakers dealt with so far. So my title of my talk is Radiation Resistance Insight, a lesson from transgenic animal model, Drosophila melanogaster. So radiation effect. So a lot of people talk about radiation, like how it influences, how we can use radiation as a treatment strategy for different cancer, all those things. But the question is, when we talk about this radiation effect, so we also talk about there is something very interesting phenomenon that is called radiation resistance. So if you go through the animal kingdom, so for example, man is affected by about 3 to 5 gray of radiation. But if you look at down the evolutionary line, so mice can survive up to 7 gray, and Drosophila melanogaster, that's an animal which I'm going to talk today, it can survive up to 1200 gray. And if you go down, even if bacterium, that is Deinococcus, which is known to be one of the most resistant, radiation resistance organism existing today. It can resist up to about 4,000 to 6,000 gray. So there are different possibilities, there are different hypotheses which is responsible for this radiation resistance, it could be efficient antioxidant mechanism or efficient DNA repair mechanism, or it can also be due to the hyper stress response, or it could be presence of holokinetic chromosomes where there is no much crossing over between the chromosome, and also it could be resistant to the radiation-induced apoptosis. So in my lab, so what we are looking is like we are taking Crucible as a model system to understand the mechanism of radiation resistance. The whole idea is like if you can find some mechanism or if you can find some molecule which is responsible for the radiation resistance, that can be used as a advantage for human welfare. So what is Drosophila melanoic acid? So Drosophila melanoic acid, especially for the most of the people here are from clinical background. So Drosophila melanoic acid is simply called as fruit fly, so which you can easily find in your kitchen, like it's normally roaming around the fruits. That's why it's commonly called as fruit fly, and also it's also called as signal of genetics because it has given almost like a lot of fundamental discoveries to the scientific field, and also Drosophila field also received around 6 Nobel Prize winning work. But why Drosophila? Because I already told that Drosophila has very high radiation resistance, but why Drosophila? We can also take other insect, but why Drosophila? So Drosophila offers a lot of advantages. That is, one is like it can be easily cultured in the lab, and you can have a lot of animal for your experiment, but most importantly, it has a lot of genetic tools. So one of the major genetic tools which we use, or most of the trial lab is using, is GAL4-US system. So GAL4-US system is mostly used for manipulating genetically, gene manipulation in the Drosophila. So basically it has two components, that is GAL4 and UAS component. So in the GAL4 we can clone gene of our interest so that the expression of the gene manipulation can be restricted to different tissues. So then we have second component, that is UAS. So where we can clone, some modulator like toxins or anything which can actually kill these cells. So basically GAL4-US system, in the GAL4-US system we can manipulate gene or genetic manipulation where the tissue which we are interested. So that is why Drosophila is one of the best model system currently available when we talk about the genetic manipulation. So here we use this approach and look at did a lot of genetic manipulation Drosophila and look at some of the molecules which are responsible for the radiation resistance. And due to all this reason, today Drosophila is used for almost every field in the biological research. For example, our lab basically work on the nervous system. Drosophila also has a nervous system. Most of the time it will be surprising to see that Drosophila has nervous system. Of course, it is one of the highly studied nervous systems. So people use Drosophila for different studies. So let's come back to our own study. So we knew that insects have very high resistance from the beginning. The question is, what is the mechanism? So there was some hypothesis that it could be the chitin, most of the insects have very strong chitin layer outside the body. So people initially proposed that it could be the chitin which is responsible for this high radiation resistance. You have to imagine human is around 3 to 4 gray and insect is, Drosophila is around 1200 to 1500 gray. It's a huge difference. So first we want to check whether it's really chitin is responsible for this radiation resistance. That's what we did is we took different stages of Drosophila. So it's a insect. So it starts from egg, then it undergoes different developmental stages. That is larval stages, pupa, and then adults. So adults are male and female. So female have XX and male have XY chromosome. So what we did is we took all these different stages of Drosophila. So then we put them under a gamma chamber, so we have a gamma chamber facility and treated them at different radiation level. So if you look at this graph, it's a bit complex. So only this X, what is shown in red, and the late pupal stage shows some sort of death. So this is something like a survival assay. We treat the sample under gamma chamber, then we observe for 24 to 48 hours whether they survive or not. So this is what the result coming from this experiment. So if you look at this graph, so egg and one of the stages called pre-pupal stage have some sort of death. But otherwise, most of them are surviving very high radiation level. So just to give you the brief summary of our experiment here. So if you look at different stages, so it has different stages. That is eggs, first instar larva, second instar larva, and feeding third instar larva, non-feeding third instar larva, and early pupal stage, late pupal stage, and male female. So if you look at all the stages, only at the early pupil we have some sort of death. So this is a stage where there is something called metamorphosis, like the pupa become completely into adult fly. So there are a lot of genetic, cellular events are happening. Otherwise, most of the stages are showing that very high resistance. So it clearly shows that the chitin is not responsible for the radiation resistance in insect, because larva and egg doesn't have the chitin covering, so only the adult flies have the chitin. So that's how we can exclude chitin from the. So the question is, what are the other molecules? What could be the other molecule which is responsible for radiation resistance? So we looked into one of the molecule called uric acid, because insect produce uric acid as a excretory molecule, but at the same time uric acid also acts as an antioxidant. So what we did is we used the genetic and pharmacological approach here. So we used two models. So normally the uric acid is synthesized from the hypoxanthine, and hypoxanthine is converted into xanthine, and xanthine is converted into uric acid. So this is controlled by two enzymes, that is xanthine dehydrogenase and xanthine oxidase. So what we can do is we can genetically block this enzyme. So that's what we did in the first one, that is called mutant, that is called ROSI mutant. We took one of the mutant, which is ROSI mutant, where xanthine oxidase is mutated. That means these flies cannot produce uric acid as expected like normal flies. And also we used another approach that is called pharmacological approach, where we used allopurinol. So allopurinol is one of the drug which is used by patient with gout, because to reduce the accumulation of uric acid in the gout patient. So we use these two approaches. That is, one is genetic mutant and there is allopurinol. Both the cases the xanthine oxidase is blocked. So that means there is no production of uric acid in these flies. And then what we did is we took these flies again to the gamma chamber. Then we checked in so we exposed these flies with like ROSI mutant, and also the flies treated with allopurinol, along with the wild-type flies, the normal flies with the proper amount of uric acid. So as you can see here, so these are coming from CS means controlled flies. So you can see CS untreated, ROSI untreated, and CS irradiated and ROSI irradiated. So these flies are irradiated with very high doses of gamma radiation, about more than 1000 gray. So as you can see from here, so the ROSI flies shows the early death compared to the normal ROSI, and also so after irradiation, the ROSI flies shows early death compared to the control and as well as the untreated ROSI flies. So we also measured the uric acid level in these flies. As you can see here, in the CS male and CS female, there is controlled male and female uric acid is very high, but in the ROSI male and ROSI female, the uric acid is lower. But when these ROSI flies are treated or exposed to radiation, they show higher death. That means uric acid could be one of the molecules which is responsible for the this radiation resistance. So again, we also use this pharmacological model that is using allopurinol. So again, the same result, we can see this flies treated with allopurinol, they die faster than normal untreated flies, that is controlled flies. So from these experiments, it is clear that uric acid could be one of the molecules. So we are not saying that it's only one molecule which is responsible for the radiation resistance. So, and also it is also known that uric acid is one of the antioxidant which was proposed earlier, so it could be one of the reasons. And so this was already published, and we are looking at like the molecular mechanism, how uric acid can induce this radiation resistance insect. At the same time, we also looked into another molecule that is called trehalose. So what we did is we measured trehalose, that is just by biochemical estimation, we measured trehalose. Trehalose is nothing but it's a stored glucose in insect, like just like in human, vertebrate, we have like glucose will get converted into glycogen. So glycogen is a stored glucose molecule, whereas in the case of insect, trehalose is a stored glucose molecule. Trehalose is a disaccharide actually. So there is also some report that trehalose also act as one of the antioxidants. So here what we did is we measured trehalose level from all the different stages with the biochemical analysis. As you can see again in the early pupal stage where we saw that early death in the radiation resistance experiment. So again, the early pupal stages shows again lower level of radiation resistance, or early pupal stages have started dying faster than expected. So now what we are trying to do is, can we manipulate trehalose level in Drosophila? So that's where the Drosophila genetic comes very important part. So in Drosophila again, so Drosophila also has insulin. So just like vertebrate, Drosophila also has insulin. So these insulins are expressed or released by cells called DLPs, or in the Drosophila insulin these insulins are produced by cells called IPCs, that is insulin protein cells. So what we can do is we can take a Calvert driver specific for these cells. Okay, so that what we can do is we can just kill this slice, and so that the insulin production will be reduced. Just like in the vertebrate system, in insect also insulin is responsible for converting glucose into trehalose. Just like in human, vertebrate system, it is the glucose will get converted into glycogen by insulin, but in the case of insect, it is the insulin-like protein will convert glucose into trehalose. So what we are currently doing is, interestingly, there are only about 8 cells in the entire insect Drosophila brain. So we can basically kill these cells using some of the genetic tool. So what you see in the reds, magenta, these are the cells which actually produces the insulin-like protein. So what we are currently doing is we are taking delete, that is the Calvert driver specific for this cells. So then it will be crossed with UAS delete to RNAi. This is RNAi line. So again, this is an advantage in Drosophila field. You don't need to induce any RNAi, just like a transformation or something. All the fine lines are available. You can just cross them so that you can reduce some of this protein. So in this slides, so these experiments are currently we are doing in the lab actually. So we already did some sort of estimation. So in these slides, the trehalose levels are completely almost 50% reduced, and now we are exposing these slides for high radiation in the gamma chamber. So believing that or hoping that these slides will also show some sort of radiation resistance decrease. So in summary, what we are trying to do is we are looking at all the use Drosophila as a model system. We already know that insects are highly resistant, but using Drosophila as a model system because it offers a lot of genetic tools. So where we can use this molecule in the longer run, for example, trehalose can be used as a cream or paste especially for the people who are working in the radiation facility, all those things. So that's what the whole idea. I think with that, I would like to close this talk, and these are the people the first three, that is Rashika Patil, Jagdish Paitankar, and Avinash Kudu are the people who are part of this work, and I would like to acknowledge all other people my PhD student, my collaborators, and of course we all always say that research doesn't need much funding, but we are very fortunate that to get a lot of funding from different funding agencies. So with that, I would like to close this talk and thank you very much.
Thank you, sir.
I just want to ask one question. About, do you have any experience in snail work? Like, we have in market nowadays snail, product from snail, which is used to prevent darkening of skin radiation. So, probably it also works, I think.
So it could be same like tree halos. So the people are already starting using. So most invertebrate produces tree halos. Like, so that's maybe one of the reason. So we don't know the exact mechanisms. But I'm not working with the snake. But so here for us, we are using genetics of reservoir to enter this question. But it could be due to tree halos.
Is available is available in market?
Yeah, it's available. It's a nice sector actually, so it's artificially, it's a synthesized.
Now let's move the conceptual radiation as well. Like to see that. Two questions. We're always focusing on the systemic level, the conceptual as a whole, but we do know that the insects at the cellular level are more resistant than the mammalian cells. So, halos, again, we are responsible for the differences in the radiation sensitivity at the cellular level, number one. Number two, how does this compare with lepidopteran cells? SF9 versus, let's say, Drosophila cells in vitro.
You mean cell line?
Yes. SF9
Yeah.
is a lepidopteran cell line. At the cellular level, it shows resistance as a cell to mammalian.
Yeah.
And this is one. The other one is within the melanoma acid, within the Drosophila. If you prepare a primary culture of the target cells and look at radiation response, this will be the whole. To what extent you think the halos would be the contributing factor for resistance?
Yeah, I mean, it's a very interesting question because, of course, we didn't look at the cellular level. So we can go to it individual tissue level, actually, because that's an advantage with Drosophila field. Like, we can reduce halos. I mean, we can block halos at the tissue level. Like, for example, in digestive tract, because we have all the driver lines available for. That's the beauty of Drosophila field. Like, so we have lacks of Galford drivers, so we can specifically look at. But this, of course, we initiated this work, but after that, we didn't get much funding from VRNS because VRNS thinks that radiation resistance is not very important now because radiation, they are thinking in a different way. So we didn't get much funding, but we are looking at this part, like at the cellular level. Yeah. So I mean, we should get some. I mean, VRNS should support us with this type of. This is your part of.
I cannot agree more than that. I can't do anything here.
Sir, we want to congratulate Dr. Shyam Prasad. Varija is our proud scientist of nation who got highest Ramalingam award. Give him a big applause.
No more questions? Okay, thank you very much.
Thank you, sir. I request the chairpersons to felicitate the speaker.
forward-looking session titled Epigenetic Silencing of the C14MC Cluster, a methylation-driven tumor suppressor in cervical cancer. The session will be delivered by a highly respected academician, Dr. Shyam Prasad. To chair this session, we are honored to have Dr. K. Kamalakshi Shenoy and Dr. Sukesh Rao.
I take the opportunity to request Dr. Shyam Prasad to go ahead with this talk on pediatric silencing of the C14 and C cluster. Over to you, sir.
Sir, please note, there will be 15 minutes and 5 minutes post that.
Thank you. First of all, I would like to thank the organizer, Sujata Madam, for the invitation. So what I am going to talk to you today about an ongoing work which we are undertaking at our institute. I am basically a biologist and works on a basic principles connected with how cancer develops and it progresses. So that is what we are working. And the title of my presentation is Epigenetic Silencing of Chromosome 14 microRNA cluster, and how this cluster helps in the development and progression of cervical cancer. So that is what I am going to talk to you today. So before I go into the details of what we do, so I would like to briefly introduce what is epigenetics. So many of us know what is genetics, right? So where we have mutations, deletions, insertions, copy number, variations, and all these changes are connected with diverse group of diseases, including cancers. And now these changes are being used as a markers for early diagnosis, then treatment prediction, and many other things, right? So now, like genetics, so there is another term called as epigenetics, and these epigenetic changes are equally important, and many of these epigenetic changes are in fact currently considered as an early diagnostic and prognostic marker, especially in case of cancer. So if you see epigenetics, the term epigenetics refers to changes in gene expression which is not associated with the changes in any DNA sequence. For example, DNA histone modifications, then DNA methylation and non-coding RNAs, especially microRNAs, PV interacting RNA, long non-coding RNA. So all these entities comes under the umbrella epigenetics. And in many of the diseases, like genetic alterations, they show epigenetic abnormalities. And these epigenetic abnormalities are connected with a wide spectrum of disease. For example, how these epigenetics are connected with the diseases. Actually, if you see a gene, for a gene to express, it has to be in a accessible condition. So you can see on the right side. So where the nucleosomes have to be placed far apart, and it is the underlying DNA sequence is accessible. Basically, it is a euchromatic condition, which helps in the gene expression. But in case of epigenetic modifications, what happens is that this open chromatin structure gets converted into a closed chromatin structure, and ultimately it helps in the shutting of the gene. Naturally, if you see X chromosomes, females have 2 X chromosomes. One of the X chromosomes is inactivated, and how it is inactivated? Because of epigenetic modifications. Okay, and what this epigenetic modification does is that it makes the region inaccessible for the transcription factors and the factors which are connected with the gene expression. That's how they are getting silencing of the gene expression. So I am not going to the details of it. So today what I am going to talk to you is about some of the research work we are doing at our institute with respect to microRNA. So as we know, microRNA is a small RNA, non-coding RNA, in the range of 19 to 25 nucleotides in size. So what they do is they are synthesized during the transcription, and they are transported outside the nucleus and in the cytoplasm. What they do is they go and bind to a target RNA, and thereby they bring about silencing of the gene expression. Okay, so how does it happen, or what happens in cancer? Like we have tumor suppressor genes and oncoproteins, we also have tumor suppressive microRNA and oncogenic microRNA. So basically, the tumor suppressive microRNAs are generally silenced in cancer, while oncogenic microRNAs are generally active in cancer. So that's how they promote, they help in the development and progression of cancer. So at Manipal School of Life Sciences, so basically I am coming from Manipal University. We are number one private institute or university in the country. And I am from a biotechnology school, Manipal School of Life Sciences, where we have an epigenetics lab, where we specifically work on epigenetic discrimination, then non-coding RNA biology, mitochondrial biology and metabolic reprogramming, then a little bit of translation research on how our basic findings can be translated as a biomarker in clinic. And we also work in the area of environmental carcinogenesis, where we look into bisphenol, phthalates and its role in cervical cancer. So that is what we do. So some of the publications which we have produced in the last 3 years I have shown here. And besides, we also got a patent on microRNA as a marker for early diagnosis of cervical cancer. So we got an Indian patent. So why cervical cancer? We know that cervical cancer is one of the biggest problems of India. And India and China contributes to about 35% of global cervical cancer incidence. And if you see cervical cancer and Indian context, India contributed more number of mortality cases when compared with China. Basically, India overtook China in terms of the number of deaths due to cervical cancer. So some of the factors which connected with this the high mortality is late diagnosis of the cancer when we talk about from a clinical perspective. And when, as a molecular biologist, when I speak, we don't know the complete set of genes and proteins which are responsible for development and progression of cervical cancer. So in this context, so what we are doing at Manipal is we look microRNA and how this microRNA and HPV interaction basically helps in the development and progression of cervical cancer. That is what we do. Okay. So you can see a series of papers which we have published from our lab related to microRNA and cervical cancer. So these papers are available online. So I'm not going to the details of that. So what I'm going to talk to you is, like protein coding gene, microRNA can also be regulated by epigenetic mechanisms. So when we did a small RNA sequencing in a panel of cervical cancer samples, which is provided by Mr. Sharan sir, who is sitting here. So he used to help me a lot with the clinical samples. So we took this sample and we got clinical samples and we did a small RNA sequencing. And what we found is that large proportion of microRNAs are downregulated. Now, since our lab works on epigenetic mechanism and there were not much report connecting the epigenetic regulation with the microRNA expression, we started working on whether the aberrant DNA methylation is responsible for the downregulation of microRNA. So we did a lot of experiments. We took clinical samples, then we did a mechanistic study using cell lines, and what we found is that, which is published in this paper, to show that many microRNAs can be downregulated because of the promoter hypermethylation. And we did the functional studies which I am going to talk to you in the subsequent slide. And then we wanted to see whether the microRNAs what we see in tissue, so whether the same thing can be seen if you profile from a blood sample. So we took the blood of cervical cancer patients and we did a small RNA sequencing. We found 60 to 70% of microRNA what we see in a tissue as aberrantly expressed are in fact downregulated or in fact shows a correlation with the small RNA sequencing what we have done from in the blood. So one of the potential markers we identified are listed here. So it is shown in the figure which I am going to explain in the subsequent slide. And then what we did is we wanted to compare our data with what is publicly available. We did a lot of bioinformatics analysis. So due to lack of time, I am not going to the details of that. But what I wanted to highlight is, in this context or in this bioinformatics analysis, what we identified is that a microRNA cluster, belonging to chromosome 14 is actually downregulated. So why microRNA cluster is important, I am going to tell you, I am going to talk to you. So this microRNA cluster, what I am going to talk to you, has two parts. One is cluster A consisting of 9 microRNA, and we have a cluster B consisting of about 42 microRNA. The beauty of this cluster is all these 52 microRNAs, they are downregulated in cervical cancer, co-downregulated. So what we wanted to understand is that, and another point what we identified is that even this microRNA cluster downregulation we are seeing in HPV positive samples more than HPV negative samples. And even in LSL state we are able to see the downregulation of this cluster. So we thought that the downregulation of this microRNA cluster may be very critical for an early transformation of a normal epithelial cells into a malignant one. Because when we did a many bioinformatics study, we also noted that this microRNA cluster can in fact target HPV, human papilloma virus. So it has a binding site in the HPV 16 and 18 proteins basically in the RNA transcript we are able to see. So that's why. So we thought studying this microRNA cluster is very critical, and we started investigating its function. So this is the expression of some of the microRNAs shortlisted from that cluster, and we can see on the panel A shows that they are significantly downregulated in squamous cell carcinoma samples. All these samples are clinical samples are HPV positive, and normal samples are HPV negative. We can clearly see that these microRNAs are downregulated, and then we compared our data with what is publicly available, and we identified a strong positive correlation. What we see as downregulated in our cohort of samples are in fact downregulated in the data which is coming out from DCGA. Then what we did is we shortlisted few candidate genes from this cluster and validated in an independent cohort of samples, which I have shown on the extreme other side. And where SCC refers to squamous cell carcinoma samples and NC refers to normal cervical epithelial cells. And we found a significant downregulation of that microRNA cluster. We did a lot of machine learning and artificial intelligence based screening, and we found when we use this combination of these three markers, so we are able to achieve approximately 0.9 AUC of 0.9. So that is what we are able to identify, and this data is published. And since our lab works in the area of epigenetics, and we are interested to look at the DNA methylation. So what we wanted to see whether this downregulation of microRNA cluster is in fact because of DNA methylation. So we did a lot of bioinformatic analysis, we mapped the promoter region, and then we did an experiment called as bisulfite Sanger sequencing. And what we showed is that, you can see here, I think this pointer doesn't show. So basically if you see the panel D, okay, so what we have shown is the methylation of different CBG sites in the promoter region of this microRNA cluster. So what we are able to see is that the promoter region of this microRNA cluster is hypermethylated. And then what we did is we did a lot of experiments to say whether this methylation in this region is connected with hypermethylation. And what we found is that when we treat them with a demethylating agents like pyrazosectin, we are able to activate the expression. So what we show with this set of experiments using clinical samples, cell lines, that the promoter region of this microRNA cluster is hypermethylated, and because of the hypermethylation, this microRNA cluster is downregulated. To understand further, so we wanted to see what are the transcription factors which can transcribe this microRNA cluster. We again did bioinformatic analysis, and our bioinformatic analysis suggested that MEF2A is a transcription factor which has the binding site in the promoter region of this microRNA cluster. And we are performing many experiments to understand like chip sequencing and chip PCR based experiments to experimentally prove that the region which is predicted bioinformatically indeed acts as the binding site of MEF2A. So basically we wanted to prove that MEF2A acts as the promoter region of this or transcription factor for this microRNA cluster. Now, as a biologist, my interest lies in understanding whether downregulation of this microRNA cluster has any impact on the development and progression of cervical cancer. So what we did is, since this cluster is a very huge, about 55 kb in size, so we used an approach called as CRISPR-based activation approach. We designed guide RNAs against the promoter region, and we did some lentiviral based transduction, and what we showed is that by inserting and activating sequences in the region where in the clinical we identified as hypermethylated, we are able to show that this microRNA cluster is actually getting activated. And so activation has a significant impact on the growth. You can see the panel C and the quantification data which is presented in the panel D, where we showed that when we activate this microRNA cluster, the number of colonies gets significantly reduced, suggesting that it might act as a potential tumor growth regulator. Then we did standard cell biology assays, like we wanted to see whether it impacts proliferation, migration, invasion. We did a lot of cell biology experiments, and we were able to show that this microRNA cluster in fact inhibit proliferation, growth, migration, and currently we are doing some more experiments to understand what are the other hallmarks of cancer which are basically impacted because of the overexpression of this microRNA cluster. And then what we also did some experiment is to understand whether this microRNA cluster has anything to do with the senescence, EMT and bit signaling, and antibody because my entire team works in this area, and these areas are also very important in the context of cervical cancer. So what we are able to show by our preliminary studies is that when we activate this microRNA cluster, there is a induction of senescence. We can see the markers like P21, P16, P21, they are all going up. And then we also see downregulation of many EMT and Wnt signaling markers like AKT, then CME, Vimentin are strongly inhibited in the presence of this microRNA cluster. We see lipid peroxidation suggesting a potential role in fibroptosis, and glucose uptake and lactate level, there is a fluctuation suggesting that there is a strong involvement of metabolic reprogramming. I have different Ph.D. students who are working in these aspects, so I am not going to present the data. And then we wanted to understand, so when we overexpress this microRNA cluster, so how does the transcriptome of the cell get affected? So we did a transcriptomic analysis and we identified a subset of genes. And when we did the bioinformatic analysis, what we found is that metabolic reprogramming is one of the, or the genes related to metabolic reprogramming is one of the most hit pathway. So some of the genes related to metabolic programming I have shown here. And what we found is that PDK3 as one of the target of this microRNA cluster. So PDK3 is a very important molecule, which is what it does is from the glycolysis to the Krebs cycle, that shuttle is done by the PDK3 gene. And we found that in TCGA and in our cohort of samples, PDK3 is upregulated. And conversely, what we found is that wherever this PDK3 is upregulated, our chromosome 14 microRNA cluster is also downregulated. And we did a lot of molecular biology experiment to show that the members of the microRNA cluster in fact targets the PDK3. And then we did the metabolomic analysis, and one of the metabolites what we identified is the palmityl carnitine. And we found that when we activate this microRNA cluster, what we see is that there is an upregulation of palmityl carnitine inside the cell. And we did a lot of experiments to understand what does this palmityl carnitine do. And we identified that palmityl carnitine is a tumor suppressive metabolite in a cervical cancer. So when we activate chromosome 14 microRNA cluster, so there is a metabolic reprogramming. So I have not presented the data, resulting in a upregulation of palmityl carnitine. And this palmityl carnitine has, is a very significant metabolite which can, in fact, by itself can induce a tumor suppressive function. So that's what we have published recently. And since cervical cancer, when we talk, it is a cancer of HPV. And we wanted to, and we got a very strong clinical correlation between HPV infection and the downregulation of microRNA cluster. We wanted to see whether this microRNA cluster in fact targets HPV, E6 and E7. So when it comes to E6 and E7, HPV, E6 and E7 are the two important oncoproteins. So I have shown only the binding site of 40 out of 42 microRNAs, how many of them are in fact targets the E6 and E7 are only represented here. And you can see what the bottom line of this slide is that when you have this microRNA, basically it targets the HPV, E6 and E7 proteins. So that is what we have shown here. And to summarize, so it's an ongoing study. So a lot of work is going on in our lab. So what we found is that this chromosome 14 microRNA cluster is a epigenetically downregulated microRNA cluster. The promoter hypermethylation basically governs the expression of this microRNA. And when we activate this microRNA cluster, it inhibits key characteristics of tumors like growth, proliferation, migration, invasion. And we identify the molecular targets, and one of the targets which we actively pursuing is BDK3. So we believe that HPV, chromosome 14 microRNA and BDK3 axis may be targeted as a for the clinical management of cervical cancer. So this is my lab. So these are the former members I have shown, and I have the other side I have the internal collaborators, international collaborators. These are my team, and I would like to thank Krishna Sharan sir for all the clinical support when he was at Manipal. So we got a lot of support, and we published so many papers with the help of him in the area of cervical cancer and epigenetics. And I would like to thank the funding for this study from DBT Welcome Trust, then from ICMR, SPAR and DST. Thank you.
Excellent presentation. I just wanted to know about your experience in cancer cervix screening, whether this can be used in that.
Sir, what we have not done extensive screening with the microRNA vectors. We did approximately, I think 50 normal, 50 pre-malignant condition and 50 tumor we have done. We are still in the process of doing that, but what we see is that even at LSL stage, they are all down-regulated. So there is a potential scope if it is screened in a large number of samples.
How expensive is it to patient for screening? Is it very expensive right now or can be?
So it is like if you are using a Tacman assay, so you know what is the cost. So we can bring down the cost by developing a low cost like a lateral flow based detection system can be developed as a future this one. So I have a ongoing Spark grant where we are trying to develop that model, where we are having a gold nanoparticle, where we have the probes for this microRNA to be captured. And by looking into the color change, we will be able to qualitatively tell whether the microRNA is down or up.
Question, what sample do you want from the screening patient?
So we can do with the tissue, we can do with the explorative cytology samples, we can even do with the blood samples.
Thank you very much. Any questions from the audience? So we close this session.
Thank you, sir. I request our chairpersons to felicitate our speaker.
Even our scientist, BUDDA, biggest Dr. Shyam Prasad has got Welcome Group Trust Award. So all of you give me PhD applause.
Thank you, Sir Srinivas. Our sincere thanks and deep appreciation to the chairpersons for steering this session with such poise and expertise. I request the dignitaries to please felicitate our chairpersons. Chairperson, Professor Dr. Sucheta Kumari A.
To lead and oversee this session, we have our two esteemed chairpersons, Dr. Prashant T. Shetty, Professor, Allied Health Sciences, Srinivas University, and Dr. Linsel Inda, Professor, Department of OBG, Srinivas Institute of Medical Sciences and Research Center, Srinivas University.
Ma'am, please note, we will be taking the first bell at 15 minutes and second bell was 5 minutes.
So good morning to all of you. Hope we are in time, and it is my pleasure to stand before in front of so many scientists and young scientists. This topic, this is till date we were discussing a lot about radiotherapy, chemotherapy and so many other activities, research factors. This is something new, this particular, as we know, food is the most forgotten medicine in our life. So how our food will be helping to prevent the development of cancer, this part I am going to discuss. So about introduction for cancer, everybody has spoke so many things. It's abnormal growth due to lot of development of various molecules expressions, cancer molecules. So the proliferation of the cells increases. So our aim is chemotherapy or radiotherapy or different immunotherapy is blocking these various types of factors, receptors, which enhances the development of the cancer growth. So, as we know, the fatty acids, it's a very, very important factor that how fatty acid is important in our life. If you know, our life starts with one cell, zygote. But if you see the structure of zygote, there are two layers of lipids, and that lipids are showing the fluid mosaic model movement only in the presence of unsaturated fatty acids and short chain fatty acids and medium chain fatty acids. When these fatty acid supply reduces in the diet, immediately the cell will become very rigid. The receptors may not work, or it will start giving wrong signals. So the signal pathway, how this fatty acid modify the various signal to reduce the various proteins which enhance the proliferation of the cells. That's a very important point we are discussing today. So as we know, today my talk is only on medium chain fatty acid, which contains 6 to 12 carbonated compounds. These fatty acids, we have to supply through the diet, and these fatty acids are mainly present in the natural source, especially in the coconut oil. So we are discussing how the compounds, the fatty acid, helps to prevent the proliferation of the cells. So these are already I have discussed, I have told you, we are going to first see the fatty acid levels in the case and control samples, especially in the serum. We are also collecting the tissues, and we are also working on the cell lines. So these are the, then we will conclude by using the docking system. So there is a gap, knowledge gap regarding the mechanism action of medium chain fatty acid on the oncogenic signaling induction pathways in breast cancer. You know, breast cancer will have mainly triple negative breast cancer is one of the dangerous one, which is unable to cure. So we have taken two cell lines also the two types of cancer patients sample. So the objective, we are going to determine the medium chain fatty acid in the both controlled and breast cancer case in the serum, and then we also see the expression expression of the genes which are responsible to regulation of the various receptors. And also we are analyzing the impact of medium chain fatty acid on the gene expression of various pathways. And then molecular docking we are going to analyze. So these are the various methods. You see that we collect the samples and also we collect the tissues for immunohistochemistry, and we analyze the fatty acid by using the GC, FID, and also we express the RNA and cDNA synthesis, and also we estimate the RT-PCR method, and also we use the in vitro studies with the two cell lines, one triple negative and ER positive cell lines. Here we have done different varieties of method to prove whether really it will help to control the growth of the various cancer cell lines. This is the in silico docking method we used here. Here when you see the result, that is clinical characteristic, when you see here, you can see here that is in a clinical studies in the clinical statements and all about the patient, where the tobacco consumption and alcohol consumption are more observed in the case. Plus cooking oil, we analyzed it. When I was studying this, I was in Nidde University. There are many cases, mainly from the North Karnataka side, and they use the sunflower oil mainly and the cooking oil, where you can see more cases observed, those are using the sunflower oil. But in the coconut oil, the case control is from Mangalore, where they use a lot of coconut oil. So this was control is using lot of coconut oil, and case is using so much sunflower oil. Histological type, if you see, invasive ductal carcinoma, you have nodular, but ductal carcinoma is very high compared to stage 3rd and 4th stage patients are very high. Lymph node status also is positive, and receptor status, if you see triple negative breast cancer cases are very high. Grade usually second grade is very high. And when we analyze the case and control for fatty acid, there are 3 fatty acids. One is caprinic acid, capric acid, and lauric acid. Here if you see the capric acid is mainly from the milk products. So that capric acid also bad in the case, which they are using a lot of milk products. So that is dangerous. Caprinic acid, lauric acid we find low in case compared to the control. So that 3 fatty acids we have observed, which one is comparatively high, which one is comparatively low to the control. And when you see here the percentage of plasma medium chain fatty acid in triple negative breast cancer cell lines, if you see caprinic acid is in stage 3rd and 4th it shows very low, and lauric acid in stage 1st it shows very low. So there may be some mechanism which we will discuss later. And percentage of plasma medium chain fatty acid based on receptor status, when you see the lauric acid is low in ER positive cases, but in the caprinic acid is low in the triple negative breast cancer cases. Then even the plasma mRNA expression studies of EGFR downstream signaling molecules we have done for EGFR, AK3 and M2, all are increased in case of cases. Then we have also collected the tissues and we did the histochemistry study. Then we observed the same expression, all these EGFR related molecules, proteins are increased in these conditions. Then the risk factor, when we calculated also by odds ratio, we observed capric acid, lauric acid, when it is decreased, the risk of developing cancer is very, very high. And this we have just found the LC50 for medium chain fatty acid. In both the cell lines we used, one triple negative and ER positive cell lines. And we have counted or calculated the requirement dose of this caprylic acid and lauric acid, because we have taken both only two, because they found to be the low in this case. So then we have used the two cell lines. One is triple negative cell line and the ER positive cell lines here. What we observed is when we treated with the caprylic acid and the lauric acid, the colony count is, colony formation completely decreased compared to the control. So the migration of cells also, this was inhibited the migration ability of the cell lines when we treated with the caprylic acid and also the lauric acid. Then this also we observed invading ability of breast cancer also decreased when you treated with the caprylic acid and the lauric acid. So these are the various methods we are using, whether our results which are found in the case and control syrup is also going along with the cell lines. This is induction of cycle arrest. The medium chain fatty acids, we observe the medium chain fatty acid, if you see in the MDA cell lines, the caprylic acid blocks the growth, and in MDA and lauric acid growth is synthetic phase, it will stop the growth. But in case of here, if you see the caprylic and the MCF cell line, the caprylic acid and the lauric acid blocks the G1 phase. So there maybe the mechanism is completely different. So when we see this apoptosis of the cells are in the late phase, it increased by, we have done this by using the flow cytometer, and we observed the late apoptosis is observed here. Then we also intracellular reactive oxygen generation. In normal level of lauric acid and caprylic acid, the development is normal. But as this concentration will increase with the medium chain, that is lauric acid and caprylic acid, reactive oxygen level production also increase. This also we observed in this case. And effect of medium chain fatty acid expression apoptotic related proteins we see also the apoptotic proteins, fat, asparagus and cateates, all asparagus and fat, these two proteins are increased, but anti-apoptotic BC12 is a decrease. So this shows caprylic acid and lauric acid has got very good role in regulation of the proliferation of the cancer cell lines. So down regulation of even EGFR signaling pathways also we observed. For example, all the EGFR signaling pathways, phosphorylated EGFR, M2, phosphorylated M2, all these are decreased in the treatment with the caprylic acid and the lauric acid in both the cell lines. So this is finally, we could not conclude in what way it blocks here. So we have gone to the docking system. This is what we observed, especially for the caprylic acid decreasing. It has to bind to a fatty acid binding protein, it's a protein that has two N-terminal, alpha 2 alpha 1 N-terminal N. This fatty acid goes and binds to the cap, and it blocks further entry of long chain fatty acids. For example, you know the omega-6 is a inflammatory marker, omega-3 is anti-inflammatory. So such a long chain fatty acid which increase the expression of EGFR related proteins. All that fatty acid expression of proteins and genes for the development of cancer is blocked when the fatty acid bind to the cap. This is what we observed in the fatty acid binding protein is blocked, and here the binding, the energy is very high we observed. Same thing in case of caprylic acid and lauric acid. When we see here, free fatty acid receptor 1, if you see there, here it is not binding to the top. It can't bind. It can bind in this place. And further expression of various requirement of proteins also blocked here. So the predicted model where for the study what we observed here, see the caprylic acid, lauric acid in breast cancer, when you supply the dietary supplement of both the caprylic acid and lauric acid, if you see here, the protein, fatty acid binding protein, with a long chain fatty acid when it binds, it will lead to the expression of EGFR related proteins and genes. But when caprylic acid increased in your diet, so what happened here, you see, it will block the binding of the all long chain fatty acid to the fatty acid binding protein, and it will lead to decrease the expression of all the requirement of proliferation or progression of the cancer cells. But when you see here, the dietary supplementation of medium chain and long chain fatty acids, what you observe, lauric acid and other side is unsaturated oleic acid. This free fatty acid receptor protein, normally it is very fast bind to the oleic acid and other monounsaturated and long chain fatty acids. But the lauric acid is especially antagonist to this fatty acid, so it binds to the free fatty acid receptor protein, so that further development of various related proteins which related to the EGFR proteins expression will be downregulated. So another mechanism what we observed is caprylic acid, lauric acid also increase the reactive oxygen and then it leads to apoptosis. Here also we observed that first along with the inflammatory markers protein, TNF alpha and all this nitric oxide synthase, interleukin, all that together they will be binding to this caprylic acid and lauric acid, increase the ROS level, and so that ROS also increases the antioxidant related genes it increases. Then the ROS mediated apoptosis of the cells increases. So this is generally what we observed is where we are not focusing on the short chain, medium chain fatty acid, how it helps to prevent the cancer. So this is the exact picture what we can observe in this particular studies. So in fact our findings suggest that we can see the dietary fatty acid non-toxic and excessive strategy to prevent cancer, and in addition that medium chain fatty acids are inhibiting cell proliferation, apoptosis, downregulating all the pathways. And our South Canada we use a lot of coconut which contain immediately virgin coconut or direct coconut which contain medium chain fatty acids, mainly caprylic acid and lauric acid. So the medium chain fatty prevent the, or it can be start using more in our diet than compared to the other carbohydrates. And another very beautiful thing here is medium chain fatty acid easily absorbed from the intestine. So it will be fast, it is very fast required for the energy requirement to the body. That's why if you take a lot of carbohydrates, it is the energy for proliferation of the cells. That's why we completely cut down the carbohydrates, especially the direct small molecules like glucose, sucrose and all that. You can take dietary fiber content carbohydrate and increase is the medium chain fatty acid, in the ghee, coconut oil and other. So that will absorb also fast and gives energy to the body so that we can control the proliferation of the any cells. Any body, any organ of our body, there will be a cancer cell or abnormal cell. As we control our diet, so that the proliferation definitely it will decrease or control. So this is my final, and this was supported by ICMR SR fellow for my Ph.D. candidate Dr. Prof, and she done all this work. So I thank to her and whole team. Our Srinivas University Research Center and Research team, and let food be the medicine and let medicine be the food. Thank you. So we have to think our food only, we have to convert into medicine by modifying the food. Because before we become human beings or we are super apes, where we used to live in the tree and eat only nuts and fruits. When we came down to the land, we become very lazy and started to eat cereals. And cereals contain lot of sweetness, and we can, if you go to any hotel, 90% of the product made by cereals. And we think alcohol, smoke or anything is drug addict, but addicted to the carbohydrate is also equal to drug addict. We can't avoid ice cream, we can't avoid chocolate, we can't avoid junk food, we can't avoid dosa, chapati, puri or anything. So if you see whole day, 90% of the diet is with this product. So we are also addicted to all these cereal products. So just like alcohol or tobacco. So it is better, we have to control the progression of cancer. It is by diet control. Thank you very much for the given opportunity.
Thank you for the nice presentation. It is open to the audience for any questions. Yes, there is a question.
Thank you for that lucid presentation. Does coconut supplementation of medium-sized fatty acids to the people who have undergone surgery to kill the micro metastasis?
So even one of my close, she was working in my lab. She had triple negative breast cancer, and with Dr. Vijit and all that, she overcome. Within no time, it has become reached to third stage also. But what I suggested since last 7 years is very healthy. I said completely stop taking any sugar items and eat with the ghee. Coconut milk you use for all these steamed vegetables, fruits. So she is very healthy without any progression. But doctor said the lifespan only 15 years more, and many of them I'm still helping her. You don't take anything, you go, and omega-3 you take, fish liver oil. So that all these prevent the inflammation. For any disease, progression is due to inflammation. So you have to suppress the inflammation by taking anti-inflammatory diet. So that all metabolic syndromes, cancer, everything will be under control. As per lot of work is going on lipids, you can make a 5 years course, because that much knowledge. Now if you go and just search for lipids in various fields, then more than that, at present the short chain fatty acid which is produced from the microbiota of the intestine is very good, which we have to develop the microbiota required flora. That is the dietary fiber is very important. It acts as a scaffold and it increases the production of short chain fatty acids. So these are very eye-opening things. Thank you, ma'am.
little digress into breast cancer. Will it help in the liver cancer patients?
Any cancer, any cancer.
One question I had. Is there any upper limit for this one, or
we take as a dietary factor, then it's not toxic.
because now the diet fad is that keto diet and stuff, and people are taking all these medium chain fatty acids or MCTs they are taking for weight loss and those things. So
tea is very good. We don't increase the weight. Weight is only by the sweet, because all sweet convert into a long chain fatty acid saturated. Saturated we synthesize by the help of this sugar, acetyl CoA, and become fat. So fat is obesity is due to carbohydrate, not by the fat.
My question was that if we are taking medium chain fatty acids or MCTs, what we use in our diet, so is there an upper limit for the usage, or
simply you should not take 1 glass. Whatever necessary with the diet. And another beauty is the coconut oil is saturated. It won't develop trans fatty acid while you cooking or frying. But other fatty acid contain unsaturated, immediately it develops trans fatty acid. Trans fatty acid is very toxic. There is a fatty acid transporter protein. It allows only transport trans fatty acid, not the other. So once again we will get into the problems. Thank you.
I have just a simple question. Are the doctors suggest this dietary thing to the patients who are in this cancer, along with their medications?
Now even doctors also know the diet because whole world 75% of the disease we can control by the diet, only 25% from the medicine. So hope everybody will suggest the diet modification.
They are suggesting on a regular basis. I don't think so.
It is depending on the doctor's doctor.
Thank you.
Thank you, ma'am. I request the chairpersons to felicitate our speaker.
Thank you, everyone. We now move to a cutting edge and fascinating session titled Psychosocial Burden in Cancer Care. The session will be delivered by Dr. Nitin S. Acharya and chaired by Dr. Prashant Shetty and Dr. Hilda.
Requesting Dr. Pramukh, Assistant Professor.
We would like to inform you all that our organizing chairperson has become the Vice President of Indian Association of Radiation Biology. Please give her a round of applause.
Congratulations, ma'am. I request Dr. Pramukh, Assistant Professor, Department of Research, to do the rapid
Greetings everyone, respected chairpersons and all the delegates who are gathered here. Firstly, I would like to thank the signing committee, in particular Vrinda ma'am, Sushita ma'am and Guru Prasad sir for giving this opportunity to share my viewpoints on my concept idea and psychosocial burden in cancer care. So you'll be wondering what is this elephant doing here? We have to address the elephant in the room. And what is meant to social, I'll be explaining in the next few slides. So I hope all of you are aware of what is this. Can anyone tell what is this? What is the disaster? Yes, this was the Chernobyl disaster which happened. So we are talking about radiation. So one of the disasters which happened in the early 1980s, which led to mortality and also morbidity in terms of cancer. But being a psychiatrist and also doing my PhD in psychology, we have to look at the positive side of the negative. That is called Srinivas positive psychology. So where we fall, we have to rise. So where this is nothing like when a patient receives a diagnosis of cancer, it's like dropping a bomb itself. So breaking bad news to the patient has to be done very carefully. And how the patient accepts it. The stages of cancer acceptance diagnosis is nothing less than a grief. So for this, the next few slides, how to make the person or patient and also the caregiver accept the diagnosis of cancer and walk through the stages of cancer is my concept of fictional psychology. So in this disaster, there was for any boon there is a bane, for any bane there is a boon. It's the yin and the yang, that is the Chinese philosophy. It goes like a cycle. So from this disaster, there was one young girl who was in Ukraine. She was a survivor of this disaster. She had to migrate to USA. So later she took up psychology. Then later she was one of the victims. She did not develop cancer, but she had migraine seizures, physical manifestation, and she had PTSD, post-traumatic stress disorder. So for this, she was looking for some source of inspiration because she was a child. So she was looking for some, she's very fond of any cartoons or anime. Nowadays, all of us know. If we ask who is the, say, the Attorney General of India, many people know what we know. But who is Iron Man, who is Doraemon, who is Shinchan, who is Spider-Man, everyone knows. So the kid wanted some source of inspiration and she was fond of X-Men. So in that she took the Storm character, and from that we have Janina Scarlett. She developed her own unique mode of therapy called superhero therapy. Those are from the psychology background, they know that currently we are in the 4th or the 5th wave of psychotherapy. So this comes under your metacognitive therapy and superhero therapy. So superheroes are shown as some source of inspiration, symbols of inspiration. You would have seen in Facebook, Instagram, Reels, everything that in the hospitals, especially for the kids, when they are going through the cancer stages of treatment, their favorite superheroes come dressed up as mascots and try to revive them both emotionally and also physically. Why they look up to superheroes is for that it is universal, irrespective of your background, your caste, creed, color, religion, it is accepted, it is seen, and without any bias it is known by everyone. So that is the power of fiction. So she took this concept and developed the superhero therapy. So the right side, based on her concept, I developed my own concept for which I applied the copyrights from the government of India and I obtained my copyrights in 2023. Based on that, I made my own concept called Samik Healthy Aging with Suskushan. So I developed my website, my YouTube channel, then applied for my under the National Medical Council advanced course in medical education thanks to Vrinda ma'am. So in that, I did my project with the MBBS students for inducing attitude ethics communication. Next, following this was the first step at the national level. Next, at the global level, I am doing my FEMA fellowship based on this concept with the medical students. So, because this conference is on cancer, I cannot present my findings about what I did with my students, but you can find it on my website. So this, when I was during my childhood days, this very beautiful concept which now we realize that any superhero has a mask. They wear a mask and we look for that source of inspiration. In this episode, which come through Foundation for Terminally Ill Children. So cancer need not be only for the elderly. We focus on the elderly. Even for the kids, my focus of interest, my PhD is in child and adolescent psychology. So here, the girl asked, she has a cancer, and she asked Spider-Man to reveal his mask. So no superhero is going to reveal his mask. No superhero, superhuman or no divine intervention is going to reveal the mask. But he does reveal his mask. The message conveyed here is that whenever for our loved ones as caregivers, we may be wearing our mask of different aspects, our work mask, our professional mask, our emotional mask, everything. We have to unmask ourselves and show our true self, our love, care to the people of cancer patients, to our loved ones. So we don't have to wait till the end moment, the last wish. See, everyone tells we have to fulfill the last wish of the person. How do you know that is going to be the last wish? Whatever they wish for. So you have to live in the present, you have to live in the present moment, you have to be mindful of that, you have to be resilient. So because psychiatry, psychology, spirituality has a overlay of that, and most of the researchers, they know that as you advance the stages of research and as you advance in the age, we become more spiritual by default, and that is how I got connected with Sucheta also during the discussion. So whatever spirituality is there, we can derive from any form of aspect. So this was my concept. So ultimately you have to unmask yourself to your loved ones, not only at the time of their terminal illness, but to your everyday life. So this is the message which I got, and this is the message which I will be delivering when I get a chance to do my research with the cancer patients. So because spirituality is an overlapping field, and the theme of this conference is convergence, and spirituality is something which is union, which is basically an integration of all aspects of life. So 6 pillars of holistic cancer care includes physical, emotional, mental, financial, spiritual and social. So my realm comes in emotional, mental and spiritual aspect. So spirituality is a very important aspect because acceptance of the illness and commitment, as I told, acceptance and commitment, a hero's journey through acceptance and commitment therapy. Unless the patient accepts that he has cancer, he won't be able to commit to improve himself or herself through the stages of cancer, no matter what stage of cancer he or she is. So even the WHO has felt the need for incorporation of the spiritual component in the definition of health. This was as early as in 2008. But because of the confusion between religion and spirituality, this is not incorporated. But there was an amendment which was proposed by Ph.D. report, where health is a dynamic state of complete physical, mental and spiritual, social well-being, and not merely the absence of disease or infirmity. So what does spiritual health mean? It is a state of being where an individual is able to deal with the day-to-day life in a manner which leads to the realization of one's full potential. So this realization, as per the Abraham Maslow's hierarchy of needs, self-actualization, which comes at the top level. So this actualization need not be only at your terminal age or your terminal stage of the illness. Early the better, because prevention is always better than cure. So spiritual health is state of being where an individual is able to deal the day-to-day life to lead to one's full potential, or even to die with one's full potential. You have lived your life and you have to accept that in stage, back stage of your life, and you have to exit with some form of grace. For that, spiritual health is very important. So for this, for the kids, for the children and adolescents, fiction is a very important mode and can be an universal thing, and that's what my concept of fiction psychology is, and fiction spirituality is. That's what your Mitsu social and Samic aging is all about. So use of spirituality, religion and rituals, whatever it may be. So it may be like your spiritual gurus or your spiritual beliefs, your Brahmakumaris or your Isha or whatever other backgrounds. Finding a meaning of not only the life also, but also finding meaning of the death gives an acceptance of the disease in the person, and that struggling with the disease will reduce. Ultimately, gracing the illness without disgracing it and embracing it with calmness is what resilience is what we need to teach to the patients, especially we from psychiatrists, psychologists or mental professionals we deal with. So this is from the nursing care, because nurses are in constant touch with the patients. They are very well aware of the spiritual needs of the patient, so giving their spiritual angle to the patients with cancer care will definitely, not only to the patient but also the caregivers, it's a soothing journey, and that what needs to be conveyed. So my topic is basically psychosocial burden in cancer patients and cancer care. For this I have taken two, one is an umbrella review, the other one is a meta review. So this is the umbrella review, that is psychosocial factors associated with quality of life in cancer care patients undergoing treatment. So as this is a rigorous and meticulous meta-review from 2012 to 2023, where nearly 3 lakh patients were studied across diverse cancer types, primary breast cancer, prostate cancer and gastric cancers, and quality assessment and rigorous selection was done. So here the quality of life, basically. So the quality of life, we say TALI in community medicine, disability life years. So that is affected by the cancer stage. So earlier the cancer stage, the quality of life is better. Earlier the intervention, the outcome, the prognosis is better. The treatment type, chemotherapy, comorbidities. See, not only the diagnosis, even the treatment itself is very disheartening, maybe like say demotivating to the patient and to the caregivers. So providing them the 6 pillars of care in that emotional, mental and spiritual care also has a very important role. Then coming to the symptom burden. So cancer specific symptoms like fatigue and pain. So pain, more than the physical pain, psychological pain also needs to be addressed, and that is where we pitch in. So most of the thing, the major impact of cancer is basically depression. So which is present across all the studies which were done, and also anxiety and distress. Resilience and optimism is something which needs to be built on, and this cannot be done only with the medication. It can be helped with the meditation also, and also with somehow dedication. For that dedication, I am little bit poetry also, so I write sometimes of my words. So for some resilience and optimism, some fictional, especially children, that will help. So coping strategies, active coping, problem solving, emotion focused, this we do in the therapies. Your emotion focused therapy, your problem solving techniques, your mindfulness based stress reduction techniques, all that. So the power of social support, strong social support, that's why we have support groups, because not only sometimes beyond the family we need help. So we have support groups, NGOs, you have hospitals, you have caregivers, therapeutic caregivers, everything, which needs to improve in the Indian setting as well. Then the family harmony. So once a cancer patient receives a diagnosis, the entire, the balance, the homeostasis in the family is disturbed. So providing psychosocial support not only to the patient, the family also becomes important. Where along with the individual therapy, family therapy also becomes important. So resilience, as I told, in the face of an adversity, how you grace it, that is what is your resilience. So that is what needs to be built on. And earlier we build on this, the better it is. This is something which is not taught in the syllabus in, say, in your primary school or elementary school, or later on only with the experience we learn this. By experiential learning, that is what is your humanistic psychology is all about. So key takeaways is treatment is a vulnerable time, not only diagnosis. So during a vulnerable time, when they are vulnerable, they need support. Secondly, we have to manage the depression. First, foster social connections. So, like this is one of the platforms where we can make such a social support groups and form a networking, where knowledge is the best form of, they say, RANA is the best form of what you can give someone else. So knowledge that can be delivered to the patients and to the caregivers and also to the therapists, that can become a change in the attitude which can be put into practice, KAP. So a holistic care approach where the holistic, as I told, the union, the integration has to happen at different realms at different levels. So this is about the psychosocial burden of cancer care in the patient population. Now coming to the caregivers, it's almost similar, there's not much of a difference. But see, we also have to address, in addition, one aspect is the stigma. It's like receiving a diagnosis of cancer in a family. Most of you, because you are from the medical background, you are seeing the patients, and some of you, at a personal level, may have experienced that. The stigma which is attached with the cancer, it's also very high. As such, with the depression, the psychiatric stigma is very high. If you add on the cancer stigma, it becomes much more higher. So when the problem is higher, the intervention also has to be on par with the problem. So what we can learn from the evidence of psychosocial support for carers of people with cancer, how do we advance our efforts, is a meta-review. So from this review, what critical role of cancer carers were basically the healthcare partners. The mental health problem basically is again depression, anxiety, and extensive responsibility. So here I would like to quote my favorite fiction cartoon, Spider-Man. With great power comes great responsibility. So with great, hurdle also comes great responsibility. So that responsibility first has to be taken care of by the patient himself or herself. Then, because our health is our responsibility. Next comes all the other caregivers. So that responsibility, sense of responsibility, resilience has to be built in the patients. So this was a systematic research, rigorous screening was done, and quality appraisal with data synthesis from 54 included reviews were done. So again, depression, anxiety were the prominent outcomes, and these were the ones which were addressed predominantly, because depression and anxiety is the most comorbid condition which happens with cancer or any other, say, terminal illness. So quality of life and psychological distress. So with these outcomes, whatever the interventions they were done, the intervention mainly focused on psychoeducation, counseling and supportive approaches, where 44% showed significant improvement, 11 out of 25 reviews, no significant change. As I told, therapy is talk therapy. So in that integration now in the 5th wave of psychotherapy, the new thing is about integration. That is the integrative medicine or integrative psychiatry or integrative treatment. In that, all the branches of medicine, not only allopathy, your Ayush also comes into picture, and there is a lot of scope for this integrative research to happen. So yoga also is a part of the therapy which is done in IMANS, where I was a part of that. So along with the talk therapy, other therapies also need to be incorporated with the ethical approval and all the other, keeping that into the background, and with the clearance. So only with these psychoeducation counseling supportive approaches, no significant change we have seen in nearly 16% of people, and unclear results in 40%. So less than 50% improved only with the talk therapies. So other forms of therapies also they need to be incorporated. The spiritual component, if we add on, definitely there can be improvement seen, and there are a lot of research for evidence for that also. So refocusing the research agenda, design studies to consider what works and for which carers to achieve specific outcomes, and embed theory-driven approaches, document and report theoretical basis, guiding interventions including carer-specific frameworks, and target-specific populations, develop carer-specific programs addressing unique needs of particular care groups. So there is a belief, so similarly there is a health belief model. What you believe is a cancer, what is a diagnosis like your diabetes, what is your belief of diabetes. Acceptance of diabetes is universal. There is no patient who doesn't accept he does not have diabetes, because that is something which is less treatment, and they have a health belief model. Similarly, their spiritual belief model or your social belief model, your cultural belief model, all that needs to be addressed. The belief is something which needs to be addressed, because that is going to change the mind or the attitude of the patients by giving the right knowledge. So we have to target specific the population for that. Enhance the implementation, focus on translating evidence into policy and practice, addressing barriers to reality. So finally, despite the need to ensure carers have access to programs supporting their well-being, the evidence to guide program development is complex and difficult to navigate. Refocusing on research efforts is definitely needed. So with that, I would like to end my presentation. Thank you.
Thank you, Dr. Vidya. It was a very enlightening talk. Not only psychological burden is not only for the patient, but as well as for the caregiver, and that was an eye-opener for us too. And as a clinician, yes, that was quite impactful for me as well, because I've gone through the phase. As you said, we also would have had somebody in the family, and the stigma is there. Right? So yes, that was really good. Thank you. The floor is open for questions. Anybody has any questions? Thank you, Doctor.
Thank you.
Thank you very much. May I request our esteemed chairpersons to felicitate the speaker. Appreciation to our chairpersons for guiding this session with their thoughtful leadership and academic presence. May I request the dignitaries to honor our chairpersons.
Sir, please join.
Thank you, sirs and ma'ams. We now move to a truly fascinating and futuristic session titled Nanomedicine, the Future of Healthcare. We are privileged to have with us Dr. Ramakrishna Shabraya,
To chair and guide this session, we are privileged to have two highly respected academicians, Dr. Shyamu Prasad K, Professor and Head, Department of Cell and Molecular, Manipal School of Life Sciences, Manipal Academy of Higher Education, and Dr. Satish Chitneni,
I request Dr. Videsh and Dr. Purnima to chair the session. Requesting Dr. Vijay Palini Swami, Department of Physiotherapy, Srinivas University, to do the rapporteuring for the session. Sir, kindly note, the first bell will be at 15 minutes and the second bell at 20 minutes.
Morning. Sir, I'm introducing Dr. Ramakrishna Shabraya, who is the Principal and Director of Srinivas College of Pharmacy. He's going to talk to us about nanomedicine and the future of the
Thank you for the introduction, and I take the opportunity to thank the organizing committee for giving this opportunity to be a part of this international conference. I am going to present a small topic, which is the dream of healthcare, that is nanomedicine, and I am going to touch only the theoretical aspects here. The main goal of any drug discovery or drug design, drug development or drug delivery, that is to achieve the improve the safety and efficacy of the treatment. But what is the problem, challenge in the existing treatment? The primary challenge in the chemotherapy, that is non-specific delivery of potent medicaments. There is a main limitation here, which leads to the side effects. Dosage, and consequently it reduces the efficacy and non-compliance. And another challenge is many of life-threatening diseases, particularly cancers and neurodegenerative disorders like cancer, where we are understanding the concept in the later stage, where it becomes more expensive and limited options for the treatment. In such cases, this nanomedicine has taken the chance, that is early detection and specificity, site-specific treatment that brings the transforming the dynamics in the healthcare industry. Then what is the nanomedicine? It is the process of diagnosing, treating and preventing diseases and relieving pain and of preserving and improving the human health using molecular tools and molecular knowledge to human body. What do you think of nanoparticles or nanomedicine here? Just for your imagination, this nano means 10 to the power minus 9, and this, just for your imagination, that a human hair is about 80,000 nanometer thick, and the protein size, that is 3 to 10 nanometer, and red blood cells approximately 6,000 to 8,000 nanometers. This just for imagination. When we are planning for the next nano devices, we should have the idea of what is the size range. And this what is the history of nanomedicine? The first time the nanotechnology word is used by Taniguchi, that is nanotechnology department of Japan, that is 1974. And the first book is published in 1909, and National Nanotechnology Initiative is launched in 2000. But if you turn the pages of Indian scenario, even from the ancestors, this practice was done, reduction in the particle size. Metals and the medicinal, just for medicinal use, during the time of Charaka and Samhita, they used these smaller particles. However, IS-30, that is usually a nano range. IS-30 is nothing but the treatment which was used for iron deficiency or anemia. Even other sites, in the cosmetics, it was used turmeric powder with oil or ghee as nanomycelics, which is absorbed in the body, and nanometallic and nanominerals. Even it was used in the Indian traditional treatment, that is the Loha Basma as metallic or Swarna Basma, Tamra Basma, Rajat Basma and Rasaka Basma, that's zinc carbonate. For different aspects, it was used in Ayurvedic system right from the beginning in ancient time. But what is the rationale for the nanomedicine now? In the development, what we expect? That is enhancing the drug stability and solubility, achieving specific site or drug delivery, and allowing sustained release and reduced side effect. Our major target is it has to reach to the specific site as well as with the minimum side effects and for a longer time, the continuous or controlled manner. If you are turning the use of nanomedicine, it boosts the stability, easy encapsulation, targeted therapy, reduce immunorecognition, reduce medicine degradation, increase solubility, targeted drug delivery systems, and easy in absorption, increase in shelf life. Finally, it reaches the purpose of improve the bioavailability. How it is applied? Nanotechnology applications, that is for the diagnostic purpose, for therapeutic purpose and immunization purpose. In diagnostic purpose, as imaging agents or for cancer diagnosis or for diagnosing the infectious diseases. And even in therapeutic applications for treating the infectious and non-infectious diseases, for cancer treatment and gene and stem therapy. What is the difference we observe in the current as well as nano impact? You can see that first one, there is screening stage, non-specific markers are used, but here in nano impact, specific markers are used. And in case of treatment, the surgery is done, but in case of nano, it is used mini-invasive surgery and always tissue-specific targeting. And in the follow-up, macro-scale site imaging, but in case of nano, non-invasive quantitative imaging. Come to the particles, nanoparticles here. It may be polymeric mesylate, dendrimers, nanospheres, or inorganic, there's gold nanoparticles, silver nanoparticles, and iron oxide nanoparticles. Based on the lipid-based liposomes and lipid nanoparticles, and it is used for diagnostic purpose as well as therapeutic approach, and now taken the interest of personalized medicine. In imaging, nanomaterials serve as a contrast agents, invasive medical imaging, that is common contrast agents, metal oxides, dendrimers and quantum dots. What are these quantum dots? These are the semiconductor nanocrystals made of nano-cadmium and nano-zinc, and it is used to tag biological molecules in medical diagnostics and applied in targeted therapeutics with a high drug screening. It is also used for diagnostic tool, that is the colloidal gold particles used in the rapid test for pregnancy, ovulation, HIV and other conditions of the conditions, and magnetic nanoparticles, that is iron oxide nanoparticles, shows promise in detecting Alzheimer's disease. In medical instruments, increasing the miniaturization of medical instruments using nanomaterials for better efficacy. Carbon nanotubes are alternative to glass pipettes for cell delivery. And nano silver coatings on catheters and other instruments for antimicrobial protection and wound dressing, incorporating the nano silver to prevent infections. Even it is used for therapeutic purpose, mainly to improve in the pharmaceutical applications, improve the drug solubility, reduce the systemic toxicity, enhance bioavailability and boost immunocompatibility and cellular uptake. Nanotechnology enables targeted drug delivery to control release at a specific site and dose. This improves the patient safety, patient compliance and reduces the side effects because even the dose can be minimized. These are released in the form of polymeric particles, dendrimers, nanoshells, ribosomes, mesylase and magnetic nanoparticles. In drug delivery using nanoparticles, that is polymer-based, dendrimers, chitosan and mesylate types, lipid-based nanoparticles with a liposome and neosome type, metal-based nanoparticles, gold, silver, cadmium and zinc sulfide and biological nanoparticles. Yes, you can see that it is used for the different purpose, that is drug delivery enhancement, topical infections, systemic infections, eye infections, lung infections and biological synthesis. Coming to the formulation type, just want to highlight some of the formulations used in nanoparticles. That is liposomes. These are the small spherical vesicles with the lipid bilayers closing with the aqueous core. These are used to drug delivery in the cancer treatment. Example, liposomes based on the amphotericin. Even to improve the stability of these liposomes, it is a pH gelation, which increases the molecular weight, offering the pharmacological benefits, which improves the drug solubility, protects the drug from proteolytic degradation, enhances the drug stability, once again extend the circulation time in the body, allows reduced dosage frequency without losing efficacy, and potentially of lower drug toxicity. Another formulation to overcome the challenges of liposomes, that is, liposomes are less stable due to the chemical instability, and phospholipids which are used in the preparation of liposomes, the purity issues arises, and due to high cost, it can be replaced with the neosomes. Neosomes are non-ionic surfactant vesicles, and these are used for the controlled and targeted drug delivery like liposomes and improve the drug penetration. One more formulation, that is dendrimers, which are synthetic molecules with a tree-like structure having multiple small branches around the central core. They measure about 2 to 20 nanometers with the branching originating from the core, and application of these are nano devices can be used in here for cancer cell recognition, diagnosis, treating the drug delivery, monitoring the level of tumors, and detecting the cancer cell death. That's a major advantage of these dendrimers. You can see that a single dendrimer can carry a molecule that recognizes the cancer cells, and a therapeutic agent kill those cells, and a molecule that recognizes the signals of this cell death. So it is more useful in case of cancer treatment for detection. Next one is nanoshells. Consists of silica core and metallic outer layer. These nanoshells can be attached to the antibodies to target the tumor cells. Near infrared light activates the nanoshell inside the cancer cells and generates heat. This heat selectively destroys the tumor cells without harming the nearby cells. But we can see here the healthy cells cannot be destroyed. Other advantages with the nanoparticles, we can minimize the pain, especially in case of insulin for diabetic patients. The frequent injection can be minimized if it is applied in the form of inhalation. There is a Pfizer and mankind, they already entered in this field. And drug delivery, that is a drug encapsulation. When it passes in the systemic circulation, during the transit, it can be protected by encapsulation. These nanoparticles in the range of 1 to 100 size range is used, and it will improve both the diffusion as well as degradation characteristics of the encapsulation material. And this used in the detection of Parkinson's disease. Drug discovery, the cost can be minimized because we know the drug molecule, the existing molecule, the efficiency or the efficacy can be improved converting into the nanoparticles. So the expenditure on the new drug molecule can be minimized. Even these are used in the devices, especially micro and nano-sized sensors can make use of wide range of technology that most effectively detect targeted chemical and physical property. It's use of polycrystalline glycol beads with the fluorescent material. When the beads are injected to the skin for the diabetic patients, when the sugar level comes down, automatically this glucose displaces in the fluorescent molecules and creates glow, which can be seen on the tattoo placed on the arm. And it is also used in the medical devices, which automatically delivers the medicine whenever it is required. Even it is used in the retina implants. For artificial retina uses the miniature video camera attached to the blind person's eyeglass to capture the vision signals. Even surgical reports. These surgical reports, doctors or the physician will use the thumb and fingers to move the joystick handles, but to control it is very difficult. If you make use of the robots here, the robotic system, even third hand, the third robot can be used to take the three-dimensional images, and it is more specific. Precision can be. Nanobots. These nanobots are the tiny nano devices which are used to repairing or detecting the targeted damage and infections. These are enabled to early diagnosis, which is the advantage. Which is not there in the conventional system, and targeted early system can be possible and easy for cancer detection. Used in the biomedical instrumentation and surgery. How it is? Acts a single inhaler nano robot reaches the replay into the tissue where it attaches and it swims like a fish. And we can get the information. Nanomedicine innovators have taken interest to enter this field. You can see that USA and Switzerland as well as Thailand, they invested huge and they can capture the market. Even in India, Biocon, Cadila, more than 100 companies, they started to invest on nano products. You can see that till 2024, $214 billion are invested, but it is expected in 2032 it is $507 billion. There is an expectation which indicates that the future market, the market will be captured by the nanoparticles or nanomedicines. Although the realization of the full potential of nanomedicines may be years of decades, recent advances in nanotechnology related to drug delivery, diagnosis and drug development are beginning to change the landscape of medicine. These nanotherapies could in the long term be much more economical, effective and safe, could greatly reduce the cost of current medical procedures. Hence, the design, development and delivery of nanomedicine will become a key for better health care if it is overcome the regulatory issues, social issues and ethical issues. Thank you. Size does matter. Wrong with bigger things bring better results. But fact today, small things, that is nano, brings better results today and will bring tomorrow. Size matters. Of course, it is nano size or small size. Thank you.
Now the session is open for questionnaires. Any questions from the audience? Any questions?
Thank you, Dr. Ramesh Chabra. It's a very interesting overview of the nanomedicine and nanotechnology. So I have one quick question. So are there any, still drawbacks that needs to be addressed, to bring this into wide usage? Like the distribution, let's say if you're targeting cancer, right? Is distribution of the nanoparticles or nanomedicine within tumor, is it still a problem or not? Because they are dependent on UVR, right? And hence permeability and retention.
Yes, that depends upon the phospholipids what we use, that also we can use. The drug targeting, the circulation, it can be, through encapsulation we can product the drug molecules during the transit period in the system.
Thank you, sir. Thank you. Sir, then we request our chairpersons to felicitate the speaker.
Thank you.
Thank you everyone. We now move on to a scientifically enriching session, unraveling the molecular impact of bisphenol A on PCOS through genomic and metabolomic integration. We are privileged to have with us Professor Dr. Padmalata Rai, and we request Dr. Purnima, Dr. Viresh and Dr. Satish Negi to chair the session. Professor Dr. Padmalata Rai, Associate Director, Academics and HOD, Department of Biotechnology, MSLS, Manipal Academy of Higher Education, Mangalore, India. Ma'am, kindly note that the first bell will be at 15 minutes and the second bell will be at 20 minutes for you. Thank you, madam. Good morning to all. And at the outset, I would like to thank the organizers for giving me this opportunity, especially Professor Sujata Madam for inviting me here to give my talk. And coming to my topic, I'm going to speak on polycystic ovary syndrome, which is a complicated endocrinopathy, which is unclear etiology. And bisphenol A is one of the endocrine disrupting chemical, which is also an estrogen mimic, and is most commonly used in the plasticizers that we use in our day-to-day life. There are a lot of reports saying the association of PPA and polycystic ovary syndrome, but the exact molecular mechanism of PPA and polycystic ovary syndrome is not clear. Hence, we have taken up this study to unravel the molecular mechanism of bisphenol on polycystic ovary syndrome through genomic and transcriptomic integration. As all of us know, polycystic ovary syndrome is an endocrine disorder that affects around 6 to 13% of the reproductive age women, and it is one of the leading causes which contributes to around 85% of the infertility caused due to ovarian dysfunction. And coming to the symptoms of PCOS, various reproductive outcomes such as infertility, polycystic ovary morphology, irregular period, insulin resistant, obesity, high risk of metabolic syndrome, and hyperandrogenism and psychological factors contribute to risk of polycystic ovarian syndrome, with a global prevalence rate of around 4 to 20%, and the Indian prevalence of 3.5 to 22%. This syndrome has become a major concern among the reproductive age group women. There are various factors that contribute to the risk factor, contribute as a risk factor for PCOS, which could include lifestyle, ethnicity, genetic, epigenetic factors, environmental factors, metabolic consequences, and geographic locations, where I'm going to concentrate on the environmental factors as to how they're going to have an impact and a specific focus on endocrine disrupting chemicals. Coming to what these endocrine disrupting chemicals are, they are the chemicals that interfere with the body's endocrine system and produce adverse effect on reproductive, neurological or immune system, and we are really exposed to various endocrine disrupting chemicals, which includes bisphenol, dioxin, PCBs, etc. And these substances enter into our body through ingestion, inhalation or dermal routes, which can cause severe health consequences like reproductive, developmental, neurological and immune disorders. Coming to the common sources of EDC, which includes pharmaceutical, synthetic hormones, food package and preservatives, cosmetics, clothes, solvents, etc. They contribute to the source of PPA which we are using in our day-to-day life. And bisphenol A is one of the most commonly used plasticizer, which is an endocrine disrupting chemical that binds to the estrogen. It is an estrogen limit, binds to the estrogen receptors and can affect the hormone balance. The various sources are listed here, which is common to all the MCs, and the tolerant limit, daily intake limit is fixed to 0.2 nanograms per kg body weight per day as per EFSA. And as of now, BPA is banned in baby bottles and infant formula cans, but then concern is about the other sources. So if you look into the metabolism and mechanism of BPA after it gets into the body is extensively metabolized in the liver by UTT, and if there is an impairment in the enzyme activity, it may lead to unconjugated or what we call as the free BPA, which may accumulate and get into the systemic circulation, and it may bind to estrogen receptors or disrupt the metabolic pathways or detrimental effects in steroidogenesis or alter the various gene expressions that are associated with polycystic ovary syndrome and other endocrine systems such as pituitary, ovary, pancreas and adrenaline. Although there are a large number of reports in the last 1.5 decades, but the exact molecular mechanism underlying BPA-induced PCOS remains unclear. Hence, this has prompted us to take up the study to know the exact molecular pathomechanisms that may drive PCOS-exposed BPA. This study comprises of 3 components. First, we carried out the in silico study to identify the hub targets and also to find out the interaction between BPA and the hub targets of polycystic ovary syndrome. And then we also carried out, to confirm the effect on the metabolism, we also carried out a case control cohort study where we identified the different levels of BPA in the urine sample and also did the untargeted metabolomics of the plasma sample. And then since the BPA exposure in humans is uncontrolled, we wanted to see the dose-dependent effect on the various phenotypes of BPA, and we carried out a mirror model study with controlled exposure of high and low dose and looked into the various phenotypes of PCOS. So I will address each one of them one by one. In the in silico study, we identified the hub targets and also by molecular docking, we identified the binding strength of the BPA to PCOS targets, and we identified 53 targets were common to BPA and PCOS, and all the targets were found to interact with each other. We also identified the hub targets of PCOS and BPA, and they were also found to interact with each other. And then we did the gene ontology to identify the process as well as the molecular functions that are associated with PCOS and BPA, and we found the ones which are marked in red boxes are the processes or the functions that are associated with PCOS pathogenesis. Similarly, we did the CAT pathway enrichment analysis to identify the various pathways that are associated with BPA impact on polycystic ovary syndrome. We did the molecular docking study with the hub targets, and the hub targets showed docking score above -5, which implicates that there is a strong binding of BPA with the hub targets. Further, as I already mentioned, we wanted to see whether there is disturbance in key metabolites in the humans. We carried out a case control cohort study where we enrolled around 120 cases and positive cases of PCOS and age and sex matched control samples. Sorry, sex is not a concern, only females are involved. And then we estimated the BPA levels in the urine and did the metabolomic analysis in the blood plasma. And this is the criteria we followed. We enrolled females between the age group of 18 to 45, and we followed the Rotterdam criteria. Who were positive were enrolled as cases, and who were negative with the same criteria were enrolled as controls, and listed the exclusion criteria who don't fit into the inclusion criteria. demographic hormone profile, glucose profile of the glucose and the control, and we saw that there was a significant difference between the age, weight, BMI, SBP, DBP, and also in the hormone FSH and AMH showed significant difference, although others showed difference were not significant among the cases and the control groups. We also looked into the menstrual features, clinical hyperandrogenism, uric BPA concentration in both cases and control, and we found that the menstrual cramps, heavy menstrual bleeding, infertility status were high in case of cases compared to the control, and FG score was high in case of the PCOS group compared to the control. Although total BPA and free BPA showed a difference in the concentration, they were not significant. This is a scatter plot showing the impact of BPA on metabolic pathways, and we can see that there is a difference in the three cases, three groups versus the control flow group. We also looked into the proto-metabolites of the three target groups versus the low BPA control group, and we found there was a significant difference in the distribution of these metabolites. Further, we carried out, although we could find a difference in the key metabolites in the high BPA and low BPA cases, we further wanted to confirm whether this is having other consequences on other phenotypes of BPA. We carried out a mirror model study where we exposed 6-month-old spread-out female rats to BPA of 2 dose, high dose and low dose for 90 days, and then we looked into the body weight, periodicity of menstrual cycle, hormonal profile, and also we looked into the histology of the ovary and the differential expression of the genes that are associated with PCOS, and also we did the mitochondrial dynamics and cells. So when we looked into the body weight, although there was increase in body weight over the 90 days, not show much significant difference, and menstrual cycle, we could see a difference in the proestrus and estrus cycle in the exposed group compared to the control group. We also looked into the free BPA and total BPA levels in the control versus the high dose and low dose exposed group, and we could see a significant difference between the control group and the exposed group. We also looked into the sex hormones such as FSH, LH, testosterone, estradiol and progesterone, and we could see a difference between the control group and the exposed group. Similarly, we looked into the ovarian morphology and looked into corpus luteum, apical follicles, antifollicles, and also cystic follicles, and they showed a difference between the control group and the exposed group. Further, we looked into the differential gene expression of the selected genes that are involved in ovarian steroidogenesis, estrogen receptor genes, and also mitochondrial fusion genes and biogenesis genes, which show variations between the exposed group and the control group. We also looked into ROA status and also senescent cells, and we found a difference in the intensity of ROS and also the percentage of senescent cell in the exposed group versus the control group. We also looked into the senescence associated markers, CDK and 2A protein level, and also this also showed a significant difference between the exposed group and the control group in a dose-dependent manner. To sum up my study, in silico study showed that the hub targets of PCOS showed high, strong binding affinity to PCOS, and our case control cohort study showed key metabolite variation in the low and high doses group, and the in vitro rat model study of BPA low and high dose exposure showed irregular estrous cycle, altered hormonal level, differential expression of gene, cystic follicles, and mitochondrial dysfunction and cellular senescence, which are all the hallmarks of PCOS. So it is SAI has come out with a draft on October 2025 for implementing complete ban of PTA, at least in the products which come in contact with the food. As of now, it's banned in the infant bottles and infant food cans. So it is of major concern, and hope the government will implement, or else it will have a serious consequence on the reproductive age and infertility. And I thank my collaborators, Dr. Shyama Prasad, and my clinical collaborators. Dr. Navya P. Prabhu has done most of this work for her doctoral thesis. Divya has helped us with proteomics work, and Vashishta has helped us with proteomics, and Supraja and Chinmay are putting forward this work at various levels, and also thank most importantly some of the participants of this study. And funding ICMR and Manipal Academy of Higher Education. Thank you.
Thank you very much, Dr. Bhutani, for giving a very extensive work on the agents which are going to affect the PCOs. I hope any questions from the audience. So let me ask one simple question. Say whether our lifestyle can improve use of all such products. Banning of all those whatever the agents which are affecting the PCO. Because any of our lifestyle, if we change our lifestyle, can be improved. Yeah.
We may have to say no to the sources. Unless government comes out with strong regulations, it may not be possible, because there are a number of sources which are already listed. Plastic containers, even the dental seals are containing contact lens. So wherever BPA or endocrine disrupting chemicals are there, those products have to be banned or taken care. Otherwise there will be continuous leaching which will get into the system.
Exactly, any dose is there which is going to affect some.
There is a permissible limit, sir, but then beyond that limit it is better.
Thank you.
Thank you.
Thank you, ma'am. That was highly thoughtful. I request our chairpersons to felicitate our speaker.
is known as an education hub. Many students from different places come to Mangalore just for study purposes. So just take a round around a famous private research and skill-focused university, that is Srinivas University. Srinivas University is the flagship of the 18 Srinivas group of institutions started by the A. Shamrao Foundation. It is a private charitable trust founded in 1988 by an eminent chartered accountant, Dr. A. RAGHAVENDRA. Proud son of Atka Shamrao, renowned chartered accountant and visionary academician, Dr. C.A. RAGHAVENDRA started the first educational institution, that is Srinivas College of Hotel Management, in the year 1988. Today, more than 13,000 students are pursuing their higher education at Srinivas University and Srinivas Group of Institutions. Dr. A. Srinivas Rao, Vice President of A. Shamrao Foundation and Srinivas Group of Colleges, Mangalore, is an eminent personality. The university has also recognized his contribution in managing higher education institutions effectively. The group has established many milestones in the quality education in the field of healthcare, medical, dental, paramedical, allied health sciences, hotel management, engineering and technology, physiotherapy, nursing, aviation studies, port shipping and supply chain management. The university has established networking with many industries, universities and education services providers to substantially improve the quality and weightage of the course and degrees respectively. Presently, Srinivas University has 9 institutes offering innovative industry-oriented specialized courses of UG, PG and research levels. The Institute of Engineering and Technology is a constituent college under Srinivas University located at Mukka, National Highway 66, Mangalore. The college campus is developed in a lush green environment with well-qualified and experienced faculty, state-of-the-art laboratories and research facilities in the field of engineering, technology and management science as per international standards, with the aim of creating technically sound engineers and decision makers who can establish themselves successfully in the industry and global market. Robotics, artificial intelligence and machine learning, cyber security and cyber forensic, data science, cloud technology and information security and blockchain technology and distributed computing. The College of Engineering and Technology is on the threshold of a quantum leap. Institute of Allied Health Science. Srinivas University serves with medical facilities too. The college's clinical requirement is fulfilled by Srinivas Hospital, which is a 700-bedded super speciality hospital. We have separate hostels meant for boys and girls. Srinivas University Institute of Nursing was established in the year 2019 with the commencement of the BSc program under Srinivas University, located at the Srinivas University campus, Mukka. The Srinivas Hospital is also located on the same campus and offers an excellent opportunity for students to be trained in a medical college hospital. The courses are well recognized by the concerned nursing councils. The Institute of Management and Commerce, earlier named as Srinivas Institute of Management Studies at Srinivas University, is committed to providing conducive learning environment. Institute of Computer Science and Information Science is envisioned to offer courses which would build the students into enthusiastic quality professionals in the field of computer science and information technology. Since 2005, The college is associated with several MNCs like Infosys Technology, Wipro, IBM, Accenture Technology, TCS, Tech Mahindra, Dia Systems, Spanios Technology, and many other in-placement activities. All-out undergraduate and postgraduate students get good placement through College Placement Program. Institute of Aviation is a constituent of Srinivas University. The college encourages the study of airlines, airports, and business related to the aerospace industry. This also includes supervising routine operations undertaken by the ground staff of an airport or an organization in the aviation sector. Institute of Hotel Management and Tourism offers a Bachelor of Hotel Management and Catering Technology and a BSc Hotel. These programs are focused on the aspect to create professionals who would be the future leaders of the hospitality industry. Practical exposure and experienced students will get from working in Hotel Srinivas and Hotel Saffron. Srinivas Institute of Physiotherapy was founded in the year 1993. From the year 2017, the college has become a constituent of Srinivas University. It is the first college which has designed the course curriculum to meet all overseas credentials. The college also has a wide network of alumni spread across the world in various managerial responsibilities. Undergraduate and postgraduate courses in physiotherapy are available on the Pandeshwar campus. Srinivas College of Pharmacy was established in the year 2004, offering both postgraduate, diploma and doctoral courses in pharmacy. The college is located at the Valachil campus of the Srinivas Group of Colleges with picturesque surroundings. The Srinivas School of Architecture, established in 2012 with a B.Arch degree under VTU University and is a part of Srinivas Group of Colleges. The school currently offers 5 years B.Arch program, Architecture Studio, Exhibition Space, Climatology Lab, Material Museum, Survey Lab, Model Making Studio, Specialized Classroom, and good canteen facility is the highlight of this campus. Srinivas Institute of Technology was established in 2006 with the aim of producing world-class technology graduates tuned to the demands of a fast-changing global village. SIT has gained the name as the best self-financed engineering college in Karnataka in terms of infrastructural facilities. Srinivas Institute of Technology is a unit of Srinivas Group of Colleges, Mangalore. 5 departments have been recognized as research centers by VTU to pursue Ph.D. and MSc. The institute has well-qualified, committed and motivated faculty. Mangalore is known as an education hub. Many students from different places come to Mangalore just for study purposes. So just take a round around a famous private research and skill-focused university. That is Srinivas University. Srinivas University is the flagship of the 18 Srinivas Group of institutions started by the A. Shamrao Foundation. It is a private charitable trust founded in 1988 by an eminent chartered accountant, Dr. A. RAGHAVENDRA. proud son of Atka Shamrao, renowned chartered accountant and visionary academician, Dr. C.A. Raghavendra Rao started the first educational institution, that is Srinivas College of Hotel Management, in the year 1988. Today, more than 13,000 students are pursuing their higher education at Srinivas University and Srinivas Group of Institutions. Dr. A. Srinivas Rao, Vice President of A. Shamrao Foundation and Srinivas Group of Colleges, Mangalore, is an eminent personality. The university has also recognized his contribution in managing higher education institutions effectively. The group has established many milestones in the quality education in the field of healthcare, medical, dental, paramedical, allied health sciences, hotel management, engineering and technology, physiotherapy, nursing, aviation studies, port shipping and supply chain management. The university has established networking with many industries, universities and education services providers to substantially improve the quality and weightage of the course and degrees respectively. Presently, Srinivas University has 9 institutes offering innovative industry-oriented specialized courses of UG, PG and research levels. The Institute of Engineering and Technology is a constituent college under Srinivas University located at Mukka, National Highway 66, Mangalore. The college campus is developed in a lush green environment with well-qualified and experienced faculty, state-of-the-art laboratories and research facilities in the field of engineering, technology and management science as per international standards, with the aim of creating technically sound engineers and decision makers who can establish themselves successfully in the industry and global market. Robotics, artificial intelligence and machine learning, cyber security and cyber forensic, data science, cloud technology and information security and blockchain technology and distributed computing. The College of Engineering and Technology is on the threshold of a quantum leap. Institute of Allied Health Science. Srinivas University serves with medical facilities too. The college's clinical requirement is fulfilled by Srinivas Hospital, which is a 700-bedded super speciality hospital. We have separate hostels meant for boys and girls. Srinivas University Institute of Nursing was established in the year 2019 with the commencement of the BSc program under Srinivas University, located at the Srinivas University campus, Mukka. The Srinivas Hospital is also located on the same campus and offers an excellent opportunity for students to be trained in a medical college hospital. The courses are well recognized by the concerned nursing councils. The Institute of Management and Commerce, earlier named as Srinivas Institute of Management Studies at Srinivas University, is committed to providing conducive learning environment. Institute of Computer Science and Information Science is envisioned to offer courses which would build the students into enthusiastic quality professionals in the field of computer science and information technology. Since 2005, the college is associated with several MNCs like Infosys Technology, Wipro, IBM, Accenture Technology, TCS, Tech Mahindra, Dia Systems, Spanios Technology, and many other in-placement activities. All-out undergraduate and postgraduate students get good placement through College Placement Program. Institute of Aviation is a constituent of Srinivas University. The college encourages the study of airlines, airports, and business related to the aerospace industry. This also includes supervising routine operations undertaken by the ground staff of an airport or an organization in the aviation sector. Institute of Hotel Management and Tourism offers a Bachelor of Hotel Management and Catering Technology and a BSc Hotel. These programs are focused on the aspect to create professionals who would be the future leaders of the hospitality industry. Practical exposure and experience students will get from working in Hotel Srinivas and Hotel Saffron. Srinivas Institute of Physiotherapy was founded in the year 1993. From the year 2017, the college has become a constituent of Srinivas University. It is the first college which has designed the course curriculum to meet all overseas credentials. The college also has a wide network of alumni spread across the world in various managerial responsibilities. Undergraduate and postgraduate courses in physiotherapy are available on the Pandeshwar campus. Srinivas College of Pharmacy was established in the year 2004, offering both under, postgraduate, diploma and doctoral courses in pharmacy. The college is located at the Valachil campus of the Srinivas Group of Colleges with picturesque surroundings. The Srinivas School of Architecture, established in 2012 with a B.Arch degree under VTU University and is a part of Srinivas Group of Colleges. The school currently offers 5 years B.Arch program, Architecture Studio, Exhibition Space, Climatology Lab, Material Museum, Survey Lab, Model Making Studio, Specialized Classroom and good canteen facility is the highlight of this campus. Srinivas Institute of Technology was established in 2006 with the aim of producing world-class technology graduates tuned to the demands of a fast-changing global village. SIT has gained the name as the best self-financed engineering college in Karnataka in terms of infrastructural facilities. Srinivas Institute of Technology is a unit of Srinivas Group of Colleges, Mangalore. 5 departments have been recognized as research centers by VTU to pursue Ph.D. and MSc. The institute has well-qualified, committed and motivated faculty. Mangalore is known as an education hub. Many students from different places come to Mangalore just for study purposes. So just take a round around a famous private research and skill-focused university, that is Srinivas University. Srinivas University is the flagship of the 18 Srinivas Group of institutions started by the A. Shamrao Foundation. It is a private charitable trust founded in 1988 by an eminent chartered accountant, Dr. A. RAGHAVENDRA. Proud son of Atka Shamrao, renowned chartered accountant and visionary academician, Dr. C. A. RAGHAVENDRA Rao started the first educational institution, that is Srinivas College of Hotel Management, in the year 1988. Today, more than 13,000 students are pursuing their higher education at Srinivas University and Srinivas Group of Institutions. Dr. A. Srinivas Rao Vice President of A. Shamarao Foundation and Srinivas Group of Colleges, Mangalore, is an eminent personality. The university has also recognized his contribution in managing higher education institutions effectively. The group has established many milestones in the quality education in the field of healthcare, medical, dental, paramedical, allied health sciences, hotel management, engineering and technology, physiotherapy, nursing, aviation studies, port shipping and supply chain management. The university has established networking with many industries, universities, and education services providers to substantially improve the quality and weightage of the course and degrees respectively. Presently, Srinivas University has 9 institutes offering innovative industry-oriented specialized courses of UG, PG, and research levels. The Institute of Engineering and Technology is a constituent college under Srinivas University located at Mukka, National Highway 66, Mangalore. The college campus is developed in a lush green environment with well-qualified and experienced faculty, state-of-the-art laboratories and research facilities in the field of engineering, technology and management science as per international standards, with the aim of creating technically sound engineers and decision makers who can establish themselves successfully in the industry and global market. Robotics, artificial intelligence and machine learning, cyber security and cyber forensic, data science, cloud technology and information security, and blockchain technology and distributed computing. The College of Engineering and Technology is on the threshold of a quantum leap. Institute of Allied Health Science. Srinivas University serves with medical facilities too. The college's clinical requirement is fulfilled by Srinivas Hospital, which is a 700-bedded super speciality hospital. We have separate hostels meant for boys and girls. Srinivas University Institute of Nursing was established in the year 2019 with the commencement of the BSc program under Srinivas University, located at the Srinivas University campus, Mukka. The Srinivas Hospital is also located on the same campus and offers an excellent opportunity for students to be trained in a medical college hospital. The courses are well recognized by the concerned nursing councils. The Institute of Management and Commerce, earlier named as Srinivas Institute of Management Studies at Srinivas University, is committed to providing conducive learning environment. Institute of Computer Science and Information Science is envisioned to offer courses which would build the students into enthusiastic quality professionals in the field of computer science and information technology. Since 2005, the college is associated with several MNCs like Infosys Technology, Wipro, IBM, Accenture Technology, TCS, Tech Mahindra, Dia Systems, Spanios Technology, and many other in-placement activities. All-out undergraduate and postgraduate students get good placement through College Placement Program. Institute of Aviation is a constituent of Srinivas University. The college encourages the study of airlines, airports, and business related to the aerospace industry. This also includes supervising routine operations undertaken by the ground staff of an airport or an organization in the aviation sector. Institute of Hotel Management and Tourism offers a Bachelor of Hotel Management and Catering Technology and a BSc Hotel. These programs are focused on the aspect to create professionals who would be the future leaders of the hospitality industry. practical exposure and experienced students will get from working in Hotel Srinivas and Hotel Saffron. Srinivas Institute of Physiotherapy was founded in the year 1993. From the year 2017, the college has become a constituent of Srinivas University. It is the first college which has designed the course curriculum to meet all overseas credentials. The college also has a wide network of alumni spread across the world in various managerial responsibilities. Undergraduate and postgraduate courses in physiotherapy are available on the Pandeshwar campus. Srinivas College of Pharmacy was established in the year 2004, offering both postgraduate, diploma and doctoral courses in pharmacy. The college is located at the Valachil campus of the Srinivas Group of Colleges with picturesque surroundings. The Srinivas School of Architecture, established in 2012 with a B.Arch degree under VTU University and is a part of Srinivas Group of Colleges. The school currently offers 5 years B.Arch program, architecture studio, exhibition space, climatology lab, material museum, survey lab, model making studio, specialized classroom and good canteen facility is the highlight of this campus. Srinivas Institute of Technology was established in 2006 with the aim of producing world-class technology graduates tuned to the demands of a fast-changing global village. SIT has gained the name as the best self-financed engineering college in Karnataka in terms of infrastructural facilities. Srinivas Institute of Technology is a unit of Srinivas Group of Colleges, Mangalore. 5 departments have been recognized as research centers by VTU to pursue Ph.D. and MSc. The institute has well-qualified, committed and motivated faculty. Mangalore is known as an education hub. Many students from different places come to Mangalore just for study purposes. So just take a round around a famous private research and skill-focused university, that is Srinivas University. Srinivas University is the flagship of the 18 Srinivas Group of institutions started by the A. Shamrao Foundation. It is a private charitable trust founded in 1988 by an eminent chartered accountant, Dr. A. RAGHAVENDRA. Proud son of Atka Shamrao, renowned chartered accountant and visionary academician, Dr. C. A. Raghavendra Rao started the first educational institution, that is Srinivas College of Hotel Management, in the year 1988. Today, more than 13,000 students are pursuing their higher education at Srinivas University and Srinivas Group of Institutions. Dr. A. Srinivas Rao, Vice President of A. Shamrao Foundation and Srinivas Group of Colleges, Mangalore, is an eminent personality. The university has also recognized his contribution in managing higher education institutions effectively. The group has established many milestones in the quality education in the field of healthcare, medical, dental, paramedical, allied health sciences, hotel management, engineering and technology, physiotherapy, nursing, aviation studies, port shipping and supply chain management. The university has established networking with many industries, universities, and education services providers to substantially improve the quality and weightage of the course and degrees respectively. Presently, Srinivas University has 9 institutes offering innovative industry-oriented specialized courses of UG, PG, and research levels. The Institute of Engineering and Technology is a constituent college under Srinivas University located at Mukka, National Highway 66, Mangalore. The college campus is developed in a lush green environment with well-qualified and experienced faculty, state-of-the-art laboratories and research facilities in the field of engineering, technology and management science as per international standards with the aim of creating technically sound engineers and decision makers who can establish themselves successfully in the industry and global market. Robotics, artificial intelligence and machine learning, cyber security and cyber forensic, data science, cloud technology and information security and blockchain technology and distributed computing. The College of Engineering and Technology is on the threshold of a quantum leap. Institute of Allied Health Science. Srinivas University serves with medical facilities too. The college's clinical requirement is fulfilled by Srinivas Hospital, which is a 700-bedded super speciality hospital. We have separate hostels meant for boys and girls. Srinivas University Institute of Nursing was established in the year 2019 with the commencement of the BSc program under Srinivas University, located at the Srinivas University campus Mukka. The Srinivas Hospital is also located on the same campus and offers an excellent opportunity for students to be trained in a medical college hospital. The courses are well recognized by the concerned nursing councils. The Institute of Management and Commerce, earlier named as Srinivas Institute of Management Studies at Srinivas University, is committed to providing conducive learning environment. Institute of Computer Science and Information Science is envisioned to offer courses which would build the students into enthusiastic quality professionals in the field of computer science and information technology. Since 2005, the college is associated with several MNCs like Infosys Technology, Wipro, IBM, Accenture Technology, TCS, Tech Mahindra, Dia Systems, Spanios Technology, and many other in-placement activities. All-out undergraduate and postgraduate students get good placement through College Placement Program. Institute of Aviation is a constituent of Srinivas University. The college encourages the study of airlines, airports, and business related to the aerospace industry. This also includes supervising routine operations undertaken by the ground staff of an airport or an organization in the aviation sector. Institute of Hotel Management and Tourism offers a Bachelor of Hotel Management and Catering Technology and a BSc Hotel. These programs are focused on the aspect to create professionals who would be the future leaders of the hospitality industry. Practical exposure and experienced students will get from working in Hotel Srinivas and Hotel Saffron. Srinivas Institute of Physiotherapy was founded in the year 1993. From the year 2017, the college has become a constituent of Srinivas University. It is the first college which has designed the course curriculum to meet all overseas credentials. The college also has a wide network of alumni spread across the world in various managerial responsibilities. Undergraduate and postgraduate courses in physiotherapy are available on the Pandeshwar campus. Srinivas College of Pharmacy was established in the year 2004, offering both under, postgraduate, diploma and doctoral courses in pharmacy. The college is located at the Valachil campus of the Srinivas Group of Colleges with picturesque surroundings. The Srinivas School of Architecture, established in 2012 with a B.Arch degree under VTU University and is a part of Srinivas Group of Colleges. The school currently offers 5 years B.Arch program, Architecture Studio, Exhibition Space, Climatology Lab, Material Museum, Survey Lab, Model Making Studio, Specialized Classroom and good canteen facility is the highlight of this campus. Srinivas Institute of Technology was established in 2006 with the aim of producing world-class technology graduates tuned to the demands of a fast-changing global village. SIT has gained the name as the best self-financed engineering college in Karnataka in terms of infrastructural facilities. Srinivas Institute of Technology is a unit of Srinivas Group of Colleges, Mangalore. 5 departments have been recognized as research centers by VTU to pursue Ph.D. and MSc. The institute has well-qualified, committed and motivated faculty. Mangalore is known as an education hub. Many students from different places come to Mangalore just for study purposes. So just take a round around a famous private research and skill-focused university, that is Srinivas University. Srinivas University is the flagship of the 18 Srinivas Group of institutions started by the A. Shamrao Foundation. It is a private charitable trust founded in 1988 by an eminent chartered accountant, Dr. A. RAGHAVENDRA. Proud son of Atka Shamrao, renowned chartered accountant and visionary academician, Dr. C. A. Raghavendra Rao started the first educational institution, that is Srinivas College of Hotel Management, in the year 1988. Today, more than 13,000 students are pursuing their higher education at Srinivas University and Srinivas Group of Institutions. Dr. A. Srinivas Rao, Vice President of A. Shamrao Foundation and Srinivas Group of Colleges, Mangalore, is an eminent personality. The university has also recognized his contribution in managing higher education institutions effectively. The group has established many milestones in the quality education in the field of healthcare, medical, dental, paramedical, allied health sciences, hotel management, engineering and technology, physiotherapy, nursing, aviation studies, port shipping and supply chain management. The university has established networking with many industries, universities and education services providers to substantially improve the quality and weightage of the course and degrees respectively. Presently, Srinivas University has 9 institutes offering innovative industry-oriented specialized courses of UG, PG and research levels. The Institute of Engineering and Technology is a constituent college under Srinivas University located at Mukka, National Highway 66, Mangalore. college campus is developed in a lush green environment with well-qualified and experienced faculty, state-of-the-art laboratories and research facilities in the field of engineering, technology and management science as per international standards, with the aim of creating technically sound engineers and decision makers who can establish themselves successfully in the industry and global market. Robotics, artificial intelligence and machine learning, cyber security and cyber forensic, data science, cloud technology and information security and blockchain technology and distributed computing. The College of Engineering and Technology is on the threshold of a quantum leap. Institute of Allied Health Science. Srinivas University serves with medical facilities too. The college's clinical requirement is fulfilled by Srinivas Hospital, which is a 700-bedded super specialty hospital. We have separate hostels meant for boys and girls. Srinivas University Institute of Nursing was established in the year 2019 with the commencement of the BSc program under Srinivas University, located at the Srinivas University campus, Mukka. The Srinivas Hospital is also located on the same campus and offers an excellent opportunity for students to be trained in a medical college hospital. The courses are well recognized by the concerned nursing councils. The Institute of Management and Commerce, earlier named as Srinivas Institute of Management Studies at Srinivas University, is committed to providing conducive learning environment. Institute of Computer Science and Information Science is envisioned to offer courses which would build the students into enthusiastic quality professionals in the field of computer science and information technology. Since 2005, the college is associated with several MNCs like Infosys Technology, Wipro, IBM, Accenture Technology, TCS, Tech Mahindra, Dia Systems, Spanios Technology, and many other in-placement activities. All-out undergraduate and postgraduate students get good placement through College Placement Program. Institute of Aviation is a constituent of Srinivas University. The college encourages the study of airlines, airports, and business related to the aerospace industry. This also includes supervising routine operations undertaken by the ground staff of an airport or an organization in the aviation sector. Institute of Hotel Management and Tourism offers a Bachelor of Hotel Management and Catering Technology and a BSc Hotel. These programs are focused on the aspect to create professionals who would be the future leaders of the hospitality industry. Practical exposure and experience students will get from working in Hotel Srinivas and Hotel Saffron. Srinivas Institute of Physiotherapy was founded in the year 1993. From the year 2017, the college has become a constituent of Srinivas University. It is the first college which has designed the course curriculum to meet all overseas credentials. The college also has a wide network of alumni spread across the world in various managerial responsibilities. Undergraduate and postgraduate courses in physiotherapy are available on the Pandeshwar campus. Srinivas College of Pharmacy was established in the year 2004, offering both under, postgraduate, diploma and doctoral courses in pharmacy. The college is located at the Valachil campus of the Srinivas Group of Colleges with picturesque surroundings. The Srinivas School of Architecture, established in 2012 with a B.Arch degree under VTU University and is a part of Srinivas Group of Colleges. The school currently offers 5 years B.Arch program, Architecture Studio, Exhibition Space, Climatology Lab, Material Museum, Survey Lab, Model Making Studio, specialized classroom, and good canteen facility is the highlight of this campus. Srinivas Institute of Technology was established in 2006 with the aim of producing world-class technology graduates tuned to the demands of a fast-changing global village. SIT has gained the name as the best self-financed engineering college in Karnataka in terms of infrastructural facilities. Srinivas Institute of Technology is a unit of Srinivas Group of Colleges, Mangalore. 5 departments have been recognized as research centers by VTU to pursue Ph.D. and MSc. The institute has well-qualified, committed and motivated faculty.
Evidence for the role in micro-autophagy. To deliver the session, we have Dr. Anup Kumar Ji. The session will be chaired by Dr. Ramakrishna Subramanian, Dr. Shyam Prasad Varijaragu, and Dr. Shashi.
Dr. Shashi Soames.
Yes, good afternoon. So it's my privilege to introduce Dr. Anup Kumar Ji, my friend. So Dr. Anup is a distinguished molecular biologist with over 18 years of research experience and currently serves as a professor at Srinivas Institute of Medical Science and Research Center, Bangalore. As a principal investigator here, he leads multidisciplinary project involving CRISPR genetic engineering on focal and confocal microscopy, proteomics and metabolomics with a strong focus on therapeutic targets and diagnostic markers. He oversees a team of 9 researchers including Ph.D. scholar, a postdoctoral fellow and a master's student. His technical expertise include CRISPR, knockout and residue studies, stable cell line preparation, flow cytometry, immunostaining, side-directed mutagenesis, plasmid cloning, trap, protein purification, immunoprecipitation, and ultracentrifugation. He also has extensive experience with animal handling and advanced imaging techniques such as cranial nerve, cranial window surgery, perfusion fixation, and intracranial viral inoculation. He is proficient in software tools such as ImageJ, FlowView, Metamorph, Reacto, ShinyGo, and Metabol Analyst. Dr. Anup has held position as Assistant Professor at Nearby Research Center, Scientist Grade 2 at KSAT, Medical Academy Research Associate at ICET Remember, and Akira Foundation Fellow at Dickens PSI Japan. He completed his PhD at Tokyo Medical and Dental University and previously served as Project Assistant at NII New Delhi. His research contribution includes high-impact applications such as NO article with over 500 citation and paper in autophagy and scientific review. His academics includes the prestigious Next Japanese Fellowship, All India Rank 1 and the Takeda Foundation Fellowship. With this brief introduction, I would like to invite Dr. Anup for his talk.
per day, then probably our cleaning of our house may be in a very low rate, that we don't have time to clean our house. Then if we get a Saturday, Sunday holiday, we can involve in cleaning the house. And suppose we don't get a holiday, over a period of months, lot of food may accumulate in the fridge and the clothes may accumulate. So same thing happens inside a cell. If cell will not get a break from its duties, it cannot clean up. But if we get a break from this metabolic load, the food processing load, for at least for 1 or 2 days, the cell will start cleaning. So that's all about autophagy. Autophagy is an intracellular cleaning process. We can generally divide it into 3. One is autophagy, which always happens at a very basic level, unless you are fasting. And that can appropriate autophagy. It is all about double membrane structure forming inside the cell. It's a liquid bilayer that can engulf damaged, misfolded, mutated proteins or damaged organelles, and then it can fuse with the lysosome, chop up the components and recycle into basic molecules of glucose, amino acids, nucleotides, lipids, and it can be reused in a cell. And there is another chaperone-mediated autophagy where it can specifically degrade proteins, and a very less studied macroautophagy. Okay, so microautophagy is the lysosome itself or the latent ozone can invaginate and take up small particles. Okay, and it is the most understudied among all the autophagy field because of the lack of markers or the mechanistic side of it, the enzymes regulating that. Then again we can classify into non-canonical or non-specific autophagy, which randomly takes up anything, or selective autophagy, which includes mitophagy, which is a topic of our interest this time. So mitophagy is all about when your mitochondria is damaged, immediately some enzymes come there and make sure that it is removed through a specific form of autophagy where the autophagous membrane engulfs just the mitochondria and fuse with the lysosomes. And studying autophagy is extremely important, of course, because if you try to stop autophagy, animals can develop tumors. And in the brain, we can see that the increasing accumulation of ubiquitinated misfolded proteins, which can cause deep and neurodegenerative diseases. And one thing common among autophagy inhibition is the accumulation of damaged mitochondria. The left one is a healthy mitochondria, you can see intact crystal. Right side you can see the damaged mitochondria. And a bit of mechanistic side. Once the mitochondria is depolarized or damaged, ubiquitin ligase called parking will rush to the spot. Then this is an E3 ubiquitin ligase, so it can ubiquitinate and tag several proteins on the mitochondrial membrane by ubiquitination tag. And this ubiquitination tag attracts autophagosome formation exactly on the mitochondrial membrane. So the autophagosome will eventually engulf that specific mitochondria and fuse with lysosome, where everything will This is a very classic image from my colleague, Dr. Chikokishikabura. Here you can see the double membrane structure of autophagosome engulfing damaged mitochondria in a parking overexpressed condition. That's the condition. Unnaturally created condition of parking overexpression. When we started working on mitophagy 13 years back, we used carbonate cyanide. It was the most popular and most effective to use mitophagy. And here you can see we added carbonyl cyanide and whole cell mitochondria is depolarized. This is the cell, here is the nucleus. What you see in color is mitochondria. 6 hours we wait, nothing happened to the mitochondria. 12 hours we wait, 24 hours we wait 48 hours. All the mitochondria is still there. It's not going anywhere. It's not removed by any autophagosomes. And we were shocked to see that because it's a well-established stuff and there are more than 12,000 papers that damaged mitochondria is removed by mitophagy. So you can see the Western blot images of inner mitochondrial membrane protein complex 3, absolutely no change after mitochondrial damage, even after 48 hours. And if you artificially overexpress parking, the story is very different. After 6 hours and 12 hours, you can see mitochondria is getting removed. And over a period of time, all the damaged mitochondria is removed in 12 to 48 hours, as you can see here. Only under artificially overexpressed parking condition, which we do not have in most of the cell lines we are checking, FA4, HeLa, MEF. So, this is just my personal conclusion. The damaged mitochondria cannot be removed unless you artificially overexpress parking. In MEF cells, FA4 cells, HeLa cells, RANA cells. Then, if you overexpress parking artificially, you can see that mitochondrial proteins are going down, which means the damaged mitochondria is removed by autophagosome, is fused in the lysosome and degraded. If you inhibit autophagy, the 5100 knockout, this will not happen. As you can see here, the complex 3, the inner mitochondrial protein do not change. But these change, that is just proteasomal degradation by parking. So, I conclude part 1 of our experiments that damaged mitochondria cannot be removed according to the experiments done in our lab. Then, did we miss the parallel players or the actual players of mitochondrial quality control in the shadow of this my personal wish to study mitophagy? But one thing I always observe during my PhD is whenever there is mitochondrial damage, some tiny vesicles, lipid membrane vesicles come nearby. This is damaged mitochondria. Immediately, like 10 minutes or like 1 minute after damage, these endosomes are there. And I have confirmed it with live imaging microscopy. So I added carbonyl cyanide, red long are mitochondria, they are not yet damaged, and the green is endosome, multivascular endosomes. Immediately they are there, within 30 seconds. They form some kind of bangles around the elongated mitochondria, and after that, these green endosomes leave, and then comes the parking. So it's like there is an accident on the road, immediately some people stepped in, took the patient to hospital, and then they just left. You don't find them anymore. Then some other main players come. But we never observed these immediate action team in our autophagy process. Then I was really interested to know why these endosomes come there immediately. Mitochondria is not removed even after 48 hours of damage. All the mitochondria are intact and proteins are intact. But endosome come there, does something, definitely does something, and just leave. So we were very curious to find out what it is doing. I wish to play this video. It's the same movie. You can see that. Okay, can you repeat? Yeah, can you repeat? See, it may form like a bangle around. Okay, one more time. Sorry, repeat. Yeah, let's go. You see that? And this was consistent in all the movies we have taken. Next slide. Again, a hierarchical classification. Red is mitochondria, this is endosome, and top is Barking. Now let's see a time scale. After adding carbonyl cyanide, mitochondria is damaged. Mitochondria is still elongated. Then as you move in time, 5 minutes, you can see endosome coming there. 10 minutes, endosomes have surrounded mitochondria. And then until 40 minutes, endosomes are doing their job. Parking comes later, after endosome leaves. So, endosome has a half an hour job to do something with the damaged mitochondria. I don't know if it is trying to repair or is trying to do something else. Let's see. This another movie which shows that the green endosomes and the red mitochondria, the endosomes are dropping something and it just comes there, it goes back. So, endosome come, it's a kiss and run model, again it go back. Again come, again it go back. So some continuous activity keep on happening. We try to bleach the damaged mitochondria area. So this is damaged mitochondria and we bleach the green color which is the endosomal area. Within seconds you can see the tubes of endosomes are shooting to the damaged mitochondria. Here this is very clear. Endosomes shooting towards damaged mitochondria. So endosomes associate with the damaged mitochondria. I'll skip this video. So many people have it out. Is it endosome? Is some kind of artifact happening? So we did the electron microscopy, and again, this was very clear that it's not just an endosome. It's a multivascular endosome, multivascular body. You can see several multivascular bodies inside this endosome, and it is closely associated with the damaged mitochondria. And all these are confocal images of the same. This elongated mitochondria is trying to take up something, maybe just the mitochondria. Again, it all repeats the same passage, and you can see one thing. There is no autophagosome in the whole story. We did not observe any autophagosome coming near a damaged mitochondria in these cells. Only endosome come there. So let us settle with this hypothesis that endosomes rush to the spot whenever there is a mitochondrial damage. And up you can see elongated mitochondria, lower is a fragmenting mitochondria after mitochondrial damage. So, one side there is a parking story happening where ubiquitin ligase come to mitochondria, autophagosome come and engulf mitochondria, fuse in lysosomes. And now we have a new story running parallel. So I want to check how they are going simultaneously. So here the green rings you see are parking circling the damaged mitochondria. And what we were talking about is an entirely different area. This is the whole cell. And multivascular endosome and mitochondria are in another space. Parking and mitochondria is in another space. So two different things parallelly happening And when these results came, the entire team was so excited because in 2014, if this was reported, it was the first to show that endosomes can engulf or there is a parallel pathway going on along with mitophagy, because that time mitophagy was in the upcoming peak. And now I repeated the experiment and we see one problem there. Everybody was happy with this, that mitochondria is engulfing the endosome. Everybody was happy except me, because I saw one problem there. There is a gap in this endosome. And mitochondria, if it is a mitochondria, it was supposed to enter completely, but it is not there. Then I came up with this hypothesis that it is just a mitotracker, not the entire protein or lipid of mitochondria. Mitotracker, and there is endosome. Once there is mitochondrial damage, mitotracker just moves out into the endosome. Then you don't see the mitochondria anymore because this was the frozen type. So actually there is mitochondria here, but you cannot see them. So it's not a mitochondrial engulfment or mitophagy, it is just the metabolite soup of mitochondrial matrix with mitotracker entering into the endosomes. Then this was my model. After mitochondrial damage, endosomes comes there, then it just takes some part of mitochondrial fluid and just goes. Maybe infuse with lysosome and do exocytosis or do something else. Parking is an entirely different pathway. There is parking or expression. This can go for mitophagy, but two different pathways. And this experiment clarified all our confusions. Here is mito-BFP, which is a protein tagged with a glufosin protein inside mitochondria, engineered. And here is an endosome in red, and there is a purple color is mitotracker dye. And mitophysin is there in the junction. So you can clearly see mitochondria do not have the mitotracker after damage. Mitotracker went into the endosomes. So mitotracker dye went inside the endosome. This is just like, and I again confirmed with Tom 20 staining, which is again a mitochondrial protein. We have put some chloroquine so that we can see a larger vesicle. All these mitochondria in red, these are not mitotrackers, actual mitochondria, because it's Tom 20 staining. And here is the endosome vesicle. None of them enter inside. Only mitotracker can enter inside the endosome. The mitochondria itself cannot enter inside the vesicle unless you overexpress parking artificially. Here you can see endosomes in red, mitochondria in blue, parking in green. All are genetically stable, engineered in the chromosomally integrated genes with fluorescence. Only if you overexpress parking artificially, which do not happen in your body, you can see this kind of models. Otherwise, mitochondria is just like you are in a bus and bus had a breakdown. Then you are the passenger, you just come out of the bus, take a cab and go home. Mitochondria is a bus and passengers are the metabolites. Once it is damaged, they just go out. Then now the bus can be repaired. Once it is repaired, you can start the metabolism again. So this is my theory on mitochondrial quality control. And all this started when I was working on ATG2, cloning and functional character. And we always observe endosomes coming near mitochondrial membrane. And now I had a very logical theory that endosomes just fuse with autophagosome membrane to enlarge the endosome, the autophagosome membrane to engulf a large mitochondria. But now I think that this is not correct. And there are 12,000 papers on mitophagy, which is an established fact. So mitophagy is also happening in our body, but not in a measurable, detectable limit of within few hours or within one day. And we have this Western blot that none of the mitochondrial proteins reduce after mitochondrial damage. And this is the present hypothesis I have, that damaged metabolites are removed into the endosomes so that the mitochondria can repair. Maybe. So I wish to thank Srinivas University Management, Professor Sucheta, Director, Research of Srinivas University, all the encouragement last 3-4 years of support, and the entire team in Srinivas University who has worked behind all this, Dr. Praveen, Dr. Ranjita, Dr. Rupa, Dr. Ashwini, Dr. Divyarani, Mithun, Mundaman, Deva, Naveen, and my students, Rahat, Sandra, the rest of the team. And this I wish to thank the professors who invested a lot of time in training me. It's a Japanese group where we started working on autophagy, Yoshinori group. Then I was with Professor Murthy Srinivasan at ICTM, and this is a group who continued working on this endosome-mediated project. Rishi, Nikhil, and Dr. Kishi and Ravis encouraged me. And Dr. Santosh Kumar from RGCB for the super resolution images. And Professor Paul Workaday from UK. And Dr. Sahani Mayurbai, my colleague from University of Goa. Thank you so much.
Thank you very much for this interesting talk. I hope there will be a few questions from audience. Okay. If there are no more questions, so thank you very much.
I don't think so. It is present, but I
Yeah, so I just want to know how was like the first discovery?
Parking was overexposed.
Yeah, but it was.
Then it was done by Richard Hughes.
Yeah, so but it was discovered as a natural process, like mitochondrial. So I just want to know how it was like.
I feel that some cells in our body, they have parking levels sufficient to induce a very minimal mitochondrial degradation through mitophagy. Okay, but when it comes to cell lines, they don't have so much parking especially to support mitophagy. So it exists, but not in the cell lines which I observe. That's it.
How was the impact of autophagy on aging?
Yeah, it's a very important question. Now, I personally feel that if cleaning process is accelerated, cells are more healthy, can be maintained over a longer period of time. So it has a very significant role. A lot of studies are done. Yeah.
And is there any markers you have, autophagy and aging markers?
Autophagy and aging markers, thank you for that idea. Now I will search for that. So we have around 200 markers of organ aging. Each organ's aging, which are the markers. Now we want to say what are the autophagy specific markers. Thank you for that idea.
IPR is mine.
Yes. Thank you so much.
Thank you.
Thank you, sir. I request the chairpersons to felicitate our speaker. sirs and ma'ams. Moving forward to the next session titled Advanced Directives End-of-Life Care Policies in India. I proudly announce the speaker for this session. It's none other than our organizing secretary, Dr. Brinda Jacob. I request Dr. Ramakrishna Shabraya, Dr. Shyam Prasad Varija Raghu and Dr. Sashi to please chair the session.
Good evening, everybody. I was very sure that today's session will also get prolonged like yesterday, so I had confidently put good evening, but looks like we are maintaining time today, so good afternoon. So today we have come to the fag end of the conference deliberations, and my topic is Advanced Directives, End-of-Life Care Policies. At this very juncture, in fact, interestingly, I remember the first slide of our first keynote address by Dr. Nishita. If I'm not wrong, this was the slide she had put. That cancer is not a battle to be won. It's a journey which begins, and we have to, walk through that journey. So I just talked to her and I said, can I please borrow your slide? Because with this slide, I'm going to talk and say that we also need to think about the end. Because yes, I definitely did not choose this, but we have to think about the dilemmas that come across each and every person taking care of those who have come to the fag end of their journey of life, whatever way it is. So healthcare, as we all know, it is a collaborative effort. Many people are involved. It is not alone the treating physician, the nursing faculty, the pharmacist, the other caregivers, everybody, the hospice care, everyone is involved, the lab investigator, everyone is involved. But talking to the patient party is the foremost important aspect of the end of life care. It is very, very vital that we tell them that it is important that we are with you in this final phase of your life's journey. But at the same time, as Dr. Mithun had rightly said, there is so much of psychological burden on the family, on the treating clinician and the caregivers. So how do we, take care of those final moments? The ethical dilemma that comes is on the family and the doctor when to pull the plug. Am I right in pulling the plug? The person on whom I am going to pull the plug, he is my father, he is my brother, he is my mother, she is my mother. We have played together. They have reared me. How can I pull the plug? Am I doing the right thing? What will people say tomorrow? When to pull the plug? Remember, pulling the plug may be legal, but we have to decide whether it is ethical or not. When to decide it is ethical and who tells us it is ethical. And all this began with a case where this beautiful lady who went unacknowledged when she was alive, and she was sexually assaulted and became a vegetable for almost more than 4 decades of her life. And a lady activist, Pinky Virani, fought to give her the end of her disgusting life of 4 decades. And finally, in the year 2015, she was given an end for her vegetative state by the court. That particular case became the history or a historical landmark in Indian end-of-life care policy. From there on, drafting started, and we today have a beautiful advanced directive, or the living will as we have renamed it, decision-making policies for terminally ill patients. So in that particular judgment in 2018, the Supreme Court said the said directive will dispel many a doubt at the relevant time of need throughout treatment of the patient. It will strengthen the mind of the treating doctors as they can ensure, see the Supreme Court was damn sure that they said the treating doctors can ensure without any doubt in their minds that after being satisfied that there is nothing else left for them to do, they are acting in a lawful manner. Because that is one thing the doctors are always scared of, that the legal sword is always hanging on their head. What happens if I, advise the patients to pull the plug? Tomorrow can they, go and complain in the police station and put me behind bars? Who is going to protect me? Ethically I may be right, but legally I may be wrong. Who is going to protect me? So here comes the directive which is going to help all the clinicians and all the people involved in this end-of-life care. It simply says life sans dignity. Life without dignity is not worth a life at all. It is an unacceptable defeat. When life meets death with dignity, it is a value to be. Nobody wants to have a death of or an undignified death. We all, even today, I would expect to die this moment. If death comes, I would like to drop dead now, rather than lie on the bed for days together, months together, years together, tormenting my family and all those people, financially, physically draining them, mentally draining them. We all want that. That means we all want a dignified death. Nobody wants that undignified death. But who is going to help us? All these years we were saying, no, euthanasia is wrong, nursing is wrong. Yes, active euthanasia is wrong even today. But here, we are not calling it an active euthanasia, we are calling it a passive euthanasia. And this landmark judgment was given on 9th March 2018, the end-of-life care in India. And this was added as an act in MCHA, that is Mental Health Care Act, or it was also called as Mental Health Care Regulations 2020. Though it was added in the Mental Health Act, it was included for all the disorders or diseases as a final end-of-life care. So most of the terminologies here may be added as mental health, but it is applicable to all the disorders. So please bear with it. So it is for, and it is an advanced directive, an application which can be filled by anyone. Usually after 40 years is what they say, but there is no limitations for the age. Anyone can apply for it. And there is a particular protocol for it, that you can either apply it freshly. I'm just going to give a brief of it because most of us do not know about it. Believe me, even the clinicians are not aware of it. So that is why I'm just going to give a brief of it. There is an application Form A. If you go into your Google or any search engine, you will get this. You just put MHCA Act and MHCA 2020 and you will get this Form A. There are multiple forms, Form A, Form B, Form C, but for us Form A is sufficient. There are all kinds of diseases involved there. You can go on ticking which disease when you have, if it is, you don't want to be treated, you want to be, not to be treated and simply to be left untreated, but only to be treated for pain and just left alone, all those things. But I'm going to come to it. Without consultation of a doctor, please do not do it. because you may not know the consequences of the same. So it is to be submitted. There is absolutely no cost. Initially when it was implemented, it was required to be notarized and all those things. There were a lot of hassles, but it was revoked in 2023. Now there is no such thing. It is totally free of cost. It can be done any number of times. You can change it. It is something like your will. You can keep changing it. The final one is taken into consideration. And if there is a nominee, because there has to be a nominee, just when it is to be implemented, if the person who has put this advanced directive, if he is incapable of implementing it, then there should be a nominee who has to implement the living will, tell them what is there, where is the will. So a nominee has to be put up, so that nominee has to sign in this, that I am accepting to implement this living will. That is the only criteria here that has to be there. And like a will, two witnesses have to sign. Now there is a board here to which it has to be submitted. This board is, one is the judge who is the chairperson, the district judge, then the DC or the collector or the magistrate, two members, one psychiatrist and one local doctor, and then two members like one of the caregivers association or an NGO representative, anyone, two other people. So totally 6 people, they are the members of this board. And then there is a medical board, the hospital in which the treatment is being taken. There the treating physician, subject experts, 2 subject experts, and a secondary medical board. When the decision has to be taken, a secondary medical board comes into picture with the chief district medical officer and the subject experts who are not part of the treating team. So that becomes the final board who will take the decision whether it is the right time to implement the advanced directive or not. So much of care is taken. Just because the advanced directive is there, they will not simply pull the plug. It is not taken like that. The decision has to be taken unanimously by multiple people. So enough care is taken by the government while implementing this law. So every application for an advanced directive, it has to be signed by 2 witnesses like a will, and it has to be attested by them saying that it is signed by the person who has written the directive. Okay, and then it is submitted to the board and no fee charged, and it is made available to the person who has applied and also to the person who has been nominated, so that he can have a copy for himself. And no unauthorized person or media or anyone is supposed to be having a copy of it. No one else can have a copy of the same. That is what I said, that no copy should be. And then only one restriction about the changes. That is, once a directive has been changed, there is a moratorium period of 3 months. For a period of 3 months, you cannot change the will. Once the 3-month period is over, then if you want, you can change that advanced directive further. Till that 3 months period, it cannot be changed. That is the only important thing. So now, the important thing that we need to know is, who are the people who can give or who can make this advanced directive? Anyone with a sound mind, who is an adult, that is, who is more than 18 years, to communicate, relate, comprehend the objectives. He should be aware of the consequences of the advanced directive. He should be able to be of sound mind and should be able to know what will happen if there is withdrawal of treatment, withholding of the treatment, what happens. So all those consequences are to be known. That is why I said it should be done in consultation with the doctor, the treating doctor. Alright? Most importantly, it should always specify the name of the guardian, because without which implementation of the advanced directive becomes difficult. Now, if sometimes it happens, we keep on making, we have seen it in movies also. There will be so many people, even I have a will, which will is the final, the handwritten which is dated properly with witnesses, that will be taken into consideration. Completely valid one. That means there is witnesses, there is name of the implementer or the guardian nominated. That will be, or that advance directive will be considered the final one. Now, if there is no living will, because it is happening in currently, I have not come across a case in this Karavali region where there is a person who comes with a living will. If anyone here, a practitioner, a clinician, treating doctor has come across, please let me know your experience, because I have not come across anyone here. So people are unaware of it. In such cases, what is our duty? In such cases, how do we guide them? What do we do about it? That is very, very important. What do we do? We form a medical board, and I'm telling this out of my personal experience, because recently I lost a very close family member from a terminal cancer, and there was so much of confusion. Even though I had given DNR means do not resuscitate. In the enthusiasm that I am a doctor and in the Josh, they forgot about it, and when she went into cardiac arrest, they revived her. They revived her, and not only revived her, put her into ICU. Next day she was sitting and drinking tea. And she is having terminal cancer. What do I do now? And now her husband is having a doubt. What do I do? Should I continue with the treatment? Should I leave her like that? Because we had decided that this case is absolutely hopeless. It's over. And next day she is sitting in the ICU bed and drinking tea. It is a sad state that in another 2 days she left us, because she deteriorated after that. But because of that DNR, she just sat on the bed the next day morning, drank tea. And look at the state of the mental health of the family. Till then they had totally accepted that she is going to leave them, and next day morning they are seeing her in a totally different picture. Despite of the fact that in bold letters on her file, it was written DNR. Can we sue them? No, because probably multiple factors were affecting. First, they were not used to it because all these advanced directives have not been strongly implemented. And then the patient's relative is a doctor, so they wanted to impress probably. Multifactorial things happening around. So when we look at them, even I was confused whether did I take the right decision of DNR, the way I saw her sitting and drinking tea. So we have to be very, very careful and we have to see that even when we are the treating doctors, our nurses implement the orders that are being given at no condition. Well, she was sitting and drinking because of the CPR only, because of the defibrillator, because her heart became a little strong. So these things can affect the mental health badly, very badly. So that is why formation of a medical board, actually the previous evening the medical board sat together, explaining the situation. We took and then the DNR was taken just a couple of hours before she had the attack. That is the sad part. So then, within 48 hours, it has to be, opinion has to be framed, and in fact in this case within an hour or two we had come to the decision of DNR in this case, because the case was very bad. So this medical board will be formed, as I told the treating doctor and 2 other doctors from the same hospital, and 1 outside doctor is always preferred for a general consensus and unbiased consensus. So when initiated DNR orders, now remember here, when you give DNR orders, it doesn't mean you stop the treatment. Remember that DNR means do not stop the treatment, just don't resuscitate. People have that misconception, DNR means stop all the treatment. No, DNR simply means do not resuscitate. Continue with the treatment. If the person goes into an attack, do not resuscitate. That's all. Okay, so Normal care, normal treatment continues. So suggestions for end-of-life care and decision making. In the absence of the directives, a well-wisher, this is the problem. A well-wisher might refuse to take the right decision because of the emotional attachment, the flashbacks, and not able to take the right decision at the overwhelming the burden that they have over the loved ones. So it is not the time for them to take the burden of the loved ones, rather take effective decisions, because it is not only the emotional burden here, it will be a financial burden also. Remember, because already the treatment has cost them a lot of financial burden. Just keeping them on ventilator and the cost of treatment itself is shelling them out. We have to think about that also. Every day is shelling them out. We have to think about those consequences also. They have to live their own life after the end of the life of their loved ones. So giving that consideration also, the doctors have to help them come to the right decision. So reflect on the goals, values and beliefs. No, it's not a one-time event. Meet repeatedly, talk to them, let them go back and take their own meetings and talk to each other, come back, make it as have several meetings, multidisciplinary teams if they want psychiatric help or psychological help or whatever type of help they want, give it to them and discuss all the types of alternatives that are needed for them. And then finally, it is my humble request to all the clinicians and the treating doctors, well, I don't know how many of them are currently present here, but it is a sincere request to all of you that when a patient, when you are treating a patient, please make them aware of this advanced directive. Make them aware that there is an entity called as advanced directive, so that their loved ones will not be in a dilemma. Their loved ones will not end up being crushed under the burden of the emotional guilt and the financial burden of having to take a untimely decision of whether to sustain life or to end the life of their loved ones. So on that note, I say start with the end in mind, so that we give our loved ones a happy ending and a moment to celebrate our deaths. Thank you very much. I wish to acknowledge Blue Maple document prepared by Dr. Naveen Salins of Kasturba Medical College, Manipal, and his entire team, from where the healthcare institutions have taken the end-of-life care policies, and it has been added as an advanced directive policy by the Government of India. Thank you very much.
Is there time for discussion? Any questions?
Congratulations, madam, for a vibrant talk. We are really proud of you. Throughout the 2 days, you struggled and gave a very nice end speech. Thank you so much.
Thank you, ma'am. I request the chairpersons to felicitate our speaker.
All of you join. Come, all of you. Senior staff and everybody.
Now we'll break for tea for 15 minutes. I request all the delegates to assemble back at 3:15 for panel discussion.
Mangalore is known as an education hub. Many students from different places come to Mangalore just for study purposes. So just take a round around a famous private research and skill-focused university, that is Srinivas University. Srinivas University is the flagship of the 18 Srinivas group of institutions started by the A. Shamrao Foundation. It is a private charitable trust founded in 1988 by an eminent chartered accountant, Dr. A. RAGHAVENDRA. Proud son of Atka Shamrao, renowned chartered accountant and visionary academician, Dr. C.A. RAGHAVENDRA Rao started the first educational institution, that is Srinivas College of Hotel Management, in the year 1988. Today, more than 13,000 students are pursuing their higher education at Srinivas University and Srinivas Group of Institutions. Dr. A. Srinivas Rao, Vice President of A. Shamrao Foundation and Srinivas Group of Colleges, Mangalore, is an eminent personality. The university has also recognized his contribution in managing higher education institutions effectively. The group has established many milestones in the quality education in the field of healthcare, medical, dental, paramedical, allied health sciences, hotel management, engineering and technology, physiotherapy, nursing, aviation studies, port shipping and supply chain management. The university has established networking with many industries, universities and education services providers to substantially improve the quality and weightage of the course and degrees respectively. Presently, Srinivas University has 9 institutes offering innovative industry-oriented specialized courses of UG, PG and research levels. The Institute of Engineering and Technology is a constituent college under Srinivas University located at Mukka, National Highway 66, Mangalore. The college campus is developed in a lush green environment with well-qualified and experienced faculty, state-of-the-art laboratories and research facilities in the field of engineering, technology and management science as per international standards, with the aim of creating technically sound engineers and decision makers who can establish themselves successfully in the industry and global market. Robotics, artificial intelligence and machine learning, cyber security and cyber forensic, data science, cloud technology and information security and blockchain technology and distributed computing. The College of Engineering and Technology is on the threshold of a quantum leap. Institute of Allied Health Science. Srinivas University serves with medical facilities too. The college's clinical requirement is fulfilled by Srinivas Hospital, which is the 700-bedded super speciality hospital. We have separate hostels meant for boys and girls. Srinivas University Institute of Nursing was established in the year 2019 with the commencement of the BSc program under Srinivas University. located at the Srinivas University campus, Mukka. The Srinivas Hospital is also located on the same campus and offers an excellent opportunity for students to be trained in a medical college hospital. The courses are well recognized by the concerned nursing councils. The Institute of Management and Commerce, earlier named as Srinivas Institute of Management Studies at Srinivas University, is committed to providing conducive learning environment. Institute of Computer Science and Information Science is envisioned to offer courses which would build students into enthusiastic quality professionals in the field of computer science and information technology. Since 2005, the college is associated with several MNCs like Infosys Technology, Wipro, IBM, Accenture Technology, TCS, Tech Mahindra, Dia Systems, Spanios Technology, and many other in-placement activities. All-out undergraduate and postgraduate students get good placement through College Placement Program. Institute of Aviation is a constituent of Srinivas University. The college encourages the study of airlines, airports, and business related to the aerospace industry. This also includes supervising routine operations undertaken by the ground staff of an airport or an organization in the aviation sector. Institute of Hotel Management and Tourism offers a Bachelor of Hotel Management and Catering Technology and a BSc Hotel. These programs are focused on the aspect to create professionals who would be the future leaders of the hospitality industry. Practical exposure and experience students will get from working in Hotel Srinivas and Hotel Saffron. Srinivas Institute of Physiotherapy was founded in the year 1993. From the year 2017, the college has become a constituent of Srinivas University. It is the first college which has designed the course curriculum to meet all overseas credentials. The college also has a wide network of alumni spread across the world in various managerial responsibilities. Undergraduate and postgraduate courses in physiotherapy are available on the Parneshwar campus. Srinivas College of Pharmacy was established in the year 2004, offering both under, postgraduate, diploma and doctoral courses in pharmacy. The college is located at the Valachil campus of the Srinivas Group of Colleges with picturesque surroundings. The Srinivas School of Architecture, established in 2012, with a B.Arch degree under VTU University and is a part of Srinivas Group of Colleges. The school currently offers 5 years B.Arch program, architecture studio, exhibition space, climatology lab, material museum, survey lab, model making studio, specialized classroom, and good canteen facility is the highlight of this campus. Srinivas Institute of Technology was established in 2006 with the aim of producing world-class technology graduates tuned to the demands of a fast-changing global village. SIT has gained the name as the best self-financed engineering college in Karnataka in terms of infrastructural facilities. Srinivas Institute of Technology is a unit of Srinivas Group of Colleges, Mangalore. 5 departments have been recognized as research centers by VTU to pursue Ph.D. and MSc. The institute has well qualified, committed and motivated faculty. Mangalore is known as an education hub. Many students from different places come to Mangalore just for study purposes. So just take a round around a famous private research and skill-focused university, that is Srinivas University. Srinivas University is the flagship of the 18 Srinivas group of institutions started by the A. Shamrao Foundation. It is a private charitable trust founded in 1988 by an eminent chartered accountant, Dr. A. RAGHAVENDRA. Proud son of Atka Shamrao, renowned chartered accountant and visionary academician, Dr. C.A. RAGHAVENDRA Rao started the first educational institution, that is Srinivas College of Hotel Management, in the year 1988. Today, more than 13,000 students are pursuing their higher education at Srinivas University and Srinivas Group of Institutions. Dr. A. Srinivas Rao, Vice President of A. Shamrao Foundation and Srinivas Group of Colleges, Mangalore, is an eminent personality. The university has also recognized his contribution in managing higher education institutions effectively. The group has established many milestones in the quality education in the field of healthcare, medical, dental, paramedical, allied health sciences, hotel management, engineering and technology, physiotherapy, nursing, aviation studies, port shipping and supply chain management. The university has established networking with many industries, universities and education services providers to substantially improve the quality and weightage of the course and degrees respectively. Presently, Srinivas University has 9 institutes offering innovative industry-oriented specialized courses of UG, PG and research levels. The Institute of Engineering and Technology is a constituent college under Srinivas University located at Mukka, National Highway 66, Mangalore. The college campus is developed in a lush green environment with well-qualified and experienced faculty, state-of-the-art laboratories and research facilities in the field of engineering, technology and management science as per international standards, with the aim of creating technically sound engineers and decision makers who can establish themselves successfully in the industry and global market. Robotics, artificial intelligence and machine learning, cyber security and cyber forensic, data science, cloud technology and information security and blockchain technology and distributed computing. The College of Engineering and Technology is on the threshold of a quantum leap. Institute of Allied Health Science. Srinivas University serves with medical facilities too. The college's clinical requirement is fulfilled by Srinivas Hospital, which is a 700-bedded super speciality hospital. We have separate hostels meant for boys and girls. Srinivas University Institute of Nursing was established in the year 2019 with the commencement of the BSc program under Srinivas University, located at the Srinivas University campus, Mukka. The Srinivas Hospital is also located on the same campus and offers an excellent opportunity for students to be trained in a medical college hospital. The courses are well recognized by the concerned nursing councils. The Institute of Management and Commerce, earlier named as Srinivas Institute of Management Studies at Srinivas University, is committed to providing conducive learning environment. Institute of Computer Science and Information Science is envisioned to offer courses which would build the students into enthusiastic quality professionals in the field of computer science and information technology. Since 2005, The college is associated with several MNCs like Infosys Technology, Wipro, IBM, Accenture Technology, TCS, Tech Mahindra, Dia Systems, Spanios Technology, and many other in-placement activities. All-out undergraduate and postgraduate students get good placement through College Placement Program. Institute of Aviation is a constituent of Srinivas University. The college encourages the study of airlines, airports, and business related to the aerospace industry. This also includes supervising routine operations undertaken by the ground staff of an airport or an organization in the aviation sector. Institute of Hotel Management and Tourism offers a Bachelor of Hotel Management and Catering Technology and a BSc Hotel. These programs are focused on the aspect to create professionals who would be the future leaders of the hospitality industry. Practical exposure and experienced students will get from working in Hotel Srinivas and Hotel Saffron. Srinivas Institute of Physiotherapy was founded in the year 1993. From the year 2017, the college has become a constituent of Srinivas University. It is the first college which has designed the course curriculum to meet all overseas credentials. The college also has a wide network of alumni spread across the world in various managerial responsibilities. Undergraduate and postgraduate courses in physiotherapy are available on the Pandeshwar campus. Srinivas College of Pharmacy was established in the year 2004, offering both under, postgraduate, diploma and doctoral courses in pharmacy. The college is located at the Valachil campus of the Srinivas Group of Colleges with picturesque surroundings. The Srinivas School of Architecture, established in 2012 with a B.Arch degree under VTU University and is a part of Srinivas Group of Colleges. The school currently offers 5 years B.Arch program, Architecture Studio, Exhibition Space, Climatology Lab, Material Museum, Survey Lab, Model Making Studio, Specialized Classroom, and good canteen facility is the highlight of this campus. Srinivas Institute of Technology was established in 2006 with the aim of producing world-class technology graduates tuned to the demands of a fast-changing global village. SIT has gained the name as the best self-financed engineering college in Karnataka in terms of infrastructural facilities. Srinivas Institute of Technology is a unit of Srinivas Group of Colleges, Mangalore. 5 departments have been recognized as research centers by VTU to pursue Ph.D. and MSc. The institute has well-qualified, committed and motivated faculty. Mangalore is known as an education hub. Many students from different places come to Mangalore just for study purposes. So just take a round around a famous private research and skill-focused university, that is Srinivas University. Srinivas University is the flagship of the 18 Srinivas Group of institutions started by the A. Shamrao Foundation. It is a private charitable trust founded in 1988 by an eminent chartered accountant, Dr. A. RAGHAVENDRA. proud son of Atka Shamra, renowned chartered accountant and visionary academician, Dr. C.A. Raghavendra Rao, started the first educational institution, that is Srinivas College of Hotel Management, in the year 1988. Today, more than 13,000 students are pursuing their higher education at Srinivas University and Srinivas Group of Institutions. Dr. A. Srinivas Rao, Vice President of A. Shamrao Foundation and Srinivas Group of Colleges, Mangalore, is an eminent personality. The university has also recognized his contribution in managing higher education institutions effectively. The group has established many milestones in the quality education in the field of healthcare, medical, dental, paramedical, allied health sciences, hotel management, engineering and technology, physiotherapy, nursing, aviation studies, port shipping and supply chain management. The university has established networking with many industries, universities and education services providers to substantially improve the quality and weightage of the course and degrees respectively. Presently, Srinivas University has 9 institutes offering innovative industry-oriented specialized courses of UG, PG and research levels. The Institute of Engineering and Technology is a constituent college under Srinivas University located at Mukka, National Highway 66, Mangalore. The college campus is developed in a lush green environment with well-qualified and experienced faculty, state-of-the-art laboratories and research facilities in the field of engineering, technology and management science as per international standards, with the aim of creating technically sound engineers and decision makers who can establish themselves successfully in the industry and global market. Robotics, artificial intelligence and machine learning, cyber security and cyber forensic, data science, cloud technology and information security and blockchain technology and distributed computing. The College of Engineering and Technology is on the threshold of a quantum leap. Institute of Allied Health Science. Srinivas University serves with medical facilities too. The college's clinical requirement is fulfilled by Srinivas Hospital, which is a 700-bedded super specialty hospital. We have separate hostels meant for boys and girls. Srinivas University Institute of Nursing was established in the year 2019 with the commencement of the BSc program under Srinivas University, located at the Srinivas University campus, Mukka. The Srinivas Hospital is also located on the same campus and offers an excellent opportunity for students to be trained in a medical college hospital. The courses are well recognized by the concerned nursing councils. The Institute of Management and Commerce, earlier named as Srinivas Institute of Management Studies at Srinivas University, is committed to providing conducive learning environment. Institute of Computer Science and Information Science is envisioned to offer courses which would build the students into enthusiastic quality professionals in the field of computer science and information technology. Since 2005, the college is associated with several MNCs like Infosys Technology, Wipro, IBM, Accenture Technology, TCS, Tech Mahindra, Dia Systems, Spanios Technology, and many other in-placement activities. All-out undergraduate and postgraduate students get good placement through College Placement Program. Institute of Aviation is a constituent of Srinivas University. The college encourages the study of airlines, airports, and business related to the aerospace industry. This also includes supervising routine operations undertaken by the ground staff of an airport or an organization in the aviation sector. Institute of Hotel Management and Tourism offers a Bachelor of Hotel Management and Catering Technology and a BSc Hotel. These programs are focused on the aspect to create professionals who would be the future leaders of the hospitality industry.
See all the audience, please come front. Very nice session.
Good afternoon. On the outset, I would like to thank PAPINENI for organizing such a beautiful program for today's. And her background has disappeared. Anyway, it's a wonderful session for today's. I would invite all the panelists. Today the topic is about cancer itself. I will not go into great depths. Next slide. Coming to the biology per se, to Shyam Prasad sir. Initially we thought in the 1950s it was thought because of vessel genes were important. Then as of now we know it's microenvironment is also environment where immunotherapy plays an important role. Head and neck cancers, immunotherapy plays very important. So in this changing era, what are the roles of epigenetics? I know epigenetics, we have a couple of drugs, hypoimmune agents in AML stuff. What is the role of mitochondrial DNA and what is the role of RNA sequencing and proteomics? I think Erpo is doing a big job in proteomics. So over to you, Singh. Mike, I think is.
Good evening. First of all, I would like to thank Sucheta madam for the introduction and pushing me to a very uncomfortable position of being a part of a panel discussion. So I was telling Dwarkanath when I was doing my MSc at Mangalore University. So in the second year I had a question on what is DNA methylation. So last 22 years I am working in the field of DNA methylation. Started with my Ph.D. where we try to understand what are the DNA methylation regulated genes in cervical cancer. Now moving forward, we are looking into microRNAs and histone modifications. So if you see any diseases, especially in the context of cancer, both the genetic and epigenetic changes are very, very important. So both are, one determines the other. So we cannot tell whether it is the genetic changes which drives epigenetics or epigenetic changes in turn is responsible for genetic changes like mutations or deletions or copy number variations or whatever it is. And when like sir said when in 1950s and all, so to understand the concept of gene and environment interaction, people coined the term epigenetics to say something beyond genetics. So later the research in the field of epigenetics is led to the discoveries which is so huge, which not only operates inside the cell but also even outside the cell. For example, you have two types of genes, for example, like a tumor suppressor genes and oncogenes. And if you see epigenetic modifications, all these epigenetic modifications can also be considered under these two categories. Like, for example, you have hypermethylation and hypomethylation when it comes to DNA methylation. Where hypermethylation means the tumor suppressor genes are generally silent because of DNA hypermethylation, while hypomethylation contributes to the activation of proton proteins into the oncogenes. Similarly, we have tumor suppressive microRNAs and oncogenic microRNAs and other non-coding RNAs. And considering histone modifications, again which comes under epigenetic modifications, this also can be considered under two categories: tumor suppressive histone marks and oncogenic histone marks. So all these modifications have a significant impact on the biology. So anything which you see as a cell in terms of its growth, proliferation, migration, invasion, and the connected signaling pathway, most of them are in fact driven by these epigenetic modifications. So, and besides, these epigenetic modifications can be easily influenced by the environment. That's the most important thing, and it is reversible by using inhibitors for the proteins which are connected with these epigenetic modifications. It's a all new field of research where we call them as epigenetic therapy, which is coming up in a big way. So epigenetics is very important in the context of any cancer cell with respect to biology, with respect to behavior, anything you take epigenetics plays an important role. And coming to the context of mitochondria, so we know that in the human cell, we have two types of DNA. One is nuclear DNA, and the other one comes from mitochondria. And initially, mitochondria DNA was not given importance, and it was connected with only certain disorders which we call as mitochondrial disorders. So if you take human DNA, mitochondrial DNA, we have about 16.5 18 sites, and it codes for about 37 proteins. And now, many studies, including our own studies, we show that mitochondrial mutations or mitochondrial sequence variations are very critical in the development and progression of cancer. For example, our paper is the first paper where we sequence the entire mitochondrial genome in both cervical cancer as well as congenital cancer. And we identified a lot of mutations which has a good ability in a translation area with respect to the cervical cancer. And about RNA sequencing and proteomics, if you see the diseases, there are two categories of diseases. One is monogenic disorders, which follow Mendelian pattern of inheritance, where we know it's a single gene disorder. But when it comes to a multi-gene disorders and multifaceted disorders, where you require the information of lot of genes, so that's where the importance of this next generation sequencing, like RNA sequencing, small RNA sequencing and proteomics comes into picture. Because of this, we are able to identify a lot of markers and which is currently available in the public domain. For example, TCGA, a big effort in TCGA has deposited so much data, but sadly there is very limited studies on follow-up, like how we can use this data for subsequent analysis and identifying markers. And many groups, including ours, we actually reanalyze this data. We identified many markers and which we are currently working. So this next generation sequencing, including proteomics, I think they are going to a lot of developments which are happening, and we are going to get newer and newer technology in future which will identify biomarkers and especially for diagnosis, early diagnosis, treatment in a bigger way. Thank you.
Thank you, sir, for a wonderful talk. The thing in epigenetics, what we commonly see is it's
used in diagnosis. For example, Delphi is a test which is a blood test for lung cancer. In practice, what we say, patients will say, I don't want to undergo a CT scan. So there's a scan called a blood test where circulating DNA is genetic modification is seen. So that is commonly used by us for that. Whereas RNA sequencing and proteomics in oncology we commonly use when we can't, some of these, for example, if you have large mutations which may sometimes be missed in our NGS, that is where we say we need a proteomics so that we are not missing. Some of these deletions can be missed on a DNA sequencing. So those ways we always say, okay, get this. For example, ALK, commonly say ALK, and sometimes we miss. Most of your tests can miss it. If you miss the conventional test, FISH, INC, if you missed it, and DNA NGS will miss the ALK for sure. So that is where patient will be saying, I will want another costlier test, and that is where our proteomics come into the picture. For diagnosis, we have this, and these are useful for diagnosis and this. You are an expert in all of these, that's why these questions were framed for you. I don't clear your data, and that is how these questions are framed. Next slide. To Thakur sir, then I will go to Satish Mandari sir. It's simple. Sir, we have come a long way from conventional diagnosis. For example, just doing open biopsy and stuff. What is your role of saliva in diagnosing head and neck cancer? So, meaning, what are the newer techniques there? Things have gone a long way. For example, some of them just do a HPV testing. Some of them do a 19 marker and stuff like that. So, what is newer things in diagnosis of head and neck cancers is what I just want to ask. For example, we have tests, non-invasive tests, put other slides also. For urine, for prostate cancer, we have those tests. For bladder also we can pick up. Cervical cancer also can be picked up by a urine test. This is where the indication is basically when a patient undergoes a CTRP, like Shivraj Kumar underwent CTRP, and he says, I don't want to do a cystoscopy again. So we do a urine analysis to see whether the tumor is there or not. How sensitivity, because patient will see online and say, if I don't want cystoscopy every 3 months or 2 years, this is a simple test for them. They will do the urine analysis, see the tumor and give. So similarly, head and neck cancer patients, they may say, I don't want to undergo a scopy again. What are the non-invasive tests?
Thank you, Guru sir. Actually, I am not part of the panel.
You are still an expert, sir. You are our teacher.
Lectures, I was a part of the presentation by a friend from US. He talked about the prostate cancer, about the role of urine. I felt it is really amazing. So as you said, if we had some tests like that in the oral cavity, it would have been wonderful. Unfortunately, in head and neck cancer, particularly oropharyngeal cancer, I think role of HPV as the expert you are all of us aware, but unfortunately the salivary markers, I think according to my knowledge, it's still in the infancy and in the investigation stage. We have done some projects in our university, like estimation of these competent cells in the salivary fluid and diagnosing the cancer, pre-cancerous conditions. So unfortunately, at present, as far as my knowledge goes, we don't have any diagnostic tests. But HPV, as you know, it has become part of our staging also in oropharyngeal cancer. We have done a lot of study in our Indian situations. But what we see in the Western countries, we don't see in India. The number of HPV positive oropharyngeal cancer in India, it's relatively very less. So, you know, today morning also we had a clear discussion. Even TNM staging, nodal staging has completely changed in the case of HPV positive oropharyngeal cancer because it is radio sensitive and they remarkably do well radiation. And anybody can enlighten us regarding the saliva and very happy.
Oral cancer, but kind of like ahead of time, last phase 3 has been going on for what they do is by gargling near infrared molecular gargling, you can actually visualize by either light, cancerous, precancerous stuff. And then you also have OCT as well, which can be used for non-invasive intervention. I think it's not very popular, but the screening is there going on many places, many parts. We are going to go back into practice. But now I think in Bangalore there is some program there.
Right now we have one test, Cologuard, for colon cancer, but it is not very sensitive. If there is an adenoma, it is very sensitive. But if the patient doesn't have an adenoma, the CDC, the blood is not that sensitive. So this is one test which can pick up colon. So if the patient doesn't want to repeat it for a patient with a family history of colon cancer, meaning MSI syndrome, we can do this CDC, simple as that. But they should have an adenoma at least. So that is one test which we have. Other than that, I think sir has already highlighted the urine and the genitourinary cancers which can be picked up. It's a big basket of tests, the basket of cancer which can be picked up. The problem is always the sensitivity. What to do if it comes positive, what to do if it comes negative is always been the problem. So we don't have a biopsy to put it. Next slide. Rao sir. External beam RT. Anyone is an expert in RT? Sir? So first thing is what are the advantages in RT what we have seen? And since sir, okay, I'll get the question itself, sir. So what are the best nanoparticle sensitizers we have in clinical trial, whether it's gold, hafnium, iron, what are the best which we have in practice? Because in practice, finally we end up with cisplatin. That's the only thing which we end up with, cisplatin or cetuximab or something like that. We don't have any of these which we commonly use because this prevents the normal cell from getting the problems.
Actually, some of our collaborators have with bone nanoparticles.
Yes, I
for all the in US, electron radiosensitization, including analysis. One of the reasoning is of the uniformity in size, and but there are a lot of caveats. So once the trial went in from the end, lately there are more with protein, such as targeted bone nanoparticles, for again radiosensitization. I think if you go and look
I've seen
on the clinical trial database, you can see multiple bone nanoparticles.
The problem is in Indian scenario because head and neck cancer is so common here. It's almost 20 to 30% of all our cancer in the world, and the field of radiation is so vast. So the side effects is also pretty vast. They're not able to eat. It's not like rest they can walk in and go out. Head and neck cancers, eating, speaking, a lot of things, mouth opening, all the problems. So that is where all this radiosensitizer only to that tumor and not the rest of the area. It makes a huge difference for us. So we can make something because for us platinum is the same platinum. If we say for other people platinum means platinum, for us the platinum means platinum, nothing else. So that is how the difference. If we say gold, for a normal person gold will be something else, but for a normal person it's something else.
The drawback so far is the amount of gold in one particular case is one of the things which I think probably in future it can become the cell uptake enough to provide you the
It's a lot of wonderful work you're doing in this series, and thank you. Anyone in the audience has any doubts? You can ask. Right. Next slide. Yes, sir. May I go on it, sir?
Where you mentioned that there's no use case of NAP, or the widely used in the clinical practice here, we have the NAP vaccine.
Yes, sir.
It's widely used. It's also, albumin and nano together treatment.
Those are currently
used, but nobody has combined that with radiation. Of course, it's not intended to be used together with radiation, but we do, I think, have quite a few. Especially, I think has done done reasonably well in a few cases. But the results probably are not phenomenal, but I think certainly it's much better than using the backgrounds.
I'll give a question to you directly, sir. Theranostics, therapy and diagnostics. So anyone can take, but since you're having the mic, I'll ask you the question itself. So how common do we see cyclotrons in India where we can produce multiple, because in the era when we studied there was only one FPG bed, FPG. Now with things have changed, that FFPI, if you ask anyone, they know only FFPI. So a lot of things have changed because of availability of cyclotron. So what are the things which you see in the future where with the availability of cyclotron, a lot of new things will pop up. For example, ER positive PET scans, HER2 positive, trastuzumab positive, or HER2 positive PET scans. So a lot of scans are there. Before it was one scan for everyone. Now we have going to an era where HPV positive head and neck cancers. So you can just do a scan, see the tumor heterogeneity and decide whether all the cells are HPV positive or some cells, the primary is positive, the metastasis is not positive. So cyclotron has changed a lot of things. So what is your look into the future?
My guess is over the years, the FPG will probably lose the seed or take the backmost seed. You will have more and more lesion-specific PETs coming up, PET scan coming up.
Like PSMA.
That's not only for the, that's not driven by cyclotron, that's basically driven by the chemistry. It's actually the chemistry, because if you want to couple any of these markers and with the radionuclide, the synthesis is probably what is very critical. The moment we are able to synthesize with using click and other chemistry and our ability to couple the the bombard with the cyclotron, I think it will be fine. But yes, to a certain extent, one certainly should move from small energy, what we call as baby cyclotrons, which is widely used for the FPG. We need to move into intermediate energy cyclotrons, which is happening, both in terms of affordability, practicality, and running. And Rob knows, I'm not from the field of core nuclear medicine, but my tenure at the Nuclear Medicine Institute for 2 decades, and working closely with a friend of mine, I guarantee you in a few years from now, you will have multifold increase in the lesion-specific FDG probes available. Okay? Back in Delhi, more than 10 years ago, in IMAS where I work, we made, I think, about 5-6 probes for the scan, other than FDG. Okay? And as you said, PMSA is coming up. There are a number of probes that are available. I would tend to think in less than a couple of years, we will have more FDG scans which are specific to organ and lesion specific. That's not too far.
Yeah, it's
going to happen in India too.
You want to add something, sir? From 1940 to 2000, we had only one ID-131. From 2000 to 2010, we had two probably, radium and strontium samarium. Then they have gone, now 2010 to 2020, another 2-3 drugs have got 2020 onward, just boom. So it's a big field by itself. So we don't have an MD in this line, meaning what we have is all general, but in this line person, we don't have a specialized person. So we need more and more people in this line. So you want to add something?
Yeah, I mean, what you said is correct. We are also looking at the fundamental basic biology and looking at what are the new targets that will provide us a definite way to diagnose and convert that to images. So more and more information we are gaining from that cancer biology field, we are trying to, kind of develop diagnostic agents based on that. All at the end is to fulfill the need for clinical diagnosis, let's say. On the other hand, more and more targeted therapeutics, right? All the biological and monoclonal antibodies, things like that, which are not going to be useful for all patients. So we need a diagnostic tool, imaging tool to select patients for that therapy. Again, and people like me, radiopharmaceuticals are always looking for business. They're always looking for ways to build their research careers, so we're going to bring up more and more compounds. But ultimately, it's not going to, not every compound is going to make it to the clinic, and we need to get clinician support, saying that yes, there is a clinical need that needs to be useful for, helping with diagnosis or treatment. So that's on the diagnosis. On the therapy side, that's where the big interest is. Of course, that's where the big interest is. So more and more therapeutics are going to come out this way. And because of the cost of implementing those therapies, and also the difficulty of getting access to those therapies, again, diagnostic imaging would be, a valuable tool to select right patients for that therapy. So the cost of diagnostic imaging is relatively small compared to one dose of therapy with radiopharmaceuticals, for instance. So definitely you'll see more and more radiopharmaceuticals, and we have to learn that information. But right now, the field is also focused on where there is a large unmet need, trying to find new targets. And then if those targets are like right candidates for developing therapeutic radiopharmaceuticals, that's what we are trying to develop diagnosis for a lot of similar targets. Definitely in the targets of prostate cancer, they are moving from beta inhibitors to alpha inhibitor therapies, or a combination of beta plus alpha, sequential therapies. So lots of things happening on the preclinical side. But also for diseases like triple breast cancer, where there is no proper treatment, or targets to bring additional imaging plus therapeutic agents, those sorts of things.
In Mangalore, I think KSET has a very good nuclear medicine department, and they have all these scans which we speak about. Rotanak is there. You ask them, they have. So, sir, that's the show here.
Sir.
1 minute. Okay, madam. Time's up.
A couple of minutes.
Yeah, okay.
Thank you.
Next slide. And last 2 slides. So, diet and cancer. Say one answer. Diet and cancer. If madam wants to answer. Sir is also there. I think you are the expert in this. Mike is not right.
So already we have discussed about the main role of fatty acid, especially the medium chain fatty acids and plus short chain fatty acids. Actually, most of these cancers, there are so many various toxic molecules or free radicals, we say. They are the main culprit to develop various mutations, various acceleration of various upregulation of the molecules. So the prevention is better than cure. So all of us has to try to prevent developing cancer. All the scientists on this dais, they explain after cancer what are the best treatment, best way overcome with the various methods of chemotherapy, radiotherapy, immunotherapy and nanotherapy, so many things. But my consideration is now prevention is very important. For that reason, the antioxidant status are very important, and short chain, medium chain fatty acid. For example, fat, long chain fat, obesity is not the ghee or the short chain fatty acid. It is because of the carbohydrates. So all the carbohydrates are accelerate the long chain fatty acid synthesis and increase the obesity. The obesity is the culprit for increasing estrogen and insulin. So insulin growth factors, all proliferation of cells. So this leads to inflammations and then leads to various types of cancers, anything we name. So melanoma, multiple melanoma, or the kidney cancer, all this, because the culprit is free radicals. So at the end, all of us are taking a oath, prevention is better than cure. So control your diet, your lifestyle, your exercise, maintain the weight, and you should prevent the obesity. Thank you, sir.
Thanks, sir. Any last concluding remarks, sir? And that's it.
regarding the antioxidants and the cancer war. I think space medicine also is interested in that one. So one time we had a study for about 2 years or so in 20 different locations. They found out that somehow blueberries and blackberries fed mice will have a little bit of better response to the treatment from the cancer therapy. So that was the interesting one. So when it comes to the space radiation, maybe it's a good idea to transport along with human beings these blueberries and blackberries around with them. The challenge is going to be how can we physically transport them around the field. Maybe we can transport one extra person than these tons of blueberries. Another interesting point that on the ground-based experiments, as the suggestion is that if we have this persistent flow mice, blueberry extract from these mice, the response for the cancer treatment and also the effectiveness of this radiation treatment is more possible. And maybe some of those things can be used in human beings as well. So that's my comment.
Let me just add one more. This early morning you saw Dr. Shahid Umar gave a talk. I think it would be nice if I can wrap this particular part, the article. Please check out Umar, PAPINENI and RANA paper on about diet for the microbiome, which is critical both in radiation protection, radiation injury, etc. So we have a very nice paper from 2014 about the 8 or 10 items which we can eat in India. Of course, curcumin we have, broccoli, etc. Add those and we can live probably 10 more extra years. Check it out, this paper, again, Umar, PAPINENI, RANA, GREINER, classic paper of different food items to promote better microbiome, especially before cancer, post-cancer, prevention of cancer. Thank you.
To add to PAPINENI what he has said, we have done a study on pomegranate, which is funded by PR in this project. We have found it useful in prevention of mucositis, but in radiation-induced mucositis. The trial is going on in our center. We are using pomegranate extract for protecting the normal cells from cancer.
Nice. Brenda madam, all over the you, madam. Chita madam and Brenda madam, all over to you. Thank you, my panelists, for all this.
Thank you, sir. As we arrive to the concluding movement of this remarkable journey, Simsic 2K25, I request Dr. Brinda J. Bhatt, Dr. Jayashree Bola, Dr. Praveen B.M., Dr. Naveen Bapalige, Dr. Edwin Dias, Professor Pavana Krishnamurthy, Dr. Mithun S. Acharya, and Dr. Prashant Prabhu to join our dignitaries onto the dais. Dr. Shabaraya to also join the dignitaries onto the dais. It is time to reflect, to celebrate, and to express our deepest appreciation to every mind that contributed to making this academic feast a grand success. We have amongst us the chief guest of today's program, valedictory program, Dr. Satish Kumar Bandari, former Vice Chancellor, Nitti University, Vice President ISR and CRL Nitti University. Simsic 2K25 has been a vibrant platform of knowledge exchange, research inspiration, global networking, and a celebration of scientific spirit. Over these sessions, we witnessed brilliant minds, groundbreaking ideas, thought-provoking discussions, and the powerful collaboration across borders. To begin this valedictory proceeding, I invite Dr. Vrinda J. Bhatt, Organizing Secretary, to deliver the welcome address. Over to you, ma'am.
Good evening, everyone. It is with an overwhelming heart and euphoria that I am standing here with a satisfied and an accomplished feeling for a successful international conference with the dignitaries sitting here, some of them who have already left for their hometown, and the most important ones have graced us with their presence for the valedictory function. There are no words for me to express my gratefulness for their presence here, for making this event a grand success in the true sense. So with an open heart, I offer my heartfelt welcome to Dr. Satish Bandari sir, Dr. PAPINENI sir, Dr. Saganti sir, Dr. Dwarkanath sir, Dr. Satish Chitineni sir, Dr. Shyam Prasad Square sir, and Dr. Jayashree Bolar ma'am, Dr. Guru Prasad Bhatt, Suchita ma'am, louder please. This was possible only because of her, ma'am. And then all the help rendered by her team, Dr. Mithun, Dr. Praveen, Dr. Shabaraya, Dr. Pavana, Dr. Naveen Papulige, and the wonderful food that you enjoyed for 2 days. Dr. Prasanna, Dr. Prasad, I'm so sorry. The sugar rush that I had from the coffee that I just had. I'm so sorry, Dr. Prasad. So, and all the dignitaries sitting here, Dr. Divya Rani, she is busy with the expo. So, a wonderful welcome to all the delegates here who have taken time to stay back and be with us for this valediction, and all the lovely MCs and the helping team who have done a wonderful job, spoken non-stop from yesterday morning till now. My lovely Dr. Swati, Miss Rashmi, Ranjita, Dr. Ashwini, I'm forgetting names because I'm overwhelmed. Rupa, please tell your names. I'm forgetting names. Rupa, Ritu, Situ, them. Please tell your names. Okay, all you lovely girls there. And, covering us live, Sushmita, madam, my dearest, and her team, all of you, and, cooperating us, all of you, all my lovely audience, and Dr. Anup, a special mention for, readily helping us with reporting the entire conference. Thank you, Dr. Anup. Thank you so much. Dr. Devasilan for helping us with the transportation of all our faculty and students to the Mukha campus. Thank you, Deva. Thank you so much. And welcome to all of you and all of you here. Thank you so much.
Thank you, ma'am. Your welcome made us feel extremely extremely encouraged. Now it's time for the much awaited part of the session. Yes, it is the announcement of awards. I request Dr. Ashwini, Assistant Professor, Division of Research, to read out the winners.
So good evening to the dignitaries on the dais and off the dais. It's my great pleasure to read out the names of winners of the oral poster presentation competition that was held during Simsic 2K25 conference. So I request Dr. Satish Kumar Bhandari sir to give away the prizes to the winners. So first category is oral presentation, category 1, faculty. The third prize goes to Dr. Shivani, Institution of Physiotherapy, Srinivas University. Third prize goes to another faculty, that is Dr. Vijay Kumar Palanieswamy, Institution of Physiotherapy, Srinivas University. And one more faculty, the third prize goes to Dr. Prem Kumar, Institution of Physiotherapy, Srinivas University. Director, Incubation Center, Srinivas University. And Dr. Amrit R, Allied Health Sciences, Srinivas University. And the first prize goes to Dr. Bhagyajyoti Yankar, Department of Biochemistry, Srinivas Institute of Medical Science and Research Center, Mukha. Oral presentation category 2, that is under research scholars. The third prize goes to Mr. Prashik Chavan, Enapoya Research Center, Enapoya Technical University. Mr. Prashik Chavan. Anyone else can collect? Second prize goes to Samanvita Mandal, Enapoya Research Center, Enapoya University. First prize goes to Mr. Sanjay R. Nair, Enapoya Research Center, Enapoya Next category, oral presentation, category 3, postgraduates. Third prize goes to Sirin Sara Ninan, Institute of Physiotherapy, Srinivas University. Anyone from physiotherapy can collect the certificate.
Anyone from the same college can come and collect the certificates.
Second prize goes to Mansi Chavan, Institute of Physiotherapy, Srinivas University. Science and Research Center. Second prize goes to Harshita Kumar, AG Institute of Medical Science and Research Center. First prize goes to Suhair C, KS Medical Academy, Nikhil University. Category 5 poster presentation. Third prize goes to Deepti and Thomas, Enapaya Research Center, Enapaya University. Second prize goes to Abhina Gangadhar, Enapaya University. First prize goes to Priyanka, Srinivas Institute of Medical Science and Research Center. Congratulations to all the winners of oral and poster presentation. We sincerely appreciate all the participants for their enthusiastic involvement in oral and poster presentation. Thank you.
Thank you, ma'am. And once again, congratulations to all the winners. Now, we would like to hear from our speakers. I request our esteemed speakers to share their valuable feedback on Simsic 2K25.
In the audience, please come and give some feedback. Anybody? Raghavendra sir, madam. Please come and give a good feedback. Good feedback.
Dignitaries on the dais and after the dais. I am Dr. RAGHAVENDRA, Professor of Social Sciences. Actually, I am a social science background man, and the first time I am attending all the sessions without missing, because all the sessions are very much interested. Even a social science man may be considered as a layman to this field. I can easily understand all the deliberations and the presentations, and one can easily get awareness regarding the diseases which is majorly causing more and more deaths in the human society. And I would like to request the organizers, all the presentations are good. Please request all the paper presenters through mail and get all the hard copies of the papers and please publish it. It will be a record on the, it will be keep as a record in your library. It will be record for even all the interested persons, those who are not participated in this conference also, may get use of this, because all the presentations are very, very good and more applied also. So this is my kind request to the organizers to please publish all these presentations, and thank you very much for providing me this opportunity. Thank you very much.
Anyone else?
I want to express my personal gratitude for the entire team of the organizers here, and particularly for inviting us from overseas, Dr. Suchita Kumari and the entire team here. That is a wonderful thing, and we thank all of you for providing such a wonderful hospitality for us in various areas, starting from the people on the left side announcing things and organizing chairpersons here. And all these things are a wonderful experience for us. On the same note, I will say that I have the distinct privilege of participating with wonderful scientists sitting on the stage right now, and some of them were presented by the network as well as some are gone already. And we want to thank all the speakers for choosing giving their time and taking some sacrificial steps to come all the way from so many miles away from around the world. I want to thank all us feel at home with all of you. Thank you very much.
Namaskar again, thank you, first of all. And really, when I see this huge crowd on the 2 days of success, and I look to the starting point when I had a chat with Dr. Kumari a few months ago, from where it started, where it ended. Amazing success I feel, and literally when I say the milieu of scientists and speakers who came from different parts of the globe, and what we right now need is, please spend some time, go back, look at all the videos which is nicely recorded, look at it and then grasp it and things, and each of you can, in any discipline, can utilize not just the science, but the way we think, but the way Dr. Matthew Thakur is talking, all sorts of things, these are all very critical. And be it domestic speakers or international speakers, they have a lot to say, a lot to give. And I think this is the best opportunity to get into the next round for the students. And people who are not here, please pass on the information. I think, again, like Dr. Prince again mentioned, each one of the team who's made this meeting successful, helping Dr. Srinivas Kumari to bring this out. I mean, thank you very much, wholeheartedly. And people like from transportation, amazing people. Everybody really, looks like I'm going to talk. Good friends.
I don't think I can add much more to what my dear friend Prem and Rob has said. I only will multiply 10x and say whatever they said. Into 10 is what I would have loved to say. And it's true for, as he said, the left, the front, back, and the right. An amazing person is Dr. Kumari. It was very evident from the track, the way her colleagues, down from the smallest to the biggest, interacted with her, and then kind of support she has made this stream grow bigger, stronger, and last longer. Thank you.
Thank you, sir. We all felt motivated by the words you have given to us. Thank you, really, thank you so much. It is not every day that we get to listen to someone who just don't follow the knowledge but creates a new path for it. Please join me in welcoming our Chief Guest of Valedictory Program, Dr. Satish Kumar Bhandari. Sir, I request you to address the gathering.
Good evening to each one of you gathered here. It is indeed my privilege to be part of this valedictory function. I thought the Chancellor will be there in the stage. Unfortunately, he is not here. I thought I should congratulate him for leading this very bright and very upcoming multidisciplinary university. I would have been very happy to be part of this valedictory function. Dignitaries on the dais and off the dais. It is indeed my privilege to be part of this program, even for the inaugural function over there. I saw the wonderful lectures by the invited faculty from abroad, because I know few of them very intimately. Secondly and PAPINEni, I think I had opportunity to call them, invite them to my campus earlier. We had two conferences in Nita University. I am very happy to meet them once again. I think our biggest loss is Srinivas Kumari coming to this campus. But still, I am happy. I always call her my sister, because she was a very good friend, a good colleague of mine, and she has brought a lot of enthusiasm to the campus. What you see is taking everybody together is very, very important in any campus. I always say in any university, you can't get 100% good in everything. You can't get. It's very highly impossible to get, and all 25% dedicated people is enough. And Sujata Kumar is one among them. I am very happy that I should congratulate her for organizing this conference and also be inviting me to be part of this academic feast. I have made few points. I think it's not a time to give big lecture. Already all of you are tired in the last 2 days. You had a lot of talks. But what I want to convey is the theme of the conference. I think I should congratulate the team. I think congratulate Bunda. Bunda is, I saw her eye movements and Bharatanatyam postures. She's fantastic. I think she was a great help to our Sujata Kumari. I think I should thank her also for bringing all the way. The theme of the conference is the innovation from molecule to medicine. I think it's very relevant, very truly visionary topic. I think that's how conferences start. I hope in the last 2 days everybody conveyed this message how the medicine is from the molecule goes to the medicine's bedside. I want to quote William Osler. I think all of you, particularly from the medical field, may be knowing the father of modern medicine. He said, "He who studies medicine without books sails an uncharted sea, but he who studies medicine without patient does not go to the sea at all." I think those days it was very, very relevant. That what he said, library is very important, books are very important. To learn medicine, we should have patients. Keynote address. I thought how the cancer treatment is evolving. Nowadays it's not fit for all. One particular drug for every disease. I think when we are students, we should think only few drugs. Chemotherapy, cisplatin and radiotherapy, full dose. I think we had cobalt 60 unit. But things have dramatically changed in the management of cancer. Now it has become targeted and specific to particular disease. I think even every day it's changing. Guru Prasad, I think they have to read so much. They have written so many conferences to get the latest changes what is happening. When it is personalized treatment, I think the treatment being totally personalized. I think earlier, even now, people think chemotherapy means it's gone, alopecia, I'll die. It's as good as a death result. But the things have dramatically changed. I think because a lot of things were discussed in the last 2 days, and that is one carry-home message. So, as you all know, what COVID taught us, I think molecule to medicine, I think now, earlier, to bring one particular molecule as a drug or treatment, it had to go to so many rigorous, time-consuming procedures. I think the things have changed. What you saw during COVID, I think in 8 months, 10 months, you could vaccine. Thanks to the new innovations, what's happening, technological changes are happening, revolutions are happening in this particular area. I think this particular molecule to medicine is dramatically changed. I think that we could absorb in the last 2 days. I think this is one of the another carry-over message. And what I see is when we were students, medical students, I think publication was not at all our cup of tea. We never bothered publication, documentation. I think now the things have dramatically changed. Doctors have to publish or perish. I think that is a very important message from particularly this conference, what we can carry. And you may be knowing that Government of India has started recognizing importance of innovation, entrepreneurship and intellectual property. I think Atmanirbhar Bharat, what you saw during our Operation Sindhu, I think how our army, all the 3 services played extremely well to win that war. I think things have dramatically changed. I think even the medicine, the 21st century belongs to India, according to me. I think all of you should feel proud that all of you should dedicate ourselves to get into that, what our Prime Minister says. Then, very, very important thing is, even our NIRF ranking and our NAC, 50% of the score goes for research innovation. So I think nobody can have, even Srinivas or university, want to climb this ladder, getting into good NIRF ranking or NAC A+, or now we call it grade 5 or something like that, the changed NAC grading. But we should be good in research. Research, publication, innovation, entrepreneurship. I think there is the buzzwords in the campus. I think the Chancellor also in his speech said how they want to give trust to the research part in the university, and I think definitely it is going to happen. Probably this type of international conference. I think in 2 days you can't discuss multi-omics. I think what Guru Prasad was asking about how the multi-omics, genomics, proteomics are helping the research part. I think that is very, very important, and that cannot be learned in 2 days. You could get some good ideas, talking to your peers and your other experts in the particular area. Take it home. I think particularly your PhDs, your students, this should be carried and this should be made in a concept note and should be taken to the next level. I think that publication Sakayati was telling about, I think conference proceedings will naturally come out. But out of this conference proceeding, how much will turn out to be a good impact fact or Q1, Q3 or publication, very, very difficult. But definitely you could get a lot of ideas. I think this should be carried to the next level. I hope this conference is very, very successful, and all the delegates will carry very pleasant memories of this conference, both academically and socially. I think I understand that a good cultural event on the first day. I think it was wonderful. I hope and I wish the university, Srinivas University, greater heights in the future and many more such conferences, national international conferences. Wish you all the best. May God bless you.
Thank you, sir. Your guidance is truly valuable. Gratitude is the heart of every celebration. To extend the formal vote of thanks, I invite Dr. Sujita Kumari, the organizing chairperson.
Thank you. Good afternoon, everyone. My most respected dignitaries on the dais and all the dignitaries of the dais, delegates, all my dear friends. This is my pleasure to stand before you at the end of the day. I always, even in so many conferences, end of the day, I'll take this chance to say thank you to everyone. First of all, I thank God. Without any difficulties, God is given every time to bless us. So we thank God that for the success of conference, and the first and foremost is some Chancellor, Pro-Chancellor, trustees and all registrar and whole university team. When I thought of having a conference after discussing with the Dr. PAPINENI, because the span of time is only 2 months, so many things we have to arrange without hesitating. That they never been asked how many will register, what is the fund support, nothing. Without thinking, they have shown all the generosity to develop, to give hospitality, logistics, food, the essential food, everything till evening. They have given all the opportunities to conduct this conference. We are really, on behalf of everybody, will thank the university and Chancellor, Pro-Chancellor, everyone. Thank you, sir. We pray God to bless you for best in future. And the second very, very important thing is I'm thanking all 21 speakers and 42 chairpersons and judges. Without changing one single minute, single talk, all of them present in time, and they have did their work beautifully. A big applause for everyone especially our dean. Never comes front, but I made the dean and whole medical college to come and participate. When we start first step, I know Vrunda madam. When I met her, she said, I'll support for everything, don't worry. Then she introduced Guru Prasad sir, Sangeeta madam. Then Guru Prasad said, I'll take care of everything, madam, don't worry. So till evening he is with us. Like that, our journey slowly, step by step. And PAPINENI, whole night he will call, you do this, you do that, do that. Sometimes at home we had one great function also. One day my mother also sick, and second PAPINENI phone, and all delegates contacting. So whole 2 months my brain was completely packed. In between, sometimes I off also. So something we made very dreamless success. So that is why I thank each and everyone, even the medical manager, everybody round the clock helped us for the success of this conference. So now Edwin sir also, everybody, Edwin sir is here. So next I extend my thanks or appreciation for all the delegates who have participated, because most of those delegates from various universities have participated. It's a pleasure. So many university people are participated. So we are very grateful to each and everyone. I also extend my heartfelt thanks to the Dr. Subramaniam, but who is the dean of this building, this computer science. He gracefully gave all the required logistics without hesitation. We are grateful to him. Then I wanted to extend my gratitude, special acknowledgement to our Dr. Praveen. He was our director of research and administration of all that. His exceptional support in hospitality, transport, for everything we are calling Praveen do. So you may not find him here, but behind the scene, he did everything, every support. So on behalf of university and conference organizing committee, we just have to have a felicitation. Come sir, please come, please don't waste time. Come, there are a lot of things. Please come. Sir, now be ready. Come sir, please. Please, it's a hard work throughout 2 months, running around 100 times to university office and college and everything. We are very grateful to Praveen. Next we are calling, be there sir, Naveen Bappalige, please come front. He is HOD of physics and also he is taking the IPR cell. Please stand. Come here. We wanted to felicitate you for your thoughtful and assistant, hospitality, transport, everything. Without hesitation, he has given all the support to completion of the essays first. And we from the university office, we want to thank Jayashree madam. Please come front, madam. Jayashree madam, please. She has given all the support of completion of the Krishna Kumar. Devashilan, come. Thank you, thank you, thank you. And then Vrunda Madam and team, all the MC, Swati, Rashmi, please come on the stage. Swati, Rashmi, come fast. And Ranjita, Rupa, Ashwini, along with them, come please. Come. Ranjita, Ashwini, Ranjita, Rupa. Ranjita, come first. Here, madam, here. And this is a pleasure to export team. Madam Divya Rani, please come to the stage. Divya Rani, madam, and Dr. Pradeep, who organized the impressive exhibition here. And within a span of 9 days, madam arranged, Divya Rani and Pradeep arranged around 50 participants in the expo, and she is our director for international collaborations. And Pradeep is our professor in computer science and also taking charge. If your madam call the team, Dr. Sushmita is coming. Sushmita, come madam. Sushmita. Ramo, come fast. So, Ramo. Sushmita. Anu,
also here. So we have a collaboration, the first collaboration of institute and industry also we saw in this event. So I would like to call Solar Active Farmer who were actively participated in the last 2 days in the expo. Kindly come forward and receive your certificate. Kotak Mahindra, next. Quadrilant Labs. So all export team members kindly come forward. Quadrilant Labs. Dream Holidays, some of them have already left, but whoever are here, please come forward. Ecolab Tech Solutions Private Limited, who's been continuously supporting us, he is here. Ecolab Tech Solutions, yes, this is Inficicart, Kanchana Automobiles, kindly come forward quickly. Kanchana Automobiles, Edelweiss Life, Kanchana Automobiles, please come forward. Girija Surgicals, please come forward. You have wonderfully displayed all your products. Vismaya Center, ma'am, she is here all the way from Mysore. She has come here. Bonima, SKF Elixir, who have not only set up their stalls but also they have given a free coolers, hot and cold coolers for the 2 days. Mazidov Clinic, Mazidov Clinics, Mazidov Clinical Innovation, Zupaloop Solutions, Zupaloop Private Limited, Life Essential Water. So they all have left, but it's been wonderful collaborating with you all in the event. We look forward for you in the future as well. Thank you.
Thank you very much. We'll all stand for the national anthem.